So good morning, everyone. Thank you so much for being here. Welcome to the Cholangiocarcinoma Foundation's New York City Regional Symposium. My name is Kristen Spencer. I'm a medical oncologist here at NYU, and the NYU Perlmutter Cancer Center is honored to be hosting, and I am really truly honored to be chairing today's first annual meeting. And I want to thank each of you for being here. We have patients, caregivers, advocates, clinicians, researchers, and partners. Cholangiocarcinoma, as we all know, is a rare cancer, but as everyone in this room knows, its impact is anything but small. And yet, what defines this field is not only the difficulty of the disease, but the remarkable collaboration that has grown around it, and that's really what today's meeting is all about. The Cholangiocarcinoma Foundation has played a transformative role in this space. They've brought together patients, advocates, and the scientific community in a way that has accelerated awareness, research, and clinical trial development. Meetings like this one are powerful because they sit at the intersection of science and lived experience, and because they remind us that progress happens fastest when we learn from one another. For our patients and caregivers in the audience, I want to say to you clearly, you are essential partners in this work. Your presence here strengthens this entire community. For the clinicians and researchers, today is an opportunity to step outside our silos, to hear what's happening across institutions, to share lessons that we've learned, and to think creatively about how we can continue to move the field forward together. The program today is divided into two parts. So this morning, we will hear from the Cholangiocarcinoma Foundation. We will examine novel liver-directed approaches to management, and we will hear from one of our very own patients. This will be followed by, most importantly, a lunch break from 12 to 1. And in the second half of the day, we will cover systemic therapies, including emerging treatment options, as well as how nutrition supports our patients through these treatments. Most importantly, bathrooms located out these doors wrap around to your right, and you'll find them on either side there. While you're up, please be sure to stop by and visit our symposium sponsors, without which we would not be able to host today's program. Today's symposium is designed to be practical, forward-looking, and inclusive, and my hope is that today is not only informative but energizing, and that all of you leave with new knowledge, new connections, and renewed momentum. Thank you again for being here. With that, it's my pleasure to get us started by introducing Melinda Bikini from the Cholangiocarcinoma Foundation. She's a 16-year survivor of cholangiocarcinoma who was treated at the National Cancer Institute through adoptive cell therapy clinical trial. And drawing on her lived experience, she is a dedicated patient advocate, serving on national cancer research and guideline committees, and she shares her story to support patients considering clinical trials. And she's also a very, very dear friend. So please welcome me in joining her today. No problem.
I'm gonna make sure I know what I'm doing real quick. No, I don't. Good morning, everyone. Welcome. I'm so glad that you guys ventured out to come and participate today. I know everything's getting a little crazy with this big storm warning on the way, and I have to get all the way back to Montana. So glad that you guys made it and are here. I am Melinda Bikini, Chief Patient Officer with the Cholangiocarcinoma Foundation, and also a 16-year survivor of stage 4 cholangiocarcinoma. Again, I want to thank Dr. Kristen Spencer, who partnered with us and NYU to host this first New York City Symposium. Hopefully this is something we can continue every year and serve the patients and caregivers in this area. And also we want to acknowledge our supporters, without whom we could not do these events. So AstraZeneca, Incyte, Jazz, Natera, Merck, Servier, and Partner Therapeutics, thank you for helping us to sponsor these regional symposium events. I also wanted to let you guys know about the CCF staff that are present here today. We have Mel, Emma, Elizabeth, Amanda, and myself. So please connect with us throughout the day. If you have any questions about the foundation or anything we can do to support you, please let us know. I'm going to share a little bit about my story real quick, and then I'll talk a little bit more about the foundation on the way. So 2009 for me started out as a very amazing year. My husband Steve and I got married in March. We blended our families after 5 years of a lot of ups and downs. We married on the beach in Hawaii and then came home to celebrate as a family in July. Little did I know what was going on inside my body. I was working as a paramedic and I absolutely loved my job. Steve and I had both started nursing school and had planned to be traveling nurses when the kids grew up. That year I started experiencing right shoulder pain, some fatigue, heartburn, and some GI issues. But I carried a pack and monitor on my shoulder. I worked 24-hour shifts and was a mother to 6, so I thought I should be tired. And we didn't always eat at the best time of the day or tended to eat fast food and drink a lot of caffeine, so it made sense that I had GI issues. It wasn't until Steve and I were attending a critical care nursing course when I had severe upper right quadrant pain, and the docs there thought I might have gallstones. When I returned home, I had an appointment already scheduled for my normal yearly checkup, but I mentioned to my primary care doc that I might have gallstones, and I remember because my mom had them and my older sister had them, and thankfully he sent me for an ultrasound right away, which found a large mass in my liver, which led to many tests, ultimately the day I will never forget. Like many of you, those 3 words, you have cancer. I was 41 years old, a mother of 6, and there was no way I could have cancer, yet I did. Initially, I was diagnosed with cancer of unknown primary, while they ruled out any other primary cancer. The most important thing about December 1st, 2009 was it was my son Skylar's 14th birthday. I could have never imagined the effect my diagnosis would have on him at the time. I remember taking him and his sisters to dinner that day and then to the movies, and then them not knowing yet about my cancer diagnosis, and my husband and I holding hands in the dark during the movie crying. We were told it was most likely intrahepatic cholangiocarcinoma, and I just remember my husband and I were like, cholangio what? We had never heard of it before. That it was a rare bile duct cancer, usually diagnosed in late stages, and that surgery was our only option for a potential cure. And even if I was fortunate enough to get that surgery, the recurrence rate was quite high. So my first major decision to make was to find a surgeon, and I'm from Billings, Montana. There's not a lot of experts in cholangiocarcinoma in Montana, so we did seek a second opinion at Mayo Clinic, but for personal reasons, I did choose to stay home and have my surgery in Billings 3 weeks after my diagnosis. I was told that they had clean margins. They removed 2/3 of my liver, and the cancer was gone. I was not even offered adjuvant chemo back then. Back in 2009, it wasn't really standard of care at that time. My first post-op scan was about 3 months later, and it showed spots in my lungs. They tried to do a CT-guided biopsy, but couldn't get enough tissue. I had lung surgery with the VATS procedure, and they confirmed metastasis in my lungs of cholangiocarcinoma. I was now considered a stage 4 terminal cancer patient. I knew that if I wanted to beat this, it had to be something new because what was out there was not curative. This picture was taken days after my VATS procedure. 2010 was a year of graduations. This was the first with my husband Steve receiving his nursing degree. I was still in nursing school and hoping to follow him, graduating in a year. The next graduation was a few weeks later at my daughter's Sammy's high school graduation. We attended her ceremony and then immediately hit the road to drive to Minnesota to visit the Mayo Clinic for a second opinion. Cancer messes with the whole family. Unfortunately, Mayo didn't have anything more to offer than the standard of care in which I could get back home in Montana. I asked my local doc to be as aggressive as possible and to look for clinical trials. He found one, but unfortunately, in 2010, 16 years ago, my insurance did not cover the standard of care costs that went into participating in that clinical trial. That was my first taste of advocacy, is telling my story in the Capitol at Montana and helping to get a bill passed that required insurance companies to cover those costs so that we could participate in clinical trials. But because I didn't want to leave my family in financial stress, I chose not to participate in that clinical trial, and I went ahead with standard of care. I soon found out that chemo stinks for me. At least I had my weekly chemo cocktail of Gemcis with a side of steroids, antiemetics, Benadryl, followed by 4 days of Neupogen shots. And this lasted for about 6 months until the toxicity set in. I still have ringing in my ears, the tinnitus, neuropathy in my hands and feet, And then I was blessed with a bonus of steroid psychosis. This was the third graduation, yes, of the year. My son Seth graduated from Navy boot camp. I was so proud of him for joining the Navy. I attended his graduation after being on chemo for about a month or so. I remember being so sick, but trying so hard to be brave and present for him. The second picture shows me after doing 3 different chemo options for about 18 months. It was hard to recognize myself when I looked in the mirror some days. I'd found the Cholangiocarcinoma Foundation earlier that year and had been a lurker on the discussion boards, reading and learning from others' experiences. It was October 23, 2010, when I made my first post. My words of, "It frustrates me to not have more answers to this rare cancer," still ring true today. It was comforting to chat with others who understood how it felt to be in my shoes. I was grateful to have found a community and the connection I needed. My husband titled this picture, "Rough Day." My next big decision was to choose between quality versus quantity. After 18 months of on and off chemotherapy and not really having a good quality of life, doing more sleeping, more time in bed, and not really interacting with my children, I made the decision to stop all treatments. And it was the hardest decision I ever made because I didn't want my kids to think I was giving up or that they weren't worth me suffering through the side effects. But miraculously, I found a clinical trial. About 3 weeks after I made that decision to stop chemotherapy, I stumbled upon a clinical trial. I'd stopped looking for clinical trials because back then all that was available was chemo trials, and the chemos I'd already been on, different combinations, and I really did not want to do chemo again. But I sat down to pay a medical bill, and for some reason I clicked on the NCI.gov clinical trial thing on my computer, which somehow opened up to a page where I read this clinical trial. And I remember looking at my husband and saying, "I think we should try this. You know, it has the initial chemo to wipe out your immune system for a week, and I can do anything for a week, and then it's your immune system doing what it needs to do." And so we entered into this GI tumor-infiltrating lymphocyte trial at the National Cancer Institute. We took the information to my local oncologist and said, "Hey, we really want to do this." And he said, "I'll support you in whatever you want to do. But typically we work with the University of Denver because that's where Montana is closest to." And I said, "No, I feel strongly about this. One, because the NCI is a federally funded facility and it was going to cover all the costs and I didn't have that insurance worry. And two, it also covered my costs to travel as a patient from Montana to Bethesda, Maryland to get this treatment. And so without those possibilities, I would have never been able to enter into this trial that ultimately got me here today." We went in March of 2012, were accepted, came back later. They removed 4 tumors from my lungs, pulled the T cells from those tumors, grew them in the lab, and I returned in April to receive the cell-depleting chemo, followed by 42.6 billion cells and a drug called IL-2, and another couple weeks in the hospital to recover my immune system enough to be able to go home. I remember dragging my family out so many times during this time frame, making them do family pictures because I just wanted to create memories just in case. The results of the first treatment was I was the 9th person to enter into this trial, the first one to have cholangiocarcinoma, and actually the first one to ever have a response. I had an overall shrinkage of almost 30%, not quite that partial response. But the great thing about it is it was a one-time treatment. I had no other further treatment for 18 months, and I ended up enjoying a great quality of life again. When I went into the trial, I had tumors all over both my lungs and all over my liver, and a cough that I couldn't say 5 words without going into a chronic cough. My kids couldn't hug me without me going into a chronic cough. And at the end of that 3 to 4 weeks in the hospital after receiving treatment, that cough went away. And I was like, ooh, something's working here. And of course, the doctors were very cautiously optimistic and saying, no, we have to wait and see. During that 18 months, for 6 months it shrunk, like I said, stayed stable for a while, but then it started to grow again. So they wanted to retreat me again with the same treatment because they had discovered in that 18-month time frame that one of my T cells was reactive to one of my specific mutations, and this next go around, they wanted to grow those specific T cells and try it again. I remember being at the NCI and Dr. Rosenberg coming to the foot of my bed as I was getting apheresis, and his excitement about this just was so contagious, and it made me so excited, and I was like, I couldn't wait to get started. The hope that he shared just made such a difference. So I went back for another surgery, had more tumors removed from my lungs, where they pulled those T cells again and grew them in the lab. And in October of 2013, I received 126 billion T cells, so 3 times as many, 4 doses of IL-2, which is not the funnest drug, but worth it to go through this, and tons of hope. Results for my second TIL treatment, I had an overall shrinkage of about 50 to 60%, 3 years of a great quality of life, no other treatments, just my immune cells working to keep the cancer at bay. Because of the response that I had, the data was published in the Science Journal, and because of that, the media reached out and did a story on my personal story with my family. It was published in the New York Times and the Wall Street Journal, and it spread across the world overnight, and literally in a week, I had hundreds of phone calls from patients and caregivers all over the world. They didn't need to know that the specific treatment would work for them. They just needed to know there were possibilities out there. So that's definitely the day my love for patient advocacy was born, and I just knew that I wanted to be able to help in some way. So again, I was the first person to be treated with TIL targeting a specific mutation. Went over these already, but I wanted to show you some of the CT slides of tumors in my lungs pre-treatment and then 7 months after my second treatment, and the tumors in my liver that had shrunk. Again, I really wanted to do some patient advocacy. So during treatment, I started volunteering with the Cholangiocarcinoma Foundation. I attended the second annual conference where I told my story for the first time. And it was the first time they had a patient at the conference. And there was about 100 physicians and researchers in the room. But I got off the stage and one of the researchers was crying. And he's like, "Oh my gosh, thank you so much for sharing your story. You know, I study this, but I've never ever talked to a patient." And it was just so incredible to make that connection. And my children, they got sucked into being advocates too and volunteers, and you know, they do a lot to support cholangiocarcinoma awareness. Unfortunately, 3 years after my second T-cell treatment, there started to be signs of growth again. So September 2016, Dr. Rosenberg decided to be proactive, removed the remaining tumors from my right lung and studied them. They resequenced them and did the research on those tumors. Again, back to the OR for my fourth lung surgery. This time it was a thoracotomy, which was definitely the worst. The research showed that those mutation-reactive T cells were still present, which was good. The same mutation was still there, which was good. But this time the tumors were infiltrated with the macrophages that express those high levels of PD-L1. And so my lay term for that understanding is that's the cancer's way of blocking the immune system from being able to see it. And they decided to treat me with pembrolizumab and open that window so those T cells could recognize the cancer again. I don't think the pembro would have worked had I not had the T cells there to do the job afterwards. After the second dose of pembro, we did a scan, and immediately there was already shrinkage again. So I did a total of 8 doses of pembrolizumab. I had the last one in March of 2017, which is 9 years ago, and that is the last treatment that I have ever had. There was a lot of highs and lows of doing the TIL trial. People always want to know what I did, how am I here 16 years later. I would love for them to figure out how to make it work for everyone, but unfortunately it doesn't. For me, it gave me a chance to live. There's a lot of, surgeries. The hospital stay is 3 to 4 weeks in the hospital. It's not an easy treatment. The good thing about it, though, is it is a one-time treatment, and if it works, it's a game changer. At the time, I just wanted to enter into it because I wanted to help advance the science for future patients. I got to experience the National Cancer Institute, the National Institutes of Health, which, being from Montana, I had no idea existed, and it is an incredible facility. I got to watch my niece and nephew grow up. They lived out in the DC area, which was kind of cool. But I was away from my family, and there was a lot of recovery. Because I had such a good response to that treatment, I did start volunteering with the foundation, and I wanted to find a way to pay it forward. I'm now a full-time employee with the foundation, and I serve as a patient advocate on many different panels: the NCI Hepatobiliary Task Force, Council of Research Advocates, ECOG-ACRIN, and I serve on the NCCN guideline panels as a patient advocate as well. And C3 Collaborative is a Canadian group that is now doing TIL research in Canada. I also have had the opportunity to go to the White House and present my story and talk about the need for funding for research. And I spoke to the largest gathering of U.S. senators at NCI to share how important it is to fund research for patients like us. The Cholangiocarcinoma Foundation's mission is to find a cure and improve the quality of life for cholangiocarcinoma patients. It was founded by Stacy Lindsay. She lives in Salt Lake City, Utah area, and her brother Mark was diagnosed with cholangiocarcinoma at age 38. And before Mark passed away, he asked Stacy if she would start this foundation and carry it on so that all the research and the materials and the resources they found wouldn't go to waste, and that people after him would have hope and resources and materials. The Cholangiocarcinoma Foundation likes to consider ourselves the hub of cholangiocarcinoma. We want to be the connectors to donors, patients, caregivers, scientists, industry, volunteers, professionals, the regulatory, FDA, and all our partner organizations. We have two pillars, patients and caregivers and research. I have the privilege of leading the advocacy team, which is in charge of the patient and caregiver pillar. Again, we want to connect patients and caregivers to the resources and to the materials they need early and often. We want to provide that education support, engagement to a community, empower you to be the best patient advocate you can be, and then equip our providers with resources and collaboration opportunities. We have a thriving advocacy team of 9. So if you haven't connected with us, please do. You can talk to an advocate one-on-one. You can get a mentor, and I will talk more about that in a little while, but we are here to help you, so please feel free to reach out. Our goal is to provide access to experts, to biomarker testing, to clinical trials, to treatment, and to support. We want to help you guys find the right team. We have a specialist map on the website. It's just a tool to help you look for providers in your area. We want to make sure you're getting biomarker testing early and making sure that you know if you have specific mutations that have targetable drugs, or if there's clinical trials that might be targeting one of these mutations. We have many resources. We do webinars, educational webinars twice a month. Most of those webinars talk about clinical trials that are in progress, and it's a great way to find out about what's going on in the clinical trial world. You can log in and ask questions in real time. We also do updates on side effect management, nutrition resources, and different things like that during our webinars. We have many, many virtual support groups for patients, caregivers, young adults, Spanish-speaking, our newly diagnosed and connected, and then our bereaved caregiver group. We are on all the social media channels. I can't name them all because I'm not good at that, but if you're on them, please follow us. We have a nutrition resource, and if you don't have it, I highly recommend you get on the website and download a copy or order a book. It'll give tips and tricks on what you can eat for different side effects, or why you're taking certain drugs, what you need to manage after surgery, and different things like that. We also have a 100 Q&A book that is actually being rewritten and will be coming out here shortly. We offer newly diagnosed care kits to all our newly diagnosed patients or newly connected to the foundation patients that have resources and some comfort items in them. We run a program called Cholangio Connect, which is a one-on-one mentorship program. So any of you mentors out there, thank you for mentoring. Any of you, those of you who don't have a mentor and want one, please connect with one of the staff members so that we can help you get signed up. It's an opportunity to connect with someone who's been in your shoes, who knows what's going on, who can provide that emotional support. I have seen these relationships sometimes be just a one-time phone call needing that first interaction to lifelong friendships. We've had over 1,800 matches made since we started this program. Our care teams are local community care teams in different areas. We have 6 of them so far: Baltimore, Washington, D.C., Chicago, Houston, Nashville, New England, and Seattle. This is just another opportunity for the community to do connections and bring patients and caregivers together, but also to host events at institutions and provide that awareness to cholangiocarcinoma. We have a group of research advocates. So for the physicians and researchers in the room, If you ever need somebody to give you feedback on clinical trial design, patient-facing materials, this is a group that loves to do that. They are usually a group of patients and caregivers who really want to study more about what's going on in the science of cholangiocarcinoma and then can lend their expertise as patients and caregivers. They also participate in many panels and do grant reviews for people who are doing research in cholangiocarcinoma. We partner with many of our international groups around the world, cholangiocarcinoma groups around the world. We have been very good about giving them all of our resources so that they can label them with their own logos and share with their own community. We share our expertise, we connect them with our partners, but mostly we want these international groups to grow so that they can provide the best resources to those patients in their area. We hear from international patients every day, but they're going to get better help from people in their area who know how to navigate their medical system. The second pillar is research. Dr. Juan Valle leads our research group, and we are so fortunate to have him. He is an expert in cholangiocarcinoma. We have an international cholangiocarcinoma research network. We have 480 ICRN members from 228 institutions in 36 countries all over the world. Their mission is to rapidly translate scientific discoveries into practice for cholangiocarcinoma patients through collaborative, highly translation international research network. And this is, like I said, a worldwide group of physicians and researchers who are working on finding better treatment options and a cure ultimately for us. There's many ICRN committees, and we have a scientific and medical advisory board that's led by Ben Stenger and Nilo Azad. We have a next generation committee that's led by Dr. Kristen Spencer and Dr. Nilo Azad as well, a research committee, a data committee, and a program organizing committee. So for any of your physicians in the audience, if you're interested in joining the ICRN, please reach out to Dr. Valle. We provide research grants every year. We do fellowship grants for early career researchers. It's a one-year grant. We do about 10 grants per year. We do one career development award a year. It's for mid-career, and it's a 3-year grant. And then we also do an innovation grant for senior career, early mid-career, and that's also a 3-year grant, and we do one of those a year as well. Over the years, we've given away 71 research fellowships from 2015 to 2025, totaling over $3.8 million, and these are some of the institutions who have benefited from those grants. Every year we have an annual conference in Salt Lake City. This year we will be celebrating our 20th year as a foundation. The annual conference is May 1st through 3rd, and the co-chairs are Luke Bolter and Tim Gretton. Tim is from the NCI, and Luke is from University of Edinburgh in Scotland. The conference is incredible. I can't even put into words describing the conference. If you guys get the opportunity to come, please join us. We will have over 250 patients and caregivers there. It's just one of those things that you have to experience in person to understand. I can tell you how amazing it is, but until you actually go there, you just feel the energy in the room. I see you guys back there. Yeah, many of these people here have their pictures up there, but it's incredible. So if you can't attend in person, please join virtually. It is the most amazing thing to sit in a room full of hundreds of physicians and researchers who are working on a cure for us. So for me, I feel that I've come full circle. I started my clinical trial as patient 3737. I'm now privileged to sit in audiences, big cancer conferences, and see Dr. Rosenberg present the data on my personal story, and it never ceases to give me goosebumps. I'm privileged to be one of the patient voices for the Canadian C3 Collaborative, started by Dr. Rebecca Auer. Her husband was diagnosed with cholangiocarcinoma 4 years ago. She recognized the need for more research in cholangiocarcinoma and applied for and received a $7.4 million grant specific to cholangiocarcinoma. This research will include a clinical trial with TIL like the one I did, and Dr. Simone Turcotte was my first research scientist out at the National Cancer Institute. He will be leading this trial. I'm so grateful for the NCI team, Dr. Turcotte, Dr. Rosenberg, Dr. Tran, and the entire surgery branch team who saved my life and continue researching this area for future patients. I'm even more grateful for time. When I was first diagnosed, I begged God for 5 more years to get my kids a little bit older. I have 6 children, and when I was diagnosed, the youngest was 7, the oldest was 20, and I just wanted to get that little one a little bit older. And then I hit that 5-year mark, and I'm like, all right, God, can we renegotiate? I want to be a grandma. And so I am so happy to say that I am now the grandmother to 5 beautiful grandchildren, with number 6 on the way in September. I'm so grateful for time that I never thought I would have. I've watched 3 of my kids get married. I have a 4th one getting married in July. And I'm even grateful in tragedy. I lost my 24-year-old son, unexpectedly, the same one who turned 14 on the day that I was diagnosed. I'm grateful I was there to help my family deal with this tragedy. I'm grateful I got to watch him grow and to love him for the 24 years I was so fortunate to be his mother. Grateful I was able to see him be a brother to his siblings. He loved his siblings so very much. Grateful I got to watch him get his driver's license and graduate high school. I'm also grateful to be with him through all the struggles. I'm just being a little more open about sharing this story. But cancer affects the whole family. During my journey with cholangiocarcinoma, Skylar struggled the most. Shortly after my diagnosis, he started hanging around the wrong crowd and experimenting with marijuana. That led to other drug use throughout his teenage years. It was the most challenging trial as a parent I have ever endured. I would have fought cancer 100 times over not to have had to go through this with my son. Just like cancer does not discriminate, neither does addiction. And ultimately, he went through treatment, but only for 30 days because of insurance coverage. He attended a Christian sober living home in the Seattle area and thrived for a period of time. He was rebaptized, learned to manage money, cook and clean for himself, and received a career pathway. Unfortunately, when he returned to his hometown, he relapsed again. I remember the day he told me his girlfriend was pregnant. I was terrified and thought at the time it was the worst thing in the world that could have happened. But looking back 7 years ago, it was absolutely the best thing and the biggest blessing to our family. He really did well until about 6 months after his son was born and again relapsed. He headed back to treatment, this time for about a 60-day program, which in my mind is still not long enough. This time after treatment, he went to live with his older brother, Seth, in Indiana. He got a job and was working to be able to send for his girlfriend and son, and he was doing so well and working so hard. That is why it's so hard to receive the phone call I did on January 8th, 2020 from my other son, Seth. If this call had come in a few years earlier, I would have been prepared for it. I was not ready for it. I had just spoken with Skylar the day before. He had FaceTimed his son. He had been sick with strep throat, and he was out from work with a doctor's note. That morning, Seth saw him before he left for work, and when Seth came home at lunch, he found his brother on the couch, not breathing. Seth called 911 and started CPR on his brother. There was no sign of overdose or anything unusual, and to this day, we don't know what happened for sure, but I have to believe that drug abuse contributed to his death. We rushed to his side. His entire family was with him over the next several days. I knew when I arrived, the look of my son's beautiful blue eyes, that he was gone. We as a family wanted to donate his organs as a way of making something good come out of something so terrible. I was happy to learn that Skylar had already agreed to be an organ donor on his driver's license. We worked with Donate Life in Indiana, and it was a beautiful experience. They did such an excellent job of making our family feel at peace. They helped to create memories for us by recording his heartbeat on these teddy bears. They made one for every one of his siblings, his parents and his son. They gave us imprints of his hands and a lock of his hair with a heart strip. And on the day we took him to surgery to harvest his lungs, liver, and kidneys, all the staff at the hospital lined the halls to honor his donation of life as we walked with him. The longest walk of my life, but the most proud I've ever been of my son. He saved 5 lives that day. Sorry, this was the walk of life, and I'm not sure how to skip it. Can we hear it? Do it again. One of those lives was another wife, mother, grandmother, and sister with cholangiocarcinoma. We were able to self-direct his liver to a cholangiocarcinoma patient, which I know would have made Skylar so happy to have helped someone like his mom. Leah received Skylar's liver after being diagnosed with cholangiocarcinoma a few years prior. Leah lived for 13 months after her transplant. She passed not due to liver issues or recurrence, but due to COVID. It was so unfortunate to hear this news. Her family was so scared to tell me as they were afraid I would be disappointed about giving Skylar's liver to her. I was absolutely not disappointed. I know I would have given anything for another 13 months with Skylar. Leah's husband, Wayne, said those 13 months were filled with memories of a great fishing trip with her kids, an amazing quality of life, and it was so cherished. I'm so grateful for her family that they had her for another amazing 13 months. Lastly, I'm grateful for being a part of CCF for the past 14 years. It has given me an opportunity to help others, a platform to have a voice for patients with cholangiocarcinoma, and a chance to have a family I would have not have been a part of had I not been given this path. There is such strength in community and hope in research. I want to let everyone know that tomorrow there's a local fundraiser called Because Jonah Would. Its location is McCullough's, did I say that right? In Wantagh, New York, next to the LIRR station. It's going to be from 1 to 5. Open invitation for all of you. This is Jonah. He was a patient with cholangiocarcinoma, and he touched many, many patients in this area and around the world. He was a mentor, and he provided hope. And from what I hear, he had a way of being a magnet in the infusion center, in the cancer center, and just pulling all the patients to him. His wife will be hosting this fundraiser. They've raised enough money to sponsor a research fellowship in Jonah's name, and we couldn't be happier. So I encourage you guys all to attend that tomorrow. One other thing I would like you to do is reach out to your local representatives. You can scan this QR code, join us in reaching out to elected officials to help elevate awareness on cholangiocarcinoma. We have a new platform. It's called One Click. It's really easy. You put in your information. It pulls up all your representatives. But mostly, Awareness Month is coming in February, and all we really want you to do is just say, hey, my loved one, or I have cholangiocarcinoma. We'd like you to be aware of it, and just spread the awareness so that when we're out there advocating, they'll know. They'll know. They don't have to say cholangio what. They'll hear about it. So if you could send this one click, we would surely appreciate it. Again, there is such strength in community and hope in research. And we're so grateful you all are here. Thank you.
Now I have to be up here and not cry, so thanks so much for that. Thank you so much, Melinda. Wonderful. All right, so without further ado, we'd now like to move on to our special session. I think this will be really a lot of interest from all of you, novel liver-directed approaches to cholangiocarcinoma. So rather than doing the traditional approach of talking about how we do stents and how we do radiation and how we do, surgery, we really wanted to focus on some up-and-coming, newer ways that we're managing patients with local regional disease. And so with that, I'd like to introduce Dr. Brock Hewitt, who is the hepatobiliary and pancreatic surgeon here at NYU, who is deeply committed to caring for patients with precision, empathy, and clear communication. And he specializes in complex diseases of the liver, bile ducts, and pancreas. He works with the team leading innovative approaches, including robotic surgery and non-invasive treatments for liver tumors. And he is passionate about making sure patients feel heard, supported, and informed throughout their care. So please welcome me in joining him to discuss how he incorporates histotripsy in the management of cholangiocarcinoma.
Thank you for the opportunity to speak today. I mean, what an incredible story. I mean, so powerful. And, I hope to kind of get across a similar message of hope here when talking about histotripsy and the care of these patients. But thank you so much for your testimony, and just very, very powerful. So we'll get kind of right into this. So we are an observation site here for Histosonics. We've been doing it since February of 2024. So we've been doing it for coming up on 2 years now. And so we train and teach others to use this technology. So in this talk, we're going to be talking about some high-level information here, and it's certainly going to be patient-centered. We have a Q&A session after this talk, so keep those questions in mind. We're going to go over what is histotripsy, since it is a newer technology, kind of, and then to see if, what are the criteria for becoming a potential candidate for this technology, and then what can you expect if you do get it? And then finally, at the end, talk about some future directions and how we can kind of better incorporate this in our current treatment algorithms. So Cholangiocarcinoma, I don't have to go into too much of this, but just highlighting a couple different points here in the sense that there's a couple different types when we're talking about tumors of the bile ducts. So that's what cholangiocarcinoma is, and it can be, the location matters in this sense, particularly for histotrypsy. So there's disease called intrahepatic cholangiocarcinoma, as we just talked about. So that's disease that's within the liver itself. So the bile ducts in the liver develop the disease. And then there's distal cholangio, or what's called extrahepatic, meaning disease that is in basically the tube system that comes outside of the liver. And then parahilar is kind of in between. So it's basically right there at the interface between the bile ducts and the liver itself. And it's kind of in a high real estate area, and you'll hear more about that later. So histotrypsy, though, specifically is really more so for intrahepatic and some parahilar, but really the focus is going to be on intrahepatic. So when we're talking about distal or extrahepatic cholangiocarcinoma, this will not really be applicable to that currently. So taking a 10,000-foot view, when we talk about, well, how is cholangiocarcinoma treated? And any cancer is really treated, or any solid tumor at least, of which cholangiocarcinoma is, treated with two modalities. So we're talking about local therapy, of which histotrypsy is an example of that, surgery, radiation. So these are all treatments that are designed to remove and or destroy disease that we can see, whereas systemic therapy is more global. So the most common situation would be chemotherapy, but things like TIL, things like immunotherapy, hormonal therapy, those are treatments that can be applied to patients and then go all over the body. So it gets disease you can see and disease you can't see when treating these tumors. So just keep in mind, we're talking about a local therapy here, okay? So when we treat these diseases, well, how do we know when to give local therapies versus systemic therapies? And so when it comes to cholangiocarcinoma, by far and away, the most important factor is it removable? Surgery is the definitive treatment for cholangiocarcinoma, intrahepatic cholangiocarcinoma. If you can remove the disease safely and leave enough liver behind such that the patient will, the liver will survive, then that certainly is the treatment. And then afterwards, we talk about adding systemic therapy and other modalities. If it's not resectable, we try to make it resectable. So whether that's using local therapies, something of which histotrypsy may be a part of, or whether there's other directed therapies that we can do that with. And obviously, clinical trials, any patient, if they screen positive for that and can get into a clinical trial, we always encourage that. And then, as Dr. Spencer mentioned, there's a number of components of like stenting and draining and things like that if we need to manage patients' symptoms. So now, focusing on histotrypsy. So what is histotrypsy? Well, histotrypsy is a technology that's been around since about 2001. So it's you can kind of see here over the progression, and it was initially started at the University of Michigan, and then it ended up kind of being taken over by a company called Histosonics, and that's currently the only company that will offer this technology. So just to not get confused, histotrypsy is the technology. It's what we're going to be talking about here, and Histosonics is the company. And so you can see here that over time they've done a couple clinical trials, and that ultimately here in 2023 led to the FDA approval for the use of histotrypsy in the management of primary, like cholangiocarcinoma or hepatocellular, which is liver cancers, or secondary, so tumors that go into the liver, so something coming from the lungs or coming from the liver or from the coming from the colon or the pancreas, like that. So it's for treatments of any tumors in the liver. It's FDA approved for. But just because something's FDA approved doesn't mean it applies for everybody, and so we'll talk about that in a little bit. So how does it work? What is unique about it? Well, what it specifically is, it's focused ultrasound. So the same thing that you would get, ultrasound if you're pregnant, you get an ultrasound on the belly. It's the same type of technology that way. It's focused ultrasound, though, that is delivered through the skin, so there's no incision made, and it targets on the tumor specifically. It doesn't use heat or radiation to destroy the tumor cells. What it does is it creates, due to these waves, the ultrasound waves, it creates bubbles in the tissue that it's targeting, and these bubbles expand and contract really quickly. So they end up bouncing around kind of like bowling balls in a way, and they end up breaking up and tearing the cells. And then the cells are basically broken up, and they are dead cells, so it doesn't spread the disease that way. These are dead cells with dead disease, and then the healthy liver goes in and cleans that up. And so over time, you see shrinkage of these tumors, and that's how it works. So how is it performed? Well, you can see here on the your screen left. So it's a procedure right now where you do have to be intubated, so it's under general anesthesia. You get put to sleep, and the technology you can see there sitting over the patient is a container, and that container has water in it. And you can see here in the top right, the device, that's the head, and that goes into the water, and then through that device, the patient is treated. So again, there's no incisions, no needles, nothing like that in the abdomen. And what you see here kind of in the middle is that if you were to take the top off the hood, is that you see a bunch of these, all these kind of little nodes here that then each of those directs an ultrasound wave, and it all comes in focus to one point. So how does that actually look? So you kind of see these both going at once, but on the right-hand side here, you can see this is a, oh, where did it go? To that one. All right, well, we'll just take this video once at a time here. So it shows you, so we're turning up the voltage here on this, and what happens is again, those waves come down, and what you see is this is a bubble cloud, and this is where the tumor is being treated, but it's very focused, as in it doesn't destroy any of the tumor outside of that bubble cloud. So you move around the bubble cloud, and it does it automatically. Once you target it, the machine does, and then it destroys everything within that bubble cloud, except, which is kind of a nice component of it, is it does maintain certain structures. So things like blood vessels and bile ducts, it maintains those. So this is where it is a little bit different than traditional ablation therapies, so like thermal ablation that we call like microwave ablation or radiofrequency ablation. Those types of technologies kind of act like a bomb in a sense that you put the needle in and you burn, and it basically destroys everything within the certain radius that it's treating. Histotripsy is a little bit different because of the way the technology works. Things that are very strong, like blood vessels and bile ducts, those are maintained, okay? So it's why it can be used in certain situations where other thermal ablation and other ablation technologies cannot be used. So again, part of why we like it and why we are hopeful for its application in a number of different tumor types is, for the reason I just said, is that it can preserve certain structures. It does destroy the tumors by going through the skin. It's a same-day procedure, so you go home the same day. It works with other treatments. So just because you apply histotripsy doesn't mean you can't use other different types of treatments, whereas some treatments, for example, burn a bridge, so to say, and once you get that, that's all that can be done there. Or for certain situations, like if you're going to get radiation, maybe it makes surgery not really applicable for you in the future because of the side effects of that. So this treatment, so far, we have not seen that it doesn't burn any bridges for that. It can be performed multiple times. So say, for example, you develop a lesion, you get it treated, and then in the future you have a recurrence that's close by, you can treat again with histotripsy. And I think one of the biggest things, particularly as we find out exactly how does histotripsy fit into the treatment paradigm, is that you don't have to disrupt your systemic therapy. So for patients that have metastatic disease, for example, that are being considered for histotripsy for any number of reasons we'll talk about, if you're on systemic therapy, we know that systemic therapy in that situation is by far and away the best treatment that you can get to improve your quality and quantity of life. And so anything that disrupts that, like surgery can, if you go to surgery, you have to stop therapy before and hold it for a time after while you heal. Histotripsy, you don't need to do that. So if you're getting treatments every 2 or 3 weeks, in that off week, you can get histotripsy and then maintain your current cycle. So I think it's very, very important because it is so critical to stay on that therapy, certainly if it's working. So how do you know if you're a candidate for histotripsy? Well, ultimately, as we are learning more and more about this technology, we're trying to find out, again, how do we balance this benefit versus risk when we're treating these patients, because any treatment you get, it carries a level of risk with it. And so we want to know, to the best of our ability, can we at least provide some benefit for you? As in, is it going to be greater than the risk? Because if the answer is no, if the risk is greater than the benefit, even if the risk is low, then it's probably not in your best interest to get that treatment. So when we screen candidates for histotripsy, certainly the most important thing is we take a step back and we look at the overall disease burden and we say, look, is this something, is the juice worth the squeeze, so to say? Is going through this treatment going to potentially provide any benefit? And we'll talk about that in a second. And once we do feel like there may be some benefit for that, now we kind of get down to specifics. Okay, is the tumor visible on ultrasound? Because it uses ultrasound technology. So for most patients, you need to see it on ultrasound. Now, we're making adjustments in trying to bring in CT scans and other types of imaging to enhance our ability to pick up these tumors so that way we can treat them. But obviously, if we can't see it, we can't treat it. We're not just going to randomly target places in the liver. So we need to be able to see what we're treating. And then, is it in a location that is amenable to histotripsy treatment? So not every location in the liver can be treated with histotripsy. For example, those tumors that are really high up under the lung, well, the lung is filled with air, as we know, and like I mentioned early on, when histotripsy is treated, you have a water bath over you, so you need a fluid medium in between the treatment head and the target, which means if the lung is in the way, it won't work well, and it can potentially cause some complications. So we don't treat through lung to get to lesions. Now, we're able to get around that sometimes in terms of doing kind of, I think, interesting things by, for example, you can collapse the right side, the right lung, and only breathe through the left lung. We can do things like that, which then improve the area, but there are still certain locations in the liver that we just can't treat yet with histotripsy. Also, metal kind of refracts it, so if you have a metal stent in the area, that can refract sometimes, and same thing with calcifications. So if you have some firm calcium deposits in the liver, or a prior treatment has killed some of the tumor and it's calcified over time, that just makes the treatment zone or the treatment area a little less reliable because of that. And then size limitations. So currently it's around 3 centimeters. However, that being said, we have treated a number of patients where we've done overlapping treatments. And in those situations, you can treat larger lesions. So will histotripsy help you? So right now, more broadly speaking, we use histotripsy as in a number of different clinical scenarios. So we use it as a bridge to transplant. So for certain patients that are on the way to transplant, of which cholangiocarcinoma, there are some patients, particularly those with Perry-Heiler disease, that may potentially be candidates for transplant, and histotripsy can be used in certain select situations. Neoadjuvant, meaning that we can give histotripsy to patients to shrink the tumor a little bit before surgery, such that we maybe have to remove less of the liver itself, so we can kind of preserve more healthy liver. Like I mentioned, when it's really by critical structures, because the edges or the margin treatment of histotripsy is very specific, whereas traditional heat or thermal ablation techniques, the margins aren't quite as clear in terms of how the heat dissipates across the treatment zone. It's not as clean of a margin. Well, with histotripsy, it is a little bit cleaner. And then central lesions as well. Sometimes those can be difficult. Again, with traditional ablations, you're using needles and things like that to get to those locations. But histotripsy can get there, without having to use a needle or for surgery. So again, as we're learning more and more about this technology, how can we offer this technology to patients in a safe way, in a thoughtful way? Because by far, our job as clinicians is to first do no harm, right? So if there's a technology that works, but it causes an immense amount of pain and discomfort and has a number of side effects, then it's not the right thing, even if it could potentially work, because getting the treatment could be worse than the disease itself. So far, what do we know? Well, our institution and a number of others across the country have kind of pooled our data together in terms of finding out, hey, look, is it safe here? Is it safe there? When we all come together and we look at that, we published a paper last year that looked at the first 230 cases across a number of different centers, and we found out that it actually is quite safe. And again, all of us were early users, which means we were just figuring things out. So even in unexperienced hands, as we were first trying to adopt this technology, we found that it was quite safe. So over 95% of patients had very, very mild side effects from this treatment. So less than 5% had more infections or bleeding or damage to the surrounding structure. So it was, even in inexperienced hands, it was still quite safe. The most common experience from patients is they'll have a little bit of pain or discomfort around the site of treatment. Most of them take over-the-counter pain medicine. They don't even need narcotics. They'll have some flu-like symptoms for a number of days, but then that resolves, and then they're back onto their other treatments. One of the important points about histotripsy, and really with any new technology, is the need for more data, for more information. And so right now, we try to offer any patient that we're going to offer histotripsy to, we also offer them enrollment in this Boombox trial. And what Boombox is Boombox is a national trial that its goal is to enroll up to 5,000 patients as a surveillance or as a registry, which means we're going to collect a patient's information, and that's it. It doesn't randomize to getting historytrypsy or not. But what we do with that information is we follow folks over time such that in a year from now we can look back and we can say, okay, is it true? Is historytrypsy still quite safe? And if it is safe, how many patients were able to benefit from the treatment? And so we can look back at that data. So 5,000 patients is what we're on our goal is to enroll. And this is a national study. So every patient we treat, we try to enroll them into a clinical trial. And so, okay, so does historytrypsy work? So we talked about it is quite safe, and now does it work, right? And when you're going to go through all this. And so there was another paper that we were part of that we published earlier this past year, or last year, that showed that when we treat a tumor, a year later, we looked at all those tumors, and 90% of those remained treated, as in they did not grow. And so that's pretty reassuring. That is in line with other traditional therapies like microablation and radiofrequency ablation and even radiation. So it is in line with those types of treatments. You know, when you're talking about trying to treat something, you got to understand, are you trying to treat that whole lesion? And sometimes we can treat partial lesions. But again, obviously, if we're treating a partial lesion, it's not curative, as in we're not trying to cure that lesion. We may be more interested in trying to manage symptoms. So I can give you an example of that. There was a patient that we've treated that was a partial lesion that was starting to grow into the this was a different type of cancer. It wasn't cholangiocarcinoma, but it was trying to grow into the bile ducts, and they were about to need a stent that was going to go in there, you know, to try to open that up. But we were able to treat that partial area by the bile duct and shrink that area, and then they ultimately never needed a stent for that. So we were able to avoid that stent placement, and they were able to remain on their therapy. So that's a situation where you may treat something partially with a good response. So again, what is the goal of treatment? Is the goal trying to cure, or is the goal trying to manage symptoms and or prevent additional interventions like we were talking about there? So as far as long-term, and so like I mentioned, in 1 year we know that the tumors are well controlled. But the bigger question is, and really the more important question is, does this move the needle in terms of survival? Does this extend your survival? And the answer to that is, we don't know. We don't have that data yet. So that's why we're trying to come up with clinical trials. Dr. Spencer has designed one recently, and will hopefully be getting approval soon for patients specifically with cholangio. And actually, it will be the first historytrypsy trial specifically designed for cholangiocarcinoma patients. So we're very excited for her work in this field and to partner with her and to be able to offer this to patients hopefully sooner rather than later. But this data will then go on and help us be able to educate future patients and treat future patients, because right now we don't know if it moves the needle in terms of survival. So future directions here in the last couple of minutes. Like I mentioned, you know, the most important thing is these clinical trials that are coming up. Dr. Spencer has one. We're also on the surgery side of things developing a clinical trial to see how does it work in the flow of surgery, treating patients that are surgical candidates. So certainly we cannot stress enough, as was already stressed, clinical trials are absolutely critical in terms of gaining more information and also providing maybe some new treatments to patients under close monitoring. You know, one of the other questions is, well, how do we incorporate this technology into the current treatment paradigms? Again, I can't stress this enough. The standard of care treatment is surgery. If you are a surgical candidate, I strongly would recommend that for you. If you're not, then the question is, how can we best get you to become a surgical candidate? How can we shrink the disease? And systemic therapy is a big part of that. Targeted therapy, you know, get biomarker testing to find out what mutations you have. So those are the things that should be being done. And so the question is, well, how can historytrypsy add on to what's being done? It shouldn't be a substitute. It should be an add-on for select patients. The other things is that I mentioned, you know, you have to be intubated for this. Well, how can we do this procedure without intubating a patient? Maybe more like a colonoscopy where you get some mild sedation. The other question that keeps coming up is, how does this, although this is a local therapy, as I mentioned initially, does it affect the immune system more broadly? Does it have an impact on the immune system? And so we have a number of studies looking at to say, well, can we use historytrypsy with other systemic therapies, particularly immunotherapies, to help enhance the effect of immunotherapy? And so there's a lot of ongoing work there. And then at some point, you know, in the future, if we can prove that it continues to be safe and it continues to be effective, can we test it against surgery? We're not there yet. It shouldn't be used in that way. So our hope is that with more education and with more clinical trial and more data, that we'll be able to answer these questions. So again, at the end, I just want to be very honest here with everybody. And histotripsy is not a guaranteed cure. Again, 90% over a year, that means 10% it has some growth, okay? So just kind of keep that in perspective. And it may not be right for every tumor or even every person. So, we recommend you come to a high volume center like ours and others across the country that have experience with this, that have large surgical teams, medical oncology teams, radiation oncology teams. And we review all these cases together in that multidisciplinary way to make sure we're providing the best treatment for that patient. And then we'll evaluate it, is histotripsy part of that treatment paradigm or not? And again, the most common way so far we've been using it is in one of two ways, rather as an adjunct to current therapy, or we use it in a palliative setting, meaning we're trying to treat a symptom and or prevent an intervention like the stent example I gave. But there are a number of reasons for hope. I mentioned the clinical trials that we have that are about to be ongoing. We're learning more about how to administer this technology to patients in a safer way, like I mentioned with the lung ventilation and things like that. And we're also finding unique ways to combine it with other therapies for more of a synergistic effect to treat this disease. So I'll take questions. Thank you so much for your time.
All right, so Dr. Hewitt's going to remain up for, I have to take the microphone down about 2 feet, but he will remain up here for a few moments. If anyone has any questions, we have 2 microphones up front, if anyone wants to come up and ask. Get your steps in. You've been sitting for a little while now.
Me too. 2 feet. Good morning, Dr. Hewitt. Thank you so much for that very informative discussion. My husband Eric is a almost 2-year veteran here, sitting over there. My husband has 2 cancers. He's been diagnosed with multiple myeloma 3 years ago, intrahepatic cholangiocarcinoma 2 years ago. And one of the challenges we've had with the idea of clinical trials is he's not eligible for them because of that myeloma, and it causes clinical trial groups to not want to include him. My question about histotripsy, which is something we want to pursue further, is would a clinical trial involving histotripsy also have the same requirements or difficulties where there is a secondary cancer that a candidate?
Yeah, it's a great question. So right now, for example, that boombox trial that I mentioned, it's more of a registry, so it doesn't randomize you to getting the technology or not. So if he would otherwise meet the criteria to receive histotripsy, kind of like I laid out, he can get involved in that trial. As far as the other active trials, that would be including systemic therapies and receiving histotripsy, there is an issue, with multiple myeloma. I don't know if yours will it exclude that, him, you think, or no?
I don't remember offhand, but I mean, we are admittedly trying to write trial criteria a little more broadly so that if we don't think that the cancer is, number one, going to need treatment or in any way affect the interpretation of what we're trying to study with that clinical trial, to allow patients on. I don't remember exactly for the language, but this modality is still available through your insurance, through standard of care. So while we are trying to study it under the auspices of clinical trial, you could still see Dr. Hewitt and have those questions answered and potentially receive the treatment off the trial as well.
Yeah, the nice thing about it being like a treatment modality in the sense that it's a local therapy like that, is the big issue when having another cancer is that it may impact more so for the systemic therapy treatment option and in terms of how that's interpreted. But with histotripsy, we could very clearly distinguish what would be an effect from histotripsy and then what wouldn't, more so. And so I would encourage, to get evaluated, certainly.
Great, thank you.
Yes.
You're welcome to line up, too, with the microphones. You don't have to.
Good morning, Dr. Hewitt.
Morning.
I was diagnosed on November 6th. Sister Mary Joseph, nodule that went up. I've got the intrahepatic cancer at this point.
Yes, sir.
I was told that the tumor right now is at about 3 inches roughly, which is, what, 7.2?
About 7 centimeters. Yeah.
Would that exclude me from getting the histotripsy treatments because of the size, and if so, why?
Yeah, it's a great question. Have you been evaluated by a surgeon at all yet for surgical removal?
I went to Memorial Sloan Kettering because my oncologist coordinated a surgical review where they told me basically you got 6 months and you're done.
Okay, so yeah, I would be happy to review your case, but the size alone is not a contraindication for it. Like I mentioned, you can do overlapping treatments. However, I would have to obviously review your specific imaging and seeing why it was that you weren't a candidate for surgery, because even as was talked about earlier today, her tumor was bigger than that and was still resectable. So I would have to find out why specifically it wasn't able to be removed and things like that. But that size alone, no, would not prohibit you from receiving that type of treatment.
Okay, so to further the conversation I had with Dr. Jarnigan, he said basically their goal is, number one, is it curable? And if so, then yes, they would proceed with doing the surgery. Because it was a Sister Mary Joseph, they basically said that that had metastasized, and once that has occurred, there's no chance of surgery.
So yeah, when disease has spread outside of the original organ from which it came, it is considered stage 4 disease or advanced disease. And again, in that situation, we just need to balance the risks of whatever treatment we're talking about with any potential benefits. So in your situation, for example, histotripsy would not be a curative therapy because of the disease outside of the liver. So we would just have to balance the risks of getting histotripsy, things like infections and stuff like that, with any potential benefits you could derive from it. So certainly the best treatment for you is going to be a systemic therapy, so treatment that goes everywhere in the body. So it treats disease we can see and disease we can't see. And so, I would have to look at your specific imaging and to be able to give you a more detailed answer, but I would be happy to review that and offer my personal opinion on it.
Okay, I look forward to that.
Yes, sir.
Thank you.
You're welcome.
Hi, good morning. My name is Teresa Del Corso Elman, and I'm the chair of the Cholangiocarcinoma Foundation Board. And my late husband had cholangiocarcinoma from 2015 to 2019. And thank you for your presentation. I wish this treatment modality was more developed when he was patient. And it's his case that informs my question. So he was throughout his entire time in treatment, he was a stage 4 intrahepatic cholangiocarcinoma patient, unoperable. And he was the beneficiary of a number of local therapies that gave him better outcomes than we ever had expected to have.
Wonderful.
And so I'm obviously curious, would have he been a good candidate for something like this? And it informs my question, the way his case presented, he had multiple tumors across all of the lobes in the liver. And so, he had the hepatic arterial infusion pump to control some things. He had radiation that was done up at MSK that controlled some things. He had systemic therapy. And after he failed out of the clinical trial for the pump, they were able to use other drugs in the pump to manage the disease really well. So I'm curious, if a patient has multiple tumors across the liver, would they be possibly a candidate for something like histotripsy? Like what I'm hearing is you can treat one tumor of a certain size. Can you treat multiple small tumors?
It's a great question. So just take a step back. So the biggest, or the most amount of tumors we've treated in one patient is 5. It wasn't for cholangio disease, but we can treat multiple tumors. The most we've treated one patient was 3 times, so for different tumors. And yes, local control is very, very important. Certainly, I was glad he was able to get on the hepatic artery infusion pump. There's a lot of emerging data about that. I think it's a really fantastic modality for select patients. And you mentioned radiation. So I think in your case, the case you've brought up, there are certainly select situations where maybe a patient has multiple tumors and, for example, something like hepatic artery infusion is controlling all of them, but maybe say one of them is not being controlled and it's growing a little bit. And I would say, I think it's very much worthwhile to be evaluated in that situation to say, can we provide histotripsy to that tumor to provide control? The same way you would if you were saying radiation, right? So we want to radiate that one tumor to provide that control. Now, again, we just have to be very honest and clear about, well, what is the expectation? Is this going to extend life? We don't know, but we do know, for example, if it's close to a bile duct and there's an impending stent or a drain that needs to be placed, it can shrink that tumor such that then you don't need that intervention. Okay.
Great, thank you.
You're welcome.
Hello, my name is David Montfort. I'm a patient, and I just had a clarifying question about the metal infraction. I mean, if you had a metal stents placed, would this then preclude you from getting this therapy?
It's a good question. So it doesn't preclude you from getting it. What I would say, and so far this is our experience, and again, we haven't looked this specifically, but I've noticed this when we've treated some patients, is that if there's a metal stent or there's something like calcification, that firm in an area, what can happen is because the way the treatment works is you saw the bubble cloud, and it moves around and ultimately develops and treats a spherical or an elliptical zone. That's what it treats. And so as it moves around, when it lands on an area with metal in it, it can refract slightly and move a bit. It dances in a way. And so what I would say is then at that one particular area, I can't reliably say that that zone is treated specifically. So when a metal stent is present or a calcification, I always classify that as a partial treatment, not a total treatment. And it may be a total treatment, but I can't say with confidence, due to potential refraction there, that it is treating 100% of that tumor.
Thank you.
Okay. Okay. Yeah, no problem.
Okay, I'm going to go as brief as possible. I'm stage 4. I've had 2 aborted surgeries for the same reason. And I know you said something about what you can't see as part of a problem with the word. So between my flap and my abdomen, there's like a Woodstock party going on there. Would it be able, like if you know where they are, is there a way to target them in that sense? Because essentially it's almost like that's the reason that is aborted both of my surgeries, you know what I'm saying?
Yeah.
And if that can eliminate them, is that a possibility?
Yeah, so what I would say is, we're trying to get better at using CT and MRI, which MRI is the most specific outside of an intraoperative ultrasound, and MRI is the most specific tool to pick up tumors in the liver. What I would say is we can offer you a screening ultrasound by our expert ultrasound technologists that they could see. We would be able to tell you at that moment if we can see it, and if so, can we get access to it. So for patients where we're not sure if we can actually treat, meaning we're not sure if it has access or we can see it, we do a screening ultrasound. So I would encourage that.
So I reach out to you? That's
Yeah,
okay?
definitely.
All right, cool, thanks.
Okay. Yes, last one.
Hi, Doc. Thank you so much for this. I'm just curious to hear your thoughts about the future in the histotripsy space, specifically if there's any trials being done on other organs trying to target metastatic disease to decrease the burden.
So there is. We've done one here in kidney at NYU here, and so I anticipate that at some point it will get approved by the FDA for treatment of kidney tumors. And then there is one going on in Spain right now. We're hoping to bring it to America, but a treatment for primary lesions of the pancreas. So we are working to try to expand this technology. Yeah.
Thank you.
And I'll mention that Dr. Hewitt is running to a meeting because he's the recipient of a K award. So he's, congratulations to him really changing the field here. So we're going to move on. Yep, thanks. Applause for him. Thanks, Dr. Hewitt. So we're going to move on to our surgery and transplant colleagues. I'd like to introduce Dr. Alejandro Torres Hernandez. He's a transplant surgeon here at NYU. He also specializes in treating complex liver and bile duct conditions, including cholangiocarcinoma. He also provides personalized care using advanced approaches like minimally invasive and robotic surgery, liver transplant, and other highly specialized procedures. And I think I'm just going to say, as a group for all of our doctors, we are very committed to patients' recovery, returning home safely, meaningful outcomes, patient-centered care. This is really the hallmark of our whole surgical team, so I'll say that for everyone here. And I will also point out that there are different reasons why for each of us, but Dr. Torres Hernandez is the one who managed to wear green and is in very spiffy green pants today, so we really appreciate that. So without further ado.
Thank you for that. I do want to thank the Cholangiocarcinoma Foundation for inviting me to give this talk today. And I also want to give thanks to the testimonial that was shared earlier. It was very touching and inspires me to continue doing the work that we do here. So my name is Alejandro Torres Hernandez. I'm a liver surgeon. I specialize in liver transplant as well as hepatobiliary surgery here at NYU Langone Health. And I've been asked to talk about how I choose patients for surgery and transplant for cholangiocarcinoma. This is the agenda that I'm going to be talking about today. We're going to talk about the anatomic classification of cholangiocarcinoma, how surgeons think about it. And then I'm going to go through the role of surgery as well as transplant for both intrahepatic cholangiocarcinomas as well as perihilar cholangiocarcinoma, something that Dr. Hewitt had mentioned in his previous talk. So there are many different ways of classifying cholangiocarcinomas. The pathologists have a certain way, the radiologists have a certain way, the oncologists have a certain way. Surgeons, we think about it based on its location. And I'm going to focus on two types of cholangiocarcinomas. One is the intrahepatic cholangiocarcinoma, so these are tumors of the bile duct that are growing within the liver, and then these perihilar cholangiocarcinomas that are tumors in the bile duct that exist outside of the liver, but are above the gallbladder and below the bile ducts as they enter the liver. In terms of the breakdown, in terms of the percentages of the tumors, most commonly, when folks have cholangiocarcinoma, it occurs in this area. As Dr. Hewitt mentioned, that this is an area with high real estate. This is a perihilar cholangiocarcinoma. About 50 to 60% of folks present with this, whereas the intrahepatic, or the tumors that are within the liver, about 10 to 20% of patients present with this. And this helps us think about the type of surgery we can offer to patients when they present to our office. It's a tough cancer. Less than 40% of people who have cholangiocarcinoma are amenable to surgery or transplant. And one of the first questions that I ask myself when evaluating a patient with cholangiocarcinoma is, can I completely remove the tumor? And the reason why is because our ability to completely remove the tumor really impacts a person's ability to live a long term or a long time. Specifically, if we're able to completely remove the tumor, sometimes we can achieve a 5-year survival rate up to 40%. Whereas if we're unable to completely remove the tumor, the survival rate at 5 years is quite low. So if I don't think I can remove the tumor in its entirety, I generally do not offer surgery because I'm not going to help that person. So one of the early questions, again, I ask myself is, can I successfully remove the tumor to provide them that long-term survival benefit? I'm going to focus on intrahepatic cholangiocarcinomas first. Again, these are tumors inside the liver. And I like this practice guideline. It was published in Hepatology in 2023. It's a bit dated. I just like it because of its simplicity. I'm going to focus on people who have what's considered resectable intrahepatic cholangiocarcinoma. It means it hasn't spread beyond the liver. It tends to be folks who have just one lesion within the liver. And the guidelines recommend that if they have resectable disease, they should undergo surgery, and they should undergo some form of systemic therapy after surgery. And in the United States, that's typically in the form of capecitabine, an oral medication. This is a case of a patient who presented to my office with a biopsy-proven intrahepatic cholangiocarcinoma. This is their CAT scan. And you can see here on the left side of the liver is this mass. And again, it was biopsy-proven as an intrahepatic cholangiocarcinoma. We have a tumor board, just like many other institutions. We reviewed her case at that tumor board, confirmed that there was no evidence of disease outside of the liver or outside of that spot. She was very functionally fit. She walked 3 to 4 miles a day. She had a perfectly working liver. I routinely ask folks, women particularly, to get a mammogram, endoscopy, and colonoscopy when they have an intrahepatic cholangiocarcinoma, just to make sure it's not a tumor that has spread from another site, and that was negative. And after talking to her about the different treatment options that exist, we both agreed that proceeding with surgery was the right thing to do. And so this is just a brief video of the surgery that I performed in her. This is called the robotic left lateral hepatectomy. The first part of the operation is removing the lymph nodes that are associated with that area of the liver that's close, that area that's close to the liver. Sorry, I don't know if the videos are playing. Let's see. So the first part of the operation is removing those lymph nodes. The second part of the operation, here I am, as you can see on the bottom of the screen, I'm getting around the main blood vessel that supplies the left side of the liver before clipping it and dividing it. And then lastly, I'm removing the part of the liver that contains the tumor so that I can completely remove it from this person, and I was able to do that successfully. Here you can see this 5-centimeter intrahepatic cholangiocarcinoma with negative margins. This is us walking together in the hallways. The first day after surgery, her smile was literally that big. And then the second day after surgery, she was able to go home. Despite having a successful operation, I always ask myself, what do we do after these operations? And as Dr. Hewitt had mentioned, and as previously discussed as well, you know, cholangiocarcinomas, they do have a high propensity or a high risk of recurring, and usually that recurrence happens within the first 2 years. And so, as I had mentioned, the guidelines do recommend that folks who undergo surgery, no matter how successful it is, that they get some form of systemic therapy after surgery. So in the United States, that usually is in the form of capecitabine, and that's based on a study that was published in 2019 that looked at giving capecitabine in the adjuvant setting, meaning after surgery, to people who had cholangiocarcinomas. Now, this study included folks who had all different types of biliary tract tumors, intrahepatic cholangiocarcinomas, extrahepatic cholangiocarcinomas, gallbladder cancer. And big picture, what they showed was in this solid red line that the folks who received capecitabine after surgery lived longer than the folks who did not. Now, the time difference is better than what we had previously. There's still much work to do, but the folks who received the oral chemotherapy had a median survival of 51 months, whereas those who didn't had a median survival of 37 months. So a significant improvement in overall survival, but still tons of work to be done in this field. And working with Dr. Kristen Spencer and other oncologists here in NYU as well as outside institutions, we're constantly trying to move that part of the field forward. Now, I had mentioned if somebody presents with resectable disease, they should probably undergo surgery, but sometimes people don't want surgery. And this is an example of a 79-year-old gentleman who actually had lung cancer recently, underwent a successful surgery for that, and then presented with the biopsy-proven intrahepatic cholangiocarcinoma that was localized to the right lobe of the liver. So unlike the previous patient, this is on the right side of the liver. And I thought I could do a beautiful, excellent operation removing the right side of the liver robotically, and they'd be home in 2 or 3 days, and they weren't interested in that. The most important thing to them was spending time with their family and playing golf. And so I presented his case at our multidisciplinary tumor board. Histotripsy wasn't as available as it is now at that time. So what we decided on was to pursue non-surgical treatment options. So based on a publication that came out a few years ago, we decided to offer this patient something called Y90. It's a form of radioembolization and chemotherapy. So working with our interventional radiologist, he underwent Y90, where these spheres that can emit radiation are delivered via a blood vessel that can target the tumor, as you see on this side of the screen. So they're able to selectively deliver these radiation-labeled spheres to the tumor. And then on his first surveillance imaging studies, post-treatment, he had a complete treatment response to the Y90 and no evidence of metastatic disease. And the last that I spoke, he is enjoying time with his family, and he's really enjoying his time on the golf course, although not right now because it's quite cold. I'm going to switch gears a little bit and talk about the role of liver transplant for intrahepatic cholangiocarcinoma. It's not commonly done, but there is a path for transplant for these patients. And in the United States, the criteria is generally you have a biopsy-proven intrahepatic cholangiocarcinoma. It could also be a mixed cholangiocarcinoma. That means that it has some features with what's called HCC, which is the more common liver cancer. And intrahepatic cholangiocarcinoma, you have to have cirrhosis. It can't be resectable, meaning it can't be amenable to just simply removing the tumor. It has to be 3 centimeters in size or less. And they have to have received either some form of local regional therapy, whether it be Y90, histotripsy, or systemic therapy, so some form of chemotherapy. And then they have to be fit for surgery. Transplant is a big operation. They have to have strong social support. They have to have people invested in helping them get through the recovery phase and also helping them take care of themselves while they're recovering from the transplant. And they have to be able to remain on lifelong immunosuppression. And this option really came about from a study that was published in 2016 by my mentor, Dr. Gonzalez Aposocian, who looked at people who underwent transplant for a variety of reasons who happened to have cholangiocarcinoma. And what they found was in folks who had cholangiocarcinoma that were 2 centimeters in size or less, they did a lot better than those people who had cholangiocarcinomas that were greater than 2 centimeters in size or had multifocal disease, meaning that they recurred less often. So in this line, you see that their rate of recurrence was significantly lower than those who had advanced disease, and they lived longer than those people who had advanced disease. So again, in the United States, there is a path for transplant for unresectable intrahepatic cholangiocarcinoma. It has to be small, so generally less than 3 centimeters. You have to be cirrhotic, and you have to have stable disease in response to some kind of treatment. More recently, there are data demonstrating that more advanced intrahepatic cholangiocarcinomas can be treated with liver transplantation. So this is a study that was published in 2022 that came out of Houston Methodist. It's a single center study, but these patients had advanced intrahepatic cholangiocarcinomas. So intrahepatic cholangiocarcinomas that were greater than 2 centimeters or multifocal. And in this study, there was a total of 37 patients who were listed for liver transplant. 18 underwent transplant and 14 did not. And the big picture is those patients who underwent transplant lived longer than those patients who did not undergo transplant. Now, all these patients were on systemic therapy for at least 6 months. And at the time, it was Gemcis, and then they incorporated other medications like durvalumab, as well as other targeted therapies for any targetable mutation that the patient may have. So big picture, for patients who have advanced disease, there is a pathway, if it's only limited to the liver, where in combination with systemic therapies, local regional therapies, and if they have a favorable response, there is a potential usage of liver transplantation as a curative treatment option for patients who have advanced intrahepatic cholangiocarcinomas. I'm going to switch gears a little bit and talk about perihilar cholangiocarcinomas. Again, this is the more common type, and this is the one that exists above the gallbladder or above the cystic duct and below where the bile ducts kind of enter into the liver. And again, as Dr. Hewitt had mentioned, this is a high real estate area and is one of the more challenging operations that we as surgeons face. And this is an example of somebody who I saw. It's a 51-year-old gentleman who presented with new onset of jaundice to the emergency room, otherwise completely healthy. And this is his CAT scan. And just going to summarize it, he had a perihilar mass, so in that area that we're referring to, that was localized more to the right side, and he underwent the typical kind of treatment for this. So the first step is really to decompress the bile duct or to relieve that obstruction to help resolve that jaundice. So he underwent an ERCP and he had stents placed. During that time, he had biopsies of the bile duct mass performed and it was positive for cancer. And he had an excellent performance status. And this is just an example of his MRI to show you the level of obstruction and the dilated bile ducts that he had when he presented. So using that kind of same paper that I had mentioned before, folks who have resectable perihilar cholangiocarcinomas, no evidence of extrahepatic disease, no evidence of metastasis, excellent performance status. The guidelines do recommend surgery up front followed by afterwards systemic therapy. And so the one thing that I would stress about this operation is that even in the best of hands, surgery for perihilar cholangiocarcinomas is super hard. So this is a paper that was published in 2021. This was a paper that looked at the outcomes of 708 quote-unquote straightforward patients at 24 high-volume centers. And in these people's hands, the overall complication rate for these patients was 80%. And 58.1% of these patients, or 58% of these patients, had some form of a severe complication. The rate of in-hospital mortality was 5%. The rate of mortality at 3 months was 7%. And complications such as bile leaks or liver insufficiency was close to 20%. And this is in the best of hands. I don't say this as to scare anyone, but it is I say this as a realistic view that for people who have resectable disease that undergo surgery, it is a challenging surgery. And it's important to go into that knowing that. And part of the reason why it's a challenging surgery is because the bile ducts are obstructed. And for folks who've had biliary obstruction, it's difficult. You know, you suffer from something that's called cholangitis, where you have recurrent belly pain, you have recurrent fever, you have recurrent jaundice, you have recurrent itchiness, and people get sick from that. And you can't be aggressively treating somebody if they're actively sick with cholangitis. So cholangitis delays people getting surgery, it delays people getting definitive care. So one of the first things that we have to do is relieve that obstruction, relieve the blockage of the bile duct. And oftentimes that's done with stents, and stents come in all different types of flavors. Sometimes stents are placed outside of through the skin, through the liver, and then across the area where the obstruction is. Sometimes people can do it endoscopically. They place an endoscope and they place a stent from the inside. In Asian centers, people will place these stents, which is called a nasobiliary drain, and folks will walk around with a tube kind of hanging out of the nose, and it's one of the preferred methods of draining the biliary tree in Asian centers. But the point is you have to relieve the obstruction. Now, I'm just going to briefly kind of mention this as one of the things that we think about as surgeon is which side to drain. It's a little counterintuitive, but if I had to choose a side that needed to be drained when I'm preparing someone for surgery, it's actually the side that's least affected by the tumor. I want to drain the side that I intend on keeping with the person or with the patient if I perform the operation. So if they have a right-sided tumor, it's ideal if they can drain both, but it's best if the left side of the liver gets drained, because I want the left side of the liver, if that's the side that I keep on, that I intend on keeping, to be as healthy as possible when I perform the operation. One of the things that we have to think about with liver surgery is leaving the person with enough liver to function in a normal physiologic way. We can do a perfect operation, we can remove the tumor, but if I don't leave you enough liver behind, you will unfortunately develop something called post-op or post-surgery hepatectomy liver insufficiency, meaning that the liver that I've left behind isn't big enough to work for your body's needs. And unfortunately, these folks will develop ascites, maybe not as dramatic as this person, but fluid accumulating in the belly and jaundice. And this is some of the complications that I alluded to in that paper that was published in the Best of Hand Surgeons. So one of the things that we'll routinely do, and some people have actually undergone this, is something called portal vein embolization. And we'll work with our interventional radiology colleagues to plug up the blood vessels that feed the side of the liver that we intend on removing, so that the other side of the liver can grow and provide the function necessary after surgery so that person can live a fruitful life. So just going back to the patient that I had presented who had come in with jaundice, this is his surveillance imaging. Thankfully, he has no evidence of recurrent disease, and he's 3 years and 16 days out from his surgery. We were able to get negative margins, and he's doing quite well. Just like I previously had mentioned, the standard of care in the United States today is for patients who have perihilar cholangiocarcinomas who do receive surgery to get some form of adjuvant therapy, and again, it's going to be capecitabine. In the last couple minutes, I am going to switch gears and talk about the role of transplant for perihilar cholangiocarcinomas. This is something that the Mayo Clinic has pioneered. There's the Mayo Protocol, and has really informed how we treat patients with transplant for this disease process. And in the United States, the access to transplant is dependent on a couple things. You either have to have a biopsy-proven or a cytology-proven perihilar cholangiocarcinoma, but oftentimes we're not able to get that. And so some patients are able to get transplant if they have what's called a malignant appearing stricture on an MRI or CT scan with an elevated tumor marker such as a CA19-9, or they have a malignant appearing stricture and suspicious cytology or suspicious fish, or perihilar mass with imaging features suggestive of a cancer. Now, the Mayo protocol is tough. It's really revolutionized our ability to treat this with transplant, but it's a tough protocol to undergo. These folks have to undergo external beam radiation while receiving systemic therapy, followed by intraductal brachytherapy while receiving oral capecitabine. And if they don't have any evidence of progression of disease, they have to undergo some form of operation to make sure that there's no disease outside of the liver, and we'll routinely examine the lymph nodes to make sure that there's no spread to the lymph nodes. And if all of that comes back negative, then they are a candidate for a liver transplant. As I mentioned, it's a tough protocol. There's approximately up to a 50% dropout rate. But the good thing is if you're able to undergo a transplant, we can achieve a 5-year survival rate of up to 60%. So in conclusion, the surgical management of cholangiocarcinoma is dependent on its location, and resection or transplant in conjunction with other cancer treatment options can improve survival in patients with cholangiocarcinoma. Thank you.
So he was going to have the task of introducing our next speaker, but I took his speaker's notes, so no, you're fine. Go ahead. No, you're good. You're good. So, okay, so together we're going to introduce Dr. Flavio Rocca, who's one of our invited guest speakers. Go ahead.
He's the Henninger Professor and Chair of Surgical Oncology at Oregon Health and Science University, and serves as a physician-in-chief of the Knight Cancer Institute. I've actually heard him on Spotify, one of the podcasts. It's a great talk. He trained at various institutions, including the Brigham and Women's Hospital and Memorial Sloan Kettering. His work focuses on complex diseases of the liver, bile ducts, and pancreas. He also serves as a surgical lead of the International Cholangiocarcinoma Research Network. And I wanted to welcome Dr. Flavio Rocca. Please and thank you.
And Dr. Torres Hernandez will be back just in a moment for our case panel and questions, so don't worry if you have questions for him.
Well, thank you very much, Dr. Spencer, and I'd like to thank the organizers and the Cholangiocarcinoma Foundation for holding this symposium. As I understand, this is the first one in New York City, so great to have all of you here. For me, it's kind of a homecoming. I think, as mentioned, I did my fellowship here at Sloan Kettering about 15 years ago, and I've been in Portland, Oregon, where we actually hosted the first Portland Regional Symposium last year in the summer. So we've had some great talks now about different therapies, so I want to focus now on another potential option, and this is for HAIP, which is the hepatic artery infusion pump. And so, this is great because I've already had some of these introductory slides given by Belinda and by Dr. Torres Hernandez, but really this is when we think about local therapy. As a surgeon, we always want to cure disease when we can take it out. But there's going to be a certain subset of folks who are locally unresectable. So that doesn't mean we can't help them, right? Our goal at that point is to try to control the disease progression. And I'll hopefully make the point that if we can at least preserve the liver function, we might not be able to cure the tumor, but we can keep the patient feeling well and alive and doing all the things that they want to do. And then, for the unfortunate folks, I think, that have metastatic disease that's unresponsive to therapies, can we palliate those symptoms? Can we make somebody's life a little bit easier? And so these are kind of the options when you look at the spectrum of local therapies. Of course, we had a nice discussion here on both resection and transplant. We haven't covered as much the tumor ablation technologies. Dr. Brock Hewitt did speak about histotripsy, but there are others that are available. And we're going to get into a little bit of the catheter-based kind of hepatic artery-directed ones that I've listed there. And then the other one that we haven't covered is obviously radiation, which comes in also several forms. But to give you the perspective, when I was a fellow here in New York, this was a nice paper that was published by one of our Japanese colleagues looking at what the spectrum of intrahepatic cholangiocarcinoma looked like. And so this is a series of 238 patients from Sloan Kettering. And just at that time, only about a third of patients were actually eligible for a potentially curative resection. And I always say potentially curative because as surgeons, we're always, the first question the patients always ask me is, did you get it all out? And my answer is, I got everything out that I could see and feel, right? So I think that's that's the challenge in this disease. Because even though, we can resect the disease and tell patients that we got everything out, 62% will recur, and usually at about a 2-year mark. It's about 47% in folks who have a solitary tumor with no lymph nodes that are involved. But in those that have multifocal disease, even tumor satellites, more than one lesion, or positive lymph nodes, that recurrence is going to be universal, basically saying we know it's coming. What's interesting is the pattern of recurrence. So the majority of these recurrences, when they do occur, are in the liver itself. So there is something to say about either how the tumor is spreading within the liver and its dual blood supply. And over time, we've been able to kind of find some predictors. So obviously, we talked about the number and the size of tumors, but if there's vascular invasion, particularly to the portal vein, if it goes into the bile duct, and the main bile duct needs to be resected. But I think more and more we're learning about molecular markers, just as Melinda was talking about, that may make a difference. So I'm not going to get into the nitty-gritty details about how do we define resectability, but I think this is a little bit of a challenge in this disease because we have these criteria in other tumors, like pancreas cancer, for example. And what that allowed us to do in pancreas cancer is design specific trials for specific categories of patients. And so when I think about cholangiocarcinoma, these are the things I'm thinking about when I'm considering patients for surgery. So first, is it anatomically or technically resectable? So this is, just looking at blood vessels and CT scans. I then, of course, got to look at the patient themselves. Are they good candidates for surgery? Do they have good heart function, good lung function? What does their liver look like? And then as an oncologist, I'm always looking then at what are the biologic factors, the DNA, the markers, the CA19-9. And this is the part that's not quite well defined as far as in this disease as much as in others. So, the foundation last year, the meeting that Melinda mentioned, we got together with a group of experts from around the world to try to put cholangiocarcinoma into one of these buckets, right? So on the left there, you'll see sort of the absolutely, completely resectable. On the right are the clearly unresectable. And then what we're trying to find is this sort of middle ground, right? The borderline resectable, right? So these are patients that if we can get them onto some kind of therapy and kind of convert them from that middle ground, and these are some of the categories we came up with. So where does hepatic artery infusion therapy come in? So to tell you the truth, it's not novel technology. And so I gave this talk at ASCO last year about the Back to the Future. And of course, for those of you who are familiar with the 1980s and Marty McFly and Doc Brown and the DeLorean, this technology was started in the 1950s. I mean, the pump is, you'll see there, it looks like the size of a hockey puck. It's got a little nipple valve at the top. It's got a Freon chamber with bellows, and you fill it, and then it just empties. And actually, a lot of this technology was pioneered here in New York. This is kind of what it looks like in the patient. So you'll see the diagram there behind me. The pump gets put just under the skin in the belly, and the catheter is then tunneled into an artery called the gastroduodenal artery that can then perfuse both sides of the liver. The reason this technology works is because the normal liver cells get their blood supply from the portal vein and the hepatic artery, but the tumors only get their blood supply from the hepatic artery. So that's why if we can deliver the chemotherapy to the liver through the hepatic artery, it'll treat the tumors. And you'll see these pictures in the operating room, how we're using both dye, fluorescent dye and just blue dye to make sure that one, the chemotherapy is going to get to the whole liver, but then two, in panel D there, making sure it's not going to go outside the liver, because what we don't want to do is give chemotherapy to the duodenum or the pancreas or the stomach. And so we do all sorts of, preventive techniques in order to make sure that. So we do in the operating room, we do the dye test, and then when patients are still in the hospital, we do this nuclear medicine study where we inject a radio-labeled tracer in the pump and then put them through the scanner to make sure, again, that there's nothing lighting up in the pancreas. And of course, the other piece of this is making sure we can take care of, local complications, just like wound infections, pump seromas, catheter slippages, and so forth. So even though this technology is not new, there are some nuances to it. Now, why does this work? So this is actually a more modern series from 2019. This is a study done both at Sloan Kettering and WashU in St. Louis. They pooled their patients, patients who had unresectable intrahepatic cholangiocarcinoma, and they received the combination of both systemic therapy through the vein as well as the pump chemotherapy. And you can see there that their progression-free survival was actually quite long, and even their overall survival was longer than what was expected. Interestingly, those that had an IDH mutation, which we know we have a drug for, actually did better than those who did not. And so where does this fit in the landscape? And so this was a recent meta-analysis that was done by Dr. Kortkamp, who was actually a fellow at Memorial 2 years after me and is now back in the Netherlands. So he looked at the body of literature that existed at the time here in 2022. There were 9 studies. Some were prospective trials, some were retrospective studies. But he was able to discern that with this technology, if you actually take that same population of patients, the unresectable intrahepatic cholangiocarcinomas that are not metastatic, if you just got systemic chemotherapy, your 3-year survival was around 0. But if you had the pump chemotherapy, that jumped up to about 36%. So we really kind of were able to move the needle in this very specific patient population. So he also published a couple years ago at ASCO the prospective Pump 2 trial. And again, this looked at that same population. You can see here the waterfall plot. And for those of you who are not familiar with the waterfall plots, it's just a plot of response. And so each bar is a patient. And then you'll see the tumor either getting bigger, the bar going up, or the tumor getting smaller, the bar going down. And you can clearly see here that the majority of patients had the tumor size go down or shrink, some by a significant amount. But the majority, 88% of patients, had their disease controlled or stabilized at about 6 months. And of course, then when you compare that to the historical chemotherapy survival, was much better, as we discussed, that kind of 33% versus 33%. One of my colleagues, Dr. Sky Mayo, who's on faculty with me, wrote a very nice editorial accompanying this publication in JCO, and he used this very, I guess it's a Dutch saying about all things excellent are as difficult as they are rare. And the point of this talk is really to say that we talk about the nuances of putting the pump in and the complications and the management, and I'll go into why that's important. But as Dr. Torres Hernandez also mentioned, is that sometimes, we're not going to be able to necessarily take this out. You'll see this picture here. This tumor is very central by the blood vessels. But if we can control the disease, we can actually get a grasp of getting patients out of that cycle of cholangitis and having stents and tubes. And so again, but just by being able to keep that tumor in check, can we prevent folks from coming in and out of the emergency room or being admitted to the hospital? Now, where does molecular therapy fit in? Again, we're sort of just at the cusp of this. Our colleagues in medical oncology have done a great job to kind of lay out the landscape in the advanced setting, but where does this fit in the localized setting? And so again, you'll see here, this is a retrospective study from, again, Uptown, looking at patients who have negative lymph nodes, the N0s. The top bar there is resection, the middle bar is the pump, and the lower bar is the systemic therapy. So you'll see if you have negative lymph nodes, resection really appears to be the top #1 thing. But all of a sudden, if you have positive lymph nodes, resection and the pump are completely overlapping. So there's something there about that being maybe a little more aggressive disease that the pump can perhaps control. In fact, even in that IDH mutation status that I told you about, right? So in the patients who have negative lymph nodes, it seems to be protective. But again, once you have positive lymph nodes, the curves are sort of overlapping. And certainly if you have other high-risk mutations, so particularly they looked at this combination of KRAS, P53, and CDKN2A, that particular signature, no matter what you do, the biology is pretty advanced. So the question is, again, can we spare patients from sort of ineffective therapies at that point? Including multifocal disease. I think I mentioned to you earlier that, the best outcomes we have as far as recurrence is patients who have a single tumor. And that appears to be where resection, again, has the best impact. But when you have patients that have 2, 3, 4 lesions, it's possible that the pump and resection are completely overlapping, meaning that, again, can we control the disease in the liver? We might not necessarily cure it, we might not necessarily remove it, but can we keep patients going and going in a way that they want to live? So just to shift gears a bit, in Oregon, this is how we do the pump. And you can see it takes a village, as I say, it takes a team. And so, while the surgical oncologist and the medical oncologist are in the middle, and I didn't mean to put the surgeon above the medical oncologist, I was going to let you know, Dr. Spencer. You'll see all the other team members that are around, including our radiology colleagues, pathology, GI, nursing, social work, infusion. And I got to tell you, when we do an evaluation, the note that I look the most closely at is the social work note, because that is something that we really want to make sure patients and family are engaged, they're committed and dedicated to this. And so these are just kind of our rough, indications and contraindications. And so really you'll see on the left there, pretty broad range. And again, these numbers are just, I had to pick a number, so they're not, hard and fast. But it's really more about having a good liver, having obviously the tumor and being fit for an operation. The tougher ones are the contraindications. So, because the pump only treats the disease in the liver, folks that have significant extrahepatic disease. And what I mean by that is you have a few lung nodules, I'm going to give you the benefit of the doubt. You have to have a certain arterial anatomy so that, it's conducive to the pump going in. We really want to avoid, patients that have already pre-existing strictures per se, because one of the toxicities of the pump is biliary strictures. And so we need to make sure that we're not causing those. And then the starred ones are the tougher ones. Dr. Hernandez mentioned about the Y90 treatment. And so again, folks that have had radiated livers, those livers may be already injured, so I don't know how they will tolerate pump therapy. But one of the biggest challenges for us, at least in Oregon, we're a very rural state, great distances, is the inability to travel for the fills. Because the pump, unlike your port, has to get filled every 2 weeks with drug or heparin. So patients have to come back to get that treatment. So this is the landscape of our program. So we started, Dr. Mayo started our program in 2015 before I arrived there. And you can see in blue, this technology is also used for metastatic colon cancer. That's probably where the majority of the data lies. But in orange, you'll see how we've been putting more and more of these in cholangiocarcinoma patients. And in fact, as of last year, we've actually put in more pumps for cholangiocarcinoma than for metastatic colon cancer. And again, I was to show you, for those of you who are not familiar with the map of Oregon, I know it's on the other side of the country. Portland sits up at the northwest corner, right on the border with Washington, but it's a very big state. It's about 6 to 7 hours to get down to Medford, which is on the border with California. And so, again, for ease of patients to travel, we've set up these kind of almost like gas stations, sort of fill stations, that we can have patients come in if there's a storm, if the pass is closed, so they can get their pump filled in Medford or in Bend as well. And so again, we're trying to learn more about this. So this is, again, one of the trials that we did, the initial trials. This was 5 patients that we enrolled in a prospective study looking at induction chemotherapy with the pump therapy, and this was presented at ASCO a few years ago. And I don't need, we need you to read all the details, but to show you that we did this and we showed that it was safe. And you can see kind of the treatment course of the patients there on the left. Of course, as everything gets better, the chemotherapy got better. So now, as you know, the standard of care is the chemoimmunotherapy. And so now we're launching the second trial for this, which is using the modern chemotherapy regimen with immunotherapy. And the idea here, again, is this therapy safe to be given with the pump? And so this is the trial that we're just about to open in order to answer that question. Well, where do we go in the future? Well, I think what we need is a randomized trial. And so this is the ongoing national trial that's at 5 centers, including, again, here uptown at Sloan. And this is going to compare, then, systemic chemotherapy with the pump or without the pump. And so, of course, these trials are hard to write, even harder to execute, especially in a rare cancer population. And as I told you, the target keeps moving. So we'll see how this is accruing. But again, if you have the opportunity, this is the way we're going to move the field forward. One of my dreams is to actually, we talked about, what's the treatment after surgery. When I was a fellow here, I actually wrote a trial to give adjuvant pump therapy. So patients had surgery, then they get the pump therapy afterwards. We wrote this trial in 2011. We actually opened it at Memorial and just didn't have enough patients to accrue. But I do think this is the future. And so because now we have many more hepatic artery infusion centers, so you'll see here the growth, from the 70s now to present day. We have over 50 centers. We actually have a consortium. There's a research network, so there should be a pump center near you, including some international centers as well. So I think in summary, I just want to, highlight that, resection really remains the gold standard. I think you've heard that now more than once. But we do need to be a little more standardized in the definitions that we have for patients who are not clearly upfront, to really kind of figure out what kind of therapies are they eligible for, and can we convert them to surgery at some point. And for that, we're going to need more trials, right? We need these trials for either preoperative therapy or perioperative therapy. And I do think the pump plays a role both for multifocal disease, unresectable disease, and perhaps down the road as a bridge to transplant, as we heard. So in the last few minutes, I just want to give you, for those of you who are not familiar with Oregon, kind of a little sense of our institution. We're the only NCI cancer center between a large area of Seattle, Sacramento, and Salt Lake City. We have a huge surgical residency, the largest in the country. And our faculty there cover all sorts of different cancers. As a physician-in-chief, I'm really looking forward to opening our new cancer hospital. So So this is the tower that I think the foundation, when they came to visit, got a chance to tour. So this building is completely dedicated to cancer. We're opening 4 inpatient floors, more beds. Coming out of the pandemic, as you may have known here in New York, especially, there were no beds, there were no emergency rooms. So this is really overdue. Dedicated imaging space, ICU, urgent care, and so we are frantically opening this building in a couple months. So I'll leave you with can we play the video? I think you have to activate it.
Cancer is the biggest fight in the history of the world. We will win. It will be hard. We're built for it. We care. Every breath of every second of every day. Great things come from the most unexpected places. Look around. We're surrounded by heroes. This is not a job. This is our life. Cancer does not discriminate. Neither will we. Bold moves only. Cancer doesn't see us coming. This fight is and always will be personal. We know what we gotta do.
Thank you very much. That was Dan Lanning from the University of Oregon who was narrating that for us. His wife had osteosarcoma.
I think we echo your sentiments here in New York, so thank you so much, Dr. Roca. Please feel free to have a seat, and Dr. Torres-Hernandez, if you want to come up, and I'd like to welcome Dr. Kareem Halazoun, who is the Surgical Director of the Adult Liver Transplant Program and Chief of Hepatobiliary Surgery here at NYU. His passion for liver surgery began early, inspired by watching physicians save lives and shaped by witnessing one of the world's first auxiliary liver transplants. Today, he specializes in treating liver and bile duct cancers, including cholangiocarcinoma, using advanced surgical and transplant approaches, always with a focus on compassion and personalized care. So he is actually going to be the moderator we have a combined case presentation style where they'll go over some cases they've actually each experienced and treated, and then that will be followed by a Q&A. So we have some time to catch up on our agenda. I know we're a little bit behind, but please prepare your questions, and I will hand it over to Dr. Halazoun.
Do you want to? Yeah. Sure. I think, hi, thanks for inviting me. It's a pleasure to be here and see you all. Great presentations. And I think Dr. Roca has some cases for us that we will start with.
I always love presenting cases to my transplant colleagues because we're like otters, holding hands at night. So this is a couple cases just to give you a kind of a flavor of how we use some of the stuff that we talked about. So this was a young lady who presented to us, actually presented I think in Arizona in March with some pain, typical kind of non-specific symptoms, had some minor surgeries in the past, and we can see here what it looked like on the CT scan. So this is obviously for your benefit, but also for our transplant surgeons. And you'll get a sense of this very large kind of central lesion in the middle of the liver. All right, so this is the rest of the workup. You'll see the tumor markers really not greatly elevated. Had a biopsy, positive for adenocarcinoma. It was microsatellite stable, so it was not a immunotherapy candidate. And then the mutation analysis had this PIK3CA mutation. So just curious from the perspective of, what would they do here at NYU for this patient?
Yeah, it's a large central tumor. It's a little tricky because we couldn't see the blood vessels, but just kind of in its location and given its size, it's something that we would probably not advocate for upfront surgery, even if it's resectable. There's some data out there that large tumors, even if resectable, have a higher recurrence rate. So just given that size, given its location, and I'm imagining its relation to the blood vessels, that it's probably not an upfront resectable tumor, I would advocate for, I guess technically should be induction chemotherapy and then see the response.
Yeah, I agree. The one thing that we would do here, and it's a little bit, we would get a PET scan just to see if there's metastatic disease that's obvious, it would change the prognosis, right? Yeah. And it would help us prognosticate, and maybe decide whether local regional therapy in the form of, we don't have a pump program here for various reasons, but we don't have a pump program, whether local regional therapy in the form of radioembolization or a pump are worthwhile, because if there's 20 lymph nodes, then it's probably not, right? So.
That's a very fair point. So yeah, so like you mentioned, so patient was actually seen initially outside. So started on standard of care, triple chemotherapy, the chemoimmunotherapy, gemcitabine, cisplatin, and durvalumab. Because of sort of the advanced nature, was planned for sort of 6 months, and then sort of was restaged. And you can see here, the tumor. There's a little more vascular information for our surgeons here. I don't know if they get a sense now. Is this resectable now?
I mean, it looks like you're pointing at metastatic disease within the liver of the tumor, or are those...
Correct, yeah, those are satellite lesions.
Yes. So, could you take it out? Yes. Is it the right thing to do? Probably not. Those satellite lesions portend a worse prognosis. Technically, can you do a central hepatectomy here? Possibly, but, examining this further, with satellite lesions, bilobar satellite lesions, the prognosis is bad. So, if we operate up front, or even after 6 months of chemo, most likely outcome will be recurrence. Your data that you presented before, 62%, this is probably upwards of 62%, probably in the 70 to 80 to 90% range. Almost 100% will recur. So, probably not surgery here. Now, putting my transplant hat on, if you can maintain this disease for another 6 months without progression, this is the kind of patient that at Houston Methodist, where they have a big transplant program, would get transplanted. We've done a couple of these. Could get a liver transplant if they don't progress. The tumor shrunk. It's responded beautifully.
So she did have 6 months of chemotherapy. So I think, according to Dr. Torres Hernandez, that would be a candidate for multifocal disease.
It's. I think. Go ahead. But
no, I mean, it's tricky, right? Because it's a single center study. Methodist does have their protocol. There hasn't been other centers that have published their data with regards to transplanting these patients. But as chemotherapy, immunotherapy advances, we are seeing these patients that have some kind of response. They don't develop extrahepatic disease or disease outside the liver. And then we're asking ourselves what to do, right? Which treatment options are they best served by? And quite frankly, we don't know. We know that if we can get them to transplant, replace the liver, and they don't have extrahepatic disease, we know they're going to do well. But that doesn't mean that other treatment options aren't going to be good for that patient too. So it's really important that these patients are seen at centers that have these various treatment options, transplant, local regional options, pump programs, so that we can try to figure out which patients is best served by which. But if they don't progress on immunotherapy or on chemo and immunotherapy and it's liver-only disease, according to the protocol that they have in Methodist, they may be a transplant.
6 months is too short.
Yeah.
I think 6 months is too short, right? So for us to take someone to transplant at 6 months, I think we all agree as, cancer experts in cholangiocarcinoma, 6 months is probably too short. And chemoimmunotherapy, especially immunotherapy, we're going to have to stop the immunotherapy before the transplant for a period of time, because because we have to immunosuppress the patients, that's going to be a problem. If you stop the immunotherapy, what's going to happen? Is the tumor going to explode? So that's a problem. So adding radiotherapy, adding a pump, adding something here, and giving another 6 months to watch, to make sure, I think is the appropriate thing before transplant, if transplant isn't even an option.
Well, perfect. We agreed with you. And actually, I'm blessed now to have a team of transplant surgeons where we do have a protocol at Oregon for both intrahepatic and perihilar, like you mentioned. So so we did a laparoscopy. So this is one where you may have heard, this is the kind of the keyhole surgery where, we kind of just put a camera in and look around. We do this sort of either in conjunction or instead of the PET, for example, to look for that kind of occult disease you were mentioning. Put a pump in place, again, and the hope was to either shrink the tumor more so we can get to surgery, or potentially, as both the transplant surgeons mentioned, can we get this tumor sort of controlled by another period of stability, maybe take the patient off immunotherapy, right, and see if they could be a bridge to transplant. And so had a pump study, it was great, started the FUDR, and then started systemic therapy for about 7 weeks, and then was restaged after 2 months of therapy. And you can see the response there. So the tumor is clearly smaller, still in the middle of the liver. You can see the catheter kind of coming into play there. All right, gentlemen, what now?
So this is a bit tricky, and obviously I'm only seeing, CAT scan with not a lot of time, but I just
Yes,
want to
the
review
standard
again.
disclaimers aside.
No. I say this because it seems like there's some changes to the liver. Like I see the caudate becoming bigger, the left lateral segment of the liver is becoming bigger. So even though these medications and this pump is obviously doing a great job with controlling the tumor, it seems like the liver is suffering a little bit and starting to develop some chronic changes. And at some point, there may be a time where the liver cannot tolerate more treatment. So this is where I think, again, if they don't develop any disease outside the liver and they're starting to develop consequences of chemo-induced steatohepatitis or liver insufficiency because of those medications, perhaps this is where we do begin talking about transplant. If there are other treatment options, SBRT, Y90, my understanding, just talking to the different teams, Y90 is probably a bit better for these larger tumors than SBRT. Maybe the SBRT folks will argue with me with that. But I think with those chronic changes that I'm starting to see in the liver, if we can continue to control it but start talking about transplant, that would probably be the way to go.
So just looking at the scan briefly, it looks like the only option is the left triseg here because both arteries are involved. That's a big operation, with almost always a reasonable liver remnant, but after this much chemo radiation, you probably will hurt the patient. I actually want to disagree with my partner in that I think the patients that get referred to us when they have chronic liver disease changes from the pump and the chemotherapy are actually too late for transplant because at that point we're trying to and we've transplanted them, right? But there has to be oncologic control, and then we're trying to decide whether the tumor is stable and then whether the liver disease. And what are we transplanting for? Are we transplanting for chronic liver disease or for the cancer? I think this is if this patient's had, I can't remember the time period, but it's been a year
It's been about 9 months.
Yeah. I think, consideration for transplantation is good here. I would biopsy what the remnant liver is. I'd look at the tumor volume and decide whether it's good. Now, the use of you presented some compelling data on arterial pumps, and it's good, but one of the most compelling slides that I've seen, and this is why I love talking to you, because you're very thoughtful. In 10 years of having a pump, you guys have put in less than 100 pumps, right? Whereas, there's other places that put in 800 pumps a year, right? So that's a very thoughtful approach to the pump, and I think the pump makes the transplant more difficult no matter who you ask. So this is an interesting approach where we have to talk. It makes the surgery much, much harder and much more difficult to deal with. So I think this patient could get a transplant, but they could potentially get a resection. You still had satelloidosis, which was in the left, which was in the remnant, so I would worry about that in terms of recurrence rate.
I wasn't going to present you an easy case.
Well, the one thing that I wanted to discuss with you is, so the reason I get a PET up front is to decide transplant eligibility up front. Because if I see something that's going to potentially never allow the patient to get to transplant, I want to know. I don't want to rely on 6 months of chemo, then a lymphadenectomy in a potentially irradiated field where I may not get what I want to hear, and then transplant the patient and find out that there's 5 positive lymph nodes after the fact, in the specimen because it's all irradiated. And that's happened, that happens.
And let me play devil's advocate. So let's say, PET positive, do you biopsy to confirm, or is that good enough for you?
No, I mean, we do tend to try and biopsy to confirm if we can. I mean, obviously there's PET, false positivity, but, if the tumor's positive and there's 3 bright lymph nodes, the cat's out of the bag, right? So, I mean, most people would agree that that's metastatic disease.
All right. Well, so just to give it an inch of time. So, we did have her evaluated for transplant. And, I think we're cautiously optimistic that we will get her there. But in the meantime, we're just continuing the pump therapy. She seems to tolerate it. She's able to do her kind of snowbirding in Arizona as planned.
So the most important thing about these patients, which both of you have mentioned, is, we don't want to put a carrot on a stick here and say everybody's eligible for transplant, because the majority of patients who get put into the transplant protocols actually drop out. Like 60% of patients will progress at some point and won't be eligible for transplant. So we have to continually reassess these patients every couple of months, because if The problem with transplant is if we transplant someone and they have metastatic disease, within 2 to 3 months, the disease will be everywhere, and it's worse than doing what we're doing now, and they'll die very quickly.
Yeah, I think what you're highlighting, and hopefully the audience is getting, is that multidisciplinary care is really important. Because a lot of us, know our tools, so we know what our hammer is and what our drill is, but I don't necessarily know what they have in their toolbox. And so having that discussion about sequencing of therapies, which one to get first, which one to get second, what's the backup, what's the alternative, is really important. So into this other case real quickly, if Dr. Spencer will let me, and then I do want to leave some time for questions. So this is somebody who probably, you're more used to seeing in the transplant clinics, a patient with alcoholic and hepatitis C cirrhosis, had an incidental mass on screening, so kudos to screening for picking up a mass. You'll see kind of the liver function there, and the tumor markers, which are not terribly revealing, but on the scan, you'll see this lesion right there. So small lesion, I think as Dr. Torres Hernandez mentioned, that may be, a good thing. Really nothing in the lungs, but you'll see there very prominent portal lymph nodes, cirrhosis and suggestion of portal hypertension, and then the biopsy of both the tumor and a lymph node were consistent with cancer.
So this is a problem because you have portal hypertension and metastatic disease. We're not going to offer a transplant here.
Okay, so no transplant.
No transplant.
Okay, so let me give you the options here. So these are the options.
So
I'll make it easy.
Okay, if you're asking me, I would go with a combination of 1 and 2 in the form of systemic and 190. The tumor's in a good place where you can get a radiation, effectively the same as a surgery by blasting that left lateral segment with radiation.
Can I ask your colleague and partner next door, does he disagree or
Yeah,
agree?
so I just want to confirm the lymph nodes were just, the prominent lymph nodes were periportal lymph
Portal
nodes.
lymph node.
Yeah, so,
But biopsy proven.
biopsy proven, right? So it's tricky, right? Because those are the lymph nodes that we routinely remove for surgery, and you probably can make the argument that surgery up front.
Portal hypertension.
But portal hypertension. So it's tricky because there are patients with portal hypertension that are, when well selected, can tolerate surgery depending on how much liver we, potentially remove. But I say this because even though those portal lymph nodes are in the basin that we would normally take out, we know that these folks are at high risk of recurrence. So this is somebody that I would probably shunt towards some kind of systemic therapy up front.
So you're going with number 1.
But with the caveat that this is where, we talk with our oncologists, what can the liver tolerate? Some people won't give systemic therapies with bilirubins greater than 2, bilirubins or bilirubin less than 4. So it really depends on what the liver can tolerate. And do they have to give, instead of the triple combination, do they give 1 medication or 2? So this is where I would talk with them. And
Maybe Dr. Harding can let us know. What would he do? One.
One alone, or would you combine 1?
He says systemic therapy for this game. Number 1.
I think with a single big node, right? We're not going to take out a 3-centimeter node and say we're going to do anything curative, right? But if we give chemo, we could potentially control the disease in the liver. It's because it's so localized and small. Now, if it was a gigantic tumor and we're going to have to take out half the liver, we wouldn't do it in cirrhotic. But
Yeah. So we went with number 5 because I gotta move a little thing. So we need more information. So I think we talked about mutations mattering. So we did send this for profiling, and this patient had what's called a high tumor mutation burden. So there were lots of mutations, 23, and actually had a HER2 alteration, which, as you'll see there in red, is clinically significant. You guys, anybody amending their answer?
I didn't notice that last point, but I agree with the molecular profiling.
We should get Jim up here.
yeah. I didn't notice number 5 either, to be honest.
Yeah. Great.
The complexity of the next generation profiling. What really you want to know. Yeah, for I think the sequencing component for high TMB and MSI high or mismatch repair deficiency, these are two separate things. Mismatch repair deficient or MSI high, loss of proteins that are necessary for DNA repair, right? And as a result of that, most of those patients have super high TMB, which means there's a number of mutations in proteins that then present their neoantigens to the immune system. And those patients across solid tumors are very sensitive to single-agent immunotherapy. And if I saw an MSI high or a loss of mismatch repair, I'd be very interested in giving systemic immunotherapy. Now, in this case, there isn't MSI high. I don't know what the protein status is for mismatch repair, but the TMB is high, and there is a less strong biomarker for response to immunotherapy, but would still lead me to use that at some degree. The HER2, it's hard to know, so I do gemcitabine.
Thank you.
But the conclusion brings you back to gemcis durva, right? Like whether
Correct.
you have it or not, you go back
Correct.
to giving the same combination.
So just to cut to the chase, so 7-cycle gemcis durva had stable disease. We did get a PET MRI, really minimal activity in the primary tumor, nothing outside the liver. And because of this question of portal hypertension, we did get hepatic vein wedge pressures that had a gradient of 3.
Right. So now I'd resect it.
Yeah.
Okay.
So, and this is what we refer. There's some patients who have cirrhosis and portal hypertension who can undergo surgery in select instances.
So this was the operation. So you'll see the tumor there. There was basically just a focal residual tumor that was 2 millimeters big. That's it. It really melted away, had all negative lymph nodes, ended up being a T1A. And the patient did well. And so the question then was what to do in the postoperative setting. So he's on, because the immunotherapy we think had such a good job, we put him on some maintenance durvalumab for a year.
Do you need it? Do we need the maintenance durva in a tumor that
I think
completely-
that's not an answered question, right? So, there are adjuvant studies that have just completed accrual. I think that will answer the question about immunotherapy afterward in an adjuvant setting. I think you could argue you did such a great job of controlling it with a lot of upfront therapy that you didn't need to do that, but I can understand the rationale behind it.
Yeah,
I mean,
we
but this
basically
setting is
extrapolated
rare.
from the Topaz trial because
Right,
we managed the
for
patient
sure.
sort of advanced disease.
But this is this is the kind of perfect patient, right? They come, they have potentially metastatic disease. You give them immunotherapy, and everything goes away. That's kind of rare, but it's great when it happens.
I think I'm getting the word. So I think we'll have a few minutes for questions if folks want to come up and ask our experts here.
You're one of the experts.
So just real quick background, I was not intra, I was extra. I had the 1.5 centimeter tumor down in the distal bile duct. Had a Whipple, so did the surgery 2 years ago, and then was offered the capecitabine adjuvant scenario. But I ultimately declined it, and I was looking at the same BILCAP study you put up, and it looked like the 5-year survival rates on those were not statistically significant. They did show slight improvement from doing it, but it was not statistically significant. So I ended up declining it and then ended up getting into an immunotherapy clinical trial for about a year after that. So 2 years, I'm hanging in there, clear scans, but you never know, right? It's 90% recurrence just on average. But just wanted to know your thoughts on that BILCAP study is getting aged now. It's from UK, it was like 2016 or maybe earlier. Are there better post-surgery intra or extrahepatic adjuvant options?
Yeah, it's a really great question, and I'm glad that you had such a great response after your operation. The importance of the BILCAP study was that it was the first study in the adjuvant setting that was shown to have an effect on overall survival. There are some caveats to it. So there's a lot of different statistical ways that we analyze data. And so what's called in the intention to treat trial or intention to treat arm, there was no statistical difference in overall survival between those who received the oral chemotherapy versus those who underwent observation. But in the per protocol analysis, meaning everyone that got enrolled into the study, they did show that difference in overall survival, and that's why it became the standard of care, because it was really the first study to show that advantage in overall survival, even if it wasn't perfect. Since then, there have been some studies, particularly out of Asia. S1, for instance, has been used in the adjuvant setting for cholangiocarcinomas, but it's still a work in progress. There are some adjuvant trials that are accruing, and hopefully we hope to see that data soon. But we do have clinical trials where we're enrolling folks in the adjuvant setting with chemotherapy plus immunotherapy to hopefully provide them better treatment options.
Yeah, I would add to that, and he summarized the findings very well. The BILCAP trial was highly, highly debated when it came out. And again, kudos to Dr. Primrose. Dr. Vale was actually very involved in it. It took 10 years to accrue patients to that trial. And it's a mixed population. So even though distal cholangios was the majority of patients, 33%, it included gallbladder cancer, perihilar, intrahepatic. So it's hard to pinpoint to whom it's most applicable to. And then since then, there's been other trials to see, well, can we escalate therapy? Because a lot of times we think more chemo will be better. But there was a trial in Korea, the STAMP trial, which looked at lymph node positive extrahepatic cholangiocarcinoma, comparing the BILCAP versus GemCis, and there was no difference. So, it's sometimes more is not better. Now, we do have some, pharmaceutical trials, companies that are putting together trials with new bispecific type of antibodies and treatments. So we're awaiting those results. And there is a large trial, the ACTICA trial, that accrued in Germany. We're waiting for those results as well. So again, this field evolves over time. It just takes much longer to get results because unlike breast cancer or colon cancer, there's not as many folks who are there for the disease. And a lot of them are in Asia, for example, as opposed to the US. And so we always think about, are some of the treatments that we do applicable necessarily? S1 that Dr. Torres Hernandez mentioned is not even available in the US. So. We've got another question there. Yeah.
Bear with me because I'm just getting my hands, and this is like way above my head medically to talk. So, but if there's and I'm trying to make it general for everybody to have an interest. If it's a very large tumor and it cannot be surgically removed, the first course of action then would be the systematic approach with the chemo and all of that. Why not do the Y90 or that pump system to shrink it right away?
So that's a really good question. And again, we have, in practice, we do that sometimes, but there is no really data that supports doing them at the same time for any of the cancers, actually. For the most common cancer that we deal with is hepatocellular carcinoma, where we do immunotherapy and Y90. And what we're seeing is the results of doing both immunotherapy and some form of local regional therapy are actually negative. They don't show a difference from doing one or the other. So we don't actually have the answer to that. But in practice, at least in our institution, if we think that it's reasonable to do both and the liver will tolerate it, we often do that because we feel like there, anecdotally, from our experience, there seems to be some synergistic effect from shrinking the tumor and giving chemoimmunotherapy at the same time. But it's not for everybody. Some big tumors, you can't get enough radiation in them to actually have an effect, and you can't put enough dosing in them to have an effect without actually hurting the liver quite substantially and hurting the patient and then preventing them from getting chemotherapy, which we know works in a certain percentage of people. So it's tricky. It's really individualized. It's really, really individualized. It's not that specific question is, while I know the intention is to generalize it, it's not generalizable. It's really per patient and what the vessels look like, what the interventional radiologists think they can do, what the radiation oncologists, and Dr. Roca showed a slide with everybody on it, and that's how we decide who to push to which kind of therapy.
If a patient has the hepatic nerve where the tumor is wrapping around the nerve and did already strangle some of the endings, would they still be a candidate to get the Y90 or the pump?
Yeah,
Yes.
Yes.
Thank you for your time.
Sure.
Yeah, I have a question about the preoperative evaluation. What's your feeling now as using laparoscopy, let's say, versus a laparotomy to basically evaluate patients for resection?
Yeah, it's a really good question, and in some ways a little philosophical. So here at NYU, our programmatic approach, as Dr. Halouzan had mentioned, was to get a PET scan. There's some data that PET scans can change the plan up to 30% of patients. And so not everyone agrees with that. Oftentimes, patients that we see are seeking second opinions. They go to other prominent institutions in New York, and the surgeons there don't feel a PET scan is important. And I kind of counter with them while saying, listen, if there's a 30% chance that the plan may change with the PET scan, then I think we should get the PET scan because I want to avoid an unnecessary surgery. I think there are times where we are quite confident that there is no disease outside of the liver, and if it requires a big incision to remove the tumor, then maybe we'll proceed straight with the laparotomy. But oftentimes, since we are doing the overwhelming majority of our surgeries robotically, that first step is looking around with the camera. So, we're able to do these operations robotically, and we're able to take a look around and make sure that there's no disease outside of the liver before committing them to the actual surgery. So sometimes we can do this with just 2 5-millimeter incisions, take a look around, and then proceed with the surgery robotically if we think it's a good tumor to remove surgically.
So, and I'll give you the West Coast perspective, because I think we have points where we, I think, agree, and then others that would not. So what we would do is, a good quality CT scan to me is the number one thing. A lot of times when I see folks that have been seen locally, they have sort of a general abdominal CAT scan, really hard to tell any details. So almost always we'll get sort of a multi-phase liver CT that'll have an arterial phase, a portal, venous phase, a portal phase. And then if I see any abnormalities there that I'm worried about, then I jump to the PET scan, right, to try to confirm something. But ultimately, PET scans can't detect things like peritoneal disease, meaning disease that's kind of on the lining of the abdominal wall on the inside, as you can imagine. So I laparoscope everybody. I think it's a very easy, it's a 10-minute procedure. You can look around. I do it before they get any kind of local therapy, so even before getting an embolization or a Y90, I think you gather information. I also do what's called peritoneal washings, which means I put salt water in, I wash the abdomen, and I collect it to see if there's microscopic cells floating there. And that way, the patient and I have a discussion to say, okay, look, we have as much data as possible now. Now let's talk about the different options that we can go.
I don't think that's
The question I would have is, why not, I seem to think that makes more sense, and why not just laparoscope everybody rather than take the risk of doing an open procedure and then, finding extensive disease that was not found on an imaging, therefore, subjecting the patient to, let's say, an abortive resection.
I think we do laparoscope. I think they're answering the question pretty much the same way in slightly different nuances. We get a high-quality CT scan already. We don't do the peritoneal washings, but we basically laparoscope everybody because everybody gets a laparoscopy before we decide what we're doing. Our approach has changed. We're like 75 to 80%. Even these big, massive tumors and massive resections, we're doing robotically. Even with vascular reconstructions, we've been doing them robotically. That's, just a tool. It's not any different than doing it open, but we try to look for evidence of metastases, and that's why Dr. Torres showed a case where he took the lymph nodes first, and so, oftentimes we send those and decide what to do. If things look suspicious in the belly, we take samples and we send them to pathology to figure out if these things are cancerous or not. And specifically, when we're looking at patients with transplant, we put a camera in a week before the surgery, take out all the nodes, make sure that they're all negative because we don't want to make a mistake.
And I think part of it is also practice patterns. I mean, here in New York, I know you can go to, probably 8 or 10 different centers. Out in Portland, it's kind of us. You can go up to Seattle or San Francisco, but we get patients from Hawaiian Islands, the Marshall Islands. And in those folks, the nice thing about the laparoscopy is I also put in the port. So I do all in one. They get one general anesthesia. They go home. They're already kind of got their access point. So again, a lot of it is also how, what region you are and what
also
kind of
access to PET and insurance. Yeah. I mean, it's all, yeah.
The one thing I do want to highlight, and this was kind of the point that I was trying to make, is liver surgery has changed quite dramatically in the past 10 years, 20 years. And fortunately, for me, I get to work with one of the true pioneering thought leaders in robotic liver surgery, and somebody who's able to do really complex liver surgery through really small incisions. And so oftentimes we're able to do that staging laparoscopy, look around, make sure that there's no evidence of disease, and still proceed with surgery using small incisions and avoiding that huge laparotomy that we used to have to do 10 years ago, 15 years ago. So I think with the advent of more advanced surgical techniques using smaller incisions, everybody's getting that quote-unquote diagnostic laparoscopy up front. The difference is whether you do it on the same day or next.
Last two questions?
Yeah.
I presented here 28 months ago with Klatskin tumor. Turned out to be stage 3 with perineal invasion, also one positive lymph node. Dr. Halazan did brilliant surgery. I'm very, very grateful. And my scans are clear, my blood is great, and I couldn't thank you more.
Great. What a fantastic way to, oh, we have another question, or, yeah.
So one final question. Thank you all. This is just incredible. This is one of the best things that we have, I think, is this model of what does this look like in terms of looking at a case and talking through the options. So my question comes from my experience as a layperson who's both been a caregiver to my late husband and is now the chair of the Cholangiocarcinoma Foundation Board. And what I hear in this presentation is something that my husband Steve and I experienced as he was going through his journey. And I want to pose this as maybe a final question of advice you can share with the patients and caregivers here in the auditorium. What I hear is what you've all touched on, is there are regional variations and approaches to disease. We have this great concentration of experts. Everyone has their approach, their philosophy. We see that play out through individuals. We see that play out through institutional culture. Do you have advice for how can patients and caregivers navigate those differences of approach or differences of opinion? Because speaking from experience, it can sometimes be a little confusing and confounding and even sometimes paralyzing of, should I switch institutions? Should I stay where I am? So do you have any advice just to help patients and caregivers navigate this landscape?
Why don't we go down the row, and I can start here. So, I think what you're getting, hopefully from this symposium, is the importance of an organization like the Cholangiocarcinoma Foundation, right? Because they can sort of serve as kind of your hub, right? You have advocates, caregivers, folks who have been through this, again, we are all different places, but we have, I have no pride, no hubris. I mean, if I know these colleagues, it's a small community amongst us, especially as you kind of move up the chain. And so I am more than willing, and I tell patients, always seek a second, third opinion. You do that for your car or for your house. Why wouldn't you do it for your life? And I just tell folks, I say, look, through the foundation, I know people all over the country, where do you want to go? And I sometimes make it kind of helpful. I know there's a trial, let's say, with Jim in New York, or there's a trial with Malin down in Houston. If I think you're a candidate, I'll put you in touch. I'll even send in the warm handoff email so that you can kind of bypass the usual sort of telephone tree. And so I think, because to me, it's what works for you and your family. Now, for some folks, my rural farmers in Oregon, they're not going to be able to travel to New York City. It's just not possible. So we do what we can. I think Melinda's story is also very inspirational. I mean, she did a lot of that legwork on her own, right, before the foundation was really in place. So I would say is that the take-home points really is that there should never be pride. We're all humble. I hope we're all humble. But this is a multidisciplinary process. There's a lot of things out there. The question is, how do you make those things work for, as Kareem mentioned, the individual situation? Because your tumor is not the same as the tumor of the person next to you. Your tumor's got your thumbprint on it. It's got your DNA on it.
Yeah, I have to echo everything that was just said. I think one of the advantages of being in New York is that there really are high-volume centers, multiple high-volume centers. And I always encourage any person that sees me to seek a second opinion, seek a third opinion. And then ultimately, what I've come to realize is that folks are going to just go with who they are most comfortable with, and who they've developed whatever personal relationship or whatever it is that they say to themselves, I feel comfortable with this person. And oftentimes just trusting your gut. I mean, the institutions that do this often enough have mechanisms in place to try to ensure the best possible outcome. I know we do that at NYU. I know they do it at MSK, Mount Sinai, Cornell, Columbia, and all around the country. One of the reasons why I posted that study of 24 high-volume centers and kind of the complications in the quote-unquote expert centers is because this problem is a tough one to tackle surgically. It's super important to go to expert centers, but even in the best of hands, it's a tough problem to deal with surgically. So at some point, when you do choose that provider, it has to be someone that you're comfortable taking this battle on with, and someone that you think is going to advocate for you when times get tough.
Yeah, I don't have really much to add. I think in New York, the patients with cholangio are actually lucky because they're spoiled for choice for people who will advocate for disease, and oncologists and surgeons and multidisciplinary teams that have expertise. So, I was at Cornell for 7 years before coming here, and we were across the road from Memorial until today. And what will happen is the patients will come and see me after they've seen Myron Schwartz at Sinai, and Jean Imond at Columbia, and Bill Jarnigan or Mike D'Angelica at Memorial, and then they'll come and see us for fourth opinion or fifth opinion. And I think there's a point at which, if you get 10 opinions, probably not great. A lot of people have different opinions, but there are enough experts in New York. And honestly, if someone comes and tells me, I saw Dr. Jarnigan at Memorial, he wants to do my surgery, I'm like, just stay with him. He's one of the pioneers in this. You don't need me. Why are you here? If you'd like me to operate on you, I can. But if this is his plan, this is great. Very, very, very rarely do we disagree on opinion here. And oftentimes what will happen is they'll send us a patient and say, look, we've done what we can. We're not going to be able to do anything more. Can you transplant them? And that's where sometimes we get patients where other patients, but also, different to Flavio's area where he's the only transplant center, there's 7 transplant centers all down the river. So, not everybody has cholangio protocols, but 5 of the 7 do. So, nobody's going to hurt you. Everybody's going to give you the best care possible. So I don't think New York, you're lucky in New York. Outside New York, it's very, very different.
Okay. Thank you. All right, great. Thanks. We can always use more time. Got another case for you guys.
All right, we're going to bring it back to the patient right before lunch. I'm very pleased and happy to introduce Tanya Jennison. She was diagnosed with intrahepatic cholangiocarcinoma in 2019 and brings both resilience and perspective to her journey. She's a registered nurse, a mom of 4, and an active advocate who continues to live life fully. Whether that's working full-time, spending time outdoors in central New York, or teaching Muay Thai kickboxing. Good job. Tanya shares her story to remind patients that strength and possibility can take many forms. Please join me in welcoming Tanya.
I'll try to make it brief. I'm not a great speaker by any means, and I know everybody's getting hungry. As she said, my name is Tanya Jennison. I'm a registered nurse, and back in 2019, that's when my life changed, and then forevermore remains changed. So if you look in my chart, it says 46-year-old female, Bassett labor and delivery RN, who presented with acute onset right upper quadrant abdominal pain with nausea and vomiting while at work December 7th, 2019. So December of 2019 was not a great Christmas year for us and our family. But I got some great things out of it, like some of my friends, and learning how to advocate better for myself as well as my patients. And I've also worked in oncology, and I work in a pre-op area as well. So becoming an advocate for patients and myself has been wonderful, and meeting all of my newfound friends. But when my life changed, reading that in my chart, it reduced me to the medical facts. And sometimes it's easier as a patient to become the medical facts, and sometimes it's more challenging. And the mental piece of being someone with cancer, I think is the most challenging part of it, because I've had a fairly easy way of getting through my cancer diagnosis. My final diagnosis was stage 1A, which is rarely heard of. I had an intrahepatic cholangiocarcinoma. It was about 4.5 centimeters. And I feel like that was easy compared to someone who's been fighting for a couple of years and had multiple surgeries and lots of systemic therapy and trying all sorts of avenues, although I did do some various things. So I'll just kind of quickly go through some things that I had done. I thought originally I just had a gallbladder attack. I was going to ignore it. But I had a really great colleague that I worked with, and she kept harping on me, you fit the criteria for having a gallbladder issue. And when we were in nursing school, we learned the famous Fs. Like, you're in your 40s, you're still fertile, you're a female, you're fat, because I was overweight at the time. You fit the criteria, get an ultrasound. It's going to be in your best interest. So if she hadn't harped on me, I would not have gotten an ultrasound. Nurses are the worst patients. So because of her, I called my primary care doctor a couple days later, got an ultrasound about a week later, found this mass in my liver. And then I proceeded to go on having a biopsy in December. Eventually, I went out to University of Rochester Medical Center, saw a surgical oncologist. He didn't really have a lot of great experience. He had maybe 50 surgeries over his entire career that he had participated in. And then that was on a Friday, and on the Monday I already had an appointment at Sloan Kettering to see a surgical oncologist, and that doctor really hit me hard with impressing me with not only his experience, but I felt like he was leading me down a great path. And as someone who's a nurse, I look into research. I'm really difficult to convince of certain things unless you're going to show me evidence-based practice, some published studies. My daughter had an injury to a couple of her nerves at birth, and it took me 6 months to agree to have a nerve surgery for her so that at 22 years old she has a functional arm. Thank God I agreed, but it took me 6 months doing my own research. So I need someone who's confident in giving their care and leading me down the right path and is going to show me the evidence, and that's how I felt about Dr. D'Angelica at Sloan Kettering. So that was December 2019 into January 2020. And then we all know COVID hit in March. And that's when my surgery was supposed to occur. Leading up to March, in February, I had a portal vein embolization because without it, I wouldn't have enough liver remnant left over to be able to say it wasn't too risky. So I had portal vein embolization in February. March of 20, I ended up with surgery, and I didn't know until the night before surgery if I would actually still have it because they were only doing really important necessary surgeries. And to me, cancer surgery is important, but I don't make those decisions. So I didn't know until about 6 p.m. the night before if we were still going to be able to have surgery. So we drove down in the middle of the night so I could be there at 5 a.m., dropped off on the street corner because nobody could come in with me. And then I navigated the surgery and the recovery piece for a couple days on my own. And during that time, I had a great surgery. I lost an entire day of memory. I really thought that I had maybe used my Dilaudid pump and pushed the pump one time. That's not what my chart shows when I read back on it now. I lost an entire 24 hours, which is not uncommon for people to be really hazy after they've had surgery. But I also was hallucinating on the Dilaudid, and nobody knew that because I was alone. I couldn't have any family or friend near me, so I kind of figured it out when I couldn't understand a text that someone was sending me saying that it was, I thought they were saying that it was snowing, and they were trying to tell me that I was snowed, which is a term that we use when someone is kind of like high and not really with it. And I kept saying to them, it's not snowing out my window. And finally, they were responding back with, something is wrong here, Tanya. And then I talked to the nurse, and she ended up talking to family, and I no longer had the Dilaudid PCA, and things went better. I was up moving around, wearing a mask out in the hallway, trying to get out of there as soon as possible, and then there was fear invoked because there was talk on television about closing bridges, and hotels were closed, and what if they discharged me and I couldn't get out of the city? Was this a rational fear? So I put my call bell on, called my nurse in and said, I've called for my ride. He's 4.5 hours away, so please get my paperwork ready. She called the fellow. Fellow came in. You can't go home. Your bilirubin's too high. I said, you got 4 hours to make it better. I'm leaving. I know what an AMA form is. I'll sign it. It's not a problem for me. of course, Dr. D'Angelica and the fellow were talking, and they ended up discharging me. I didn't have to do the AMA form, but I was going anyways. Because I left early, I ended up going home with a seroma. I had a wound dehiscence, which was very small, but again, I'm home with just my significant other at the time, who we had just started dating the week before I was diagnosed. And I had told him, you know, it's been great that we went on a date. You're a wonderful guy, but you do not have to stick around by all means. I probably would not have stuck around if I had dated a man for a week and they found out that they had this terminal cancer in our eyes. And we just got engaged this past December. It's been 6 years now. He's a really great guy. I can't let anybody have him. He's mine forever. But I'm just thankful for him, along with many things throughout this journey. When I had surgery, came home, I had a lot of elevated blood work. And interestingly enough, when I had that original ultrasound and I did some blood work, all my liver function tests were normal. My CA-19-9 was normal. My CEA was normal. Nothing was out of the ordinary, but I had this intense right upper abdominal pain that kind of went away after maybe a week or two and had calmed down to a dull ache in one spot. I could take my 2 fingers and put it in the one spot, and to this day I can put it in that same spot. That was my only indication that anything was going on. And if I hadn't listened to my friend who harped on me to go get an ultrasound, if I didn't have that medical knowledge where I was around this colleague who could say, go get your ultrasound, I may have ignored it and come back with a higher number than a 1A with moderate differentiation and perineural involvement. So my diagnosis was December 2019. Now we're about 6 years later. So after my surgery, got over all the recovery pieces of it. We're still in COVID. I never went back to Sloan Kettering for a single appointment. I was able to do it virtually. So if we can say anything is positive about COVID, it taught us to use technology so that we can get access to better care, we can get more access to care, and we can talk amongst each other and be able to get better care. That is a definite positive, I feel, coming out of COVID. A negative that I would say I would come out of this diagnosis was the mental health issue. So about a year ago, I was still trying to deal with the complications of my mental health issues. Just from the diagnosis. Having things like survivor's guilt. I was just talking with my friend. We're having a little girls' weekend and going to the fundraiser tomorrow. So a couple of friends are coming up. And I was talking with one of my friend, Christina, last night about some of this. And she mentioned survivor's guilt while we were here. And it's a real thing. Why am I here? But all these other friends and acquaintances that I've met along the way, why aren't they here? Why am I having an easier time, but my friends and acquaintances are suffering 100-fold? The why, I don't know, but how do I deal with it? So I started to do some mental health therapy and get a therapist, which was the greatest thing in the world. I think most people could benefit, whether you have a cancer diagnosis or you're just trying to live your life. It's always great to have someone who is even in their thought process with you, can listen to you, validate that you've been heard, but not give you the answer, help you to find the answer on your own. And that's what my great therapist has been doing, is trying to get me to understand my own internal conflict and then come up with the right decisions on my own. So we also have this, we should feel better now. It's an internal conflict that invalidates all of the procedures, the surgeries, the scanxiety that we get before every MRI, CT, PET scan. And it can be the day before, or it can be weeks ahead of time that we start having this scanxiety. Scanxiety is what we refer to it in the cholangio community. And I'm sure other cancer communities think of it in the same manner. And I like to use the word we instead of I suffer from scanxiety or I suffer from survivor's guilt, because in the community, we all have some form of this that's going on. There's a PTSD that some of us will end up having just from the cancer diagnosis and trying to travel our way through all of this chaos. I don't like to use the word journey because I love to travel and it's fun for me, and traveling is a journey. Cancer is not a journey, and it's one of my like pet peeves. And I don't mind that other people use it. Just for me personally, cancer has a negative connotation to it. And I like to journey and travel and explore, and that is all positive. So I just say it's like a path that I'm taking. Short walk, I like to say, versus a journey. So my life now, it's in 2 phases. And it's very concrete. Before cancer and after cancer. And it will never change back to before cancer, because I am now not the same person that I was before my diagnosis, before my treatment, before my side effects from doing capecitabine for 6 months. I have long-term side effects from doing the capecitabine, the mental health, looking at the support systems that I've developed, seeing how they change, finding out who your real friends are and aren't. Because when you get a cancer diagnosis, people disappear. They're afraid for whatever reason, and that's okay. That's their choice. But you also find people. You find your tribe, which is like sitting right there, my ladies. You become a different person. So I can't go back to the before cancer, and I'm in the after cancer. But it's not all I am. I hike, I do photography, I travel. I'm learning how to fly a drone for an upcoming trip that I'm taking in conjunction with the conference that's coming up in May. I do the Muay Thai kickboxing. I've been doing it for 15 years. I took a little time off to recover from surgery, and I also teach it. And when people come in and see a woman who's almost 53 years old, and they ask, hey, is the instructor here today? And they're usually in their 20s, and they're usually fit and ready to, be aggressive. I say, yep, that's me. And then they realize that I could do a lot of bad things to them if they don't know what they're doing. That's empowering. I like feeling empowered, not only as a woman, but as a middle-aged woman, but as an individual that was misjudged by my appearance, because I'm in my 50s and a woman. I'm grateful, I'm thankful, and I'm blessed for so many things. Those are 3 words I use often. As I said before, I'm thankful that I've got to meet some really great people. So that's a positive thing that came out of my diagnosis. I'm grateful for people like Dr. D'Angelica, for medications like capecitabine that can mop up all those little remnant cancer cells, for breakthrough treatments that are coming down the line. I can remember when pembrolizumab was not even an option, and then we talked about it at one of the conferences, and now it can be used often, and it's not fought over with insurance companies. And it's interesting that the first time in this symposium was a few moments ago when insurance came up. We all know whatever the doctor says doesn't go. It's whatever the insurance company says, that's what goes. And it's challenging. It's really challenging because you're fighting for your life physically, but you're fighting for your life on your phone, talking to people who are just checking a box or not. And your doctor's fighting for your life, and he's doing the same thing. And I'd rather he be able to spend time or she be able to spend time doing more surgeries rather than writing those appeals and talking on the phone on those appeals. So I want you to take away today that I'm thankful, grateful, and blessed, but I want mental health to be addressed more as the providers who are taking care of cancer patients. Sometimes the providers will ask, how is your mental health? How are you doing? How's your mood? Do you feel like you need anything? Most cancer patients will say, I'm fine, because they're fighting the physical stuff. We can only fight so many things at one time. Like you can only carry so many things in your arm at one time. So can we fight the mental health stuff and the physical health stuff simultaneously? Many times not. But when you as a provider ask me, how's your mental health? If I say it's fine, ask again. Maybe at the next appointment, ask in a different way. Because we need support, but we won't necessarily ask for the support because we just can't do it all. So more mental health benefits, more mental health assessments, more mental health support, encouraging people to be kinder to themselves, forgiving to themselves. I encourage you all to do that. I hope you enjoy lunch.
Great, wonderful. Thank you so much. That was so great to hear. And a tremendous thank you really to this morning's speakers. Let's give them all a round of applause. They were wonderful. It's been a truly transformative morning session. So we will break for lunch now, returning at 1 o'clock. And so I'll see everyone in the afternoon. Thanks. All right, welcome back, everyone. For those of you that are persisting with us and braving the grocery store after the sessions today. I actually didn't realize that I put the nutrition session or the session with our nutritionist right after lunch, but maybe that's better than doing it beforehand where your stomachs would be growling. So I hope lunch gave you an opportunity to recharge. And we have an excellent set of discussions and speakers lined up for this afternoon, so let's jump right in. Rebecca Guterman is a registered dietitian and certified oncology nutrition specialist here at NYU. She works closely with patients facing gastrointestinal cancers, providing practical evidence-based nutrition guidance before, during, and after treatment. A firm believer in food as medicine, Rebecca helps manage patient side effects and make nourishing choices that fit their real lives. So please join me in welcoming her today. Thank you.
Thank you for having me and happy to be here. And today I will be talking about how I optimize nutritional status for cholangiocarcinoma patients. So today I'll discuss the role of the oncology dietitian, a registered dietitian in an outpatient oncology setting, understand the goals of nutrition during cholangiocarcinoma treatment, and learn strategies to help everyone maximize their intake based on their treatment and their symptoms. So what does an oncology dietitian do? We really strive to help everyone to provide personalized nutrition recommendations for everyone that we're working with. We know that early intervention is important, so really trying to get involved in patients' care early on. It's recommended to have screening tools in place in cancer centers to really be able to identify patients early on in their treatment to help prevent malnutrition, help keep their nutritional status strong. We'll do a comprehensive nutrition assessment once we are speaking with people, going over their food and nutrition history, including any recent changes in their nutritional status or their ability to eat, look at any changes in their weight that they may be having, and dive into some of the symptoms that they might be presenting with, and then speak of symptoms that they might expect as they're going through their treatment. Our overall goal is to help people feel well and really get through whatever their course of treatment might be as best and easily as possible. So we understand the side effects of patients' treatments and how they might impact their nutritional status. If somebody's going through chemotherapy, radiation, or surgery, we'll think about those treatments and personalize recommendations based on those. We strive really hard to be better than Google or AI and communicate credible information. We practice in evidence-based medicine. We practice to really be that resource for people going through treatment, because we all know there's a lot of information out there, especially with nutrition, and a lot of wrong information. What works for one person might not work for another person. So we want to be the go-to resource for people going through this because we have the knowledge and background to do so. We'll also monitor any needs for nutrition support, whether it's tube feeds or recommending an appropriate nutrition supplement or any micronutrient or vitamin repletion. We would ask for lab values possibly to really make it personalized to the person and what they're going through in their treatment. Our goals, again, we want to really practice with early interventions. We want to address and prevent malnutrition or prevent it from getting any worse. We know that malnutrition is so present in the cancer population, unfortunately, and we know that people with malnutrition have increased treatment interruptions or toxicities with the treatments. We know it could increase chances of a hospital admission, increase length of stay, and unfortunately, increase mortality. There are a couple of studies that looked at patients that were undergoing curative intent resections for extrahepatic cholangiocarcinoma, and those that were severely malnourished going into surgery did have a worse overall survival. So we really want to be there to help prevent malnutrition and work with people to maximize how much they could eat and how well they could eat. So minimizing that weight loss is a top priority. Managing any of those treatment-related side effects, again, considering what the person's going through. If they're having stents, if they're having any drains or surgery or chemotherapy planned, we will take all of this into consideration and provide ongoing support. We are part of the multidisciplinary care team and involved in various times with patients going through their treatments. So if we are meeting with somebody at the beginning of their treatment, we will be there for the duration of their treatment and even into survivorship. So common side effects that we address are malabsorption or having fatty stools, being nauseous, feeling full quickly, just really a poor appetite or a low intake overall, feeling very tired, or having any ascites or abdominal fluid accumulation. Nutrition recommendations will vary. Again, we'll strive to provide personalized recommendations, but protein is something that we focus a lot on. There are various equations to estimate protein needs. Usually in somebody going through cancer treatment, they're going to range anywhere from 1 to 2.5 grams per kilogram of body weight per day. So what does that really look like? For an 150-pound person, using middle of that range, 1.2 to 1.5, which is common, we could be recommending anywhere between 81 and 102 grams of protein per day. So it's quite a high bar, but we know the importance of protein. We will recommend this along with adequate calories to help avoid unintentional weight loss, help people feel strong and have energy, because we know that being weak, being fatigued does impact quality of life. And of course, we're trying to support people through all of these treatments. An early emphasis, again, on good nutrition is important, and we do work as part of the multidisciplinary team, so always appreciate physician reinforcement of some of our recommendations. I want to emphasize weight stability versus an emphasis on gain, especially if somebody is coming into treatment having lost weight already. It is successful if they don't lose any more weight. If their weight remains stable through the duration of their treatment, that should be celebrated, because that does mean that they're eating adequately. It's very hard to regain weight during these treatments, as I'm sure a lot of you can attest to. So being weight stable or preventing those losses is really a successful intervention. Again, we know that proper nutrition helps people improve tolerance to their treatments. Poor intake could lead to making those side effects of the treatments more intense, which could lead to delaying the next cycle of treatments if it's chemotherapy, or reducing the doses, and we want to help people work to stay on their prescribed regimens. So back to protein, we want to emphasize lean protein and include protein at each meal and each snack. This helps maintain lean muscle mass, helps maintain strength and endurance going through these treatments. So what does this look like in actual foods? It could be fish, eggs, chicken, nuts or nut butters, or often I find myself recommending foods that are softer in texture. They just tend to be a little bit easier to eat, so people are a little more likely to eat more of those foods. So things like quiche or chicken, tuna or egg salad, meatloaf or meatballs, those softer textured protein foods often go over well, and if people could incorporate that as parts of their meals, they might meet their nutritional needs a little more readily. We love our plant-based foods, fruits, vegetables, whole grains, beans, lentils, nuts and seeds, but also recognize that these might not be the most appealing when somebody's appetite is poor, if somebody's feeling full quickly or having diarrhea. So we want to make adjustments and recommendations if that is the case for somebody. This might look like throwing some spinach into scrambled eggs to still get that spinach in the meal, adding some chopped peppers or shredded carrots to tomato sauce, blending fruit into a smoothie. You still get all of the benefits of those foods, you're just eating them in an easier to consume format. So these are some of the strategies that we might talk through with people. Nutrient-dense foods are important, especially with a poor appetite, any weight loss, or again, preventing additional weight loss. They're high calorie, low volume, so foods that are providing fat are going to be a focus as well. Fats have 9 calories per gram versus protein and carbs, which have 4. So you're getting double the amount of calories in fats versus protein and carbs. So trying to incorporate these as part of the meal or on top of the meal is something that we work through with people. So for example, putting avocado on eggs, adding some extra olive oil drizzled over a cooked meal. We're not talking about increasing the volume of the meal there, but we are helping maximize the calories being consumed. Adequate hydration is important and might need to be something that happens between meals. If somebody's feeling full quickly, we encourage a variety of fluids, especially if somebody has some taste changes and not so inclined to drink as much water. There are so many sports drinks and electrolyte powders out there, which could be good for rehydrating or making your own oral rehydration solution. The World Health Organization recommends a liter of water, 6 teaspoons of sugar, and half a teaspoon of salt. So something quick and easy to mix at home if those other drinks are not already in the house, especially if somebody's losing fluids rapidly, a lot of diarrhea over a short period of time. We want to make sure they are replenishing those fluids and electrolytes. I wanted to highlight oral nutrition supplements. There are so many out there, and I often find that people say, I was told to drink something, and they don't know what that something should be. So this is a variety of higher calorie, higher protein options that range are all above 200 calories, mostly above 15 grams of protein. So if somebody's really struggling to eat and using these as meal replacements or snack replacements, we want them to focus on these higher calorie protein drinks versus some of the more commercially available ones might be lower in calories and really high in protein. Like 150 calories and 30 grams of protein might not be the best balance for somebody actively going through treatment or actively needing these as part of their eating patterns. If somebody's so inclined, we do love making things at home as well, and wanting to highlight that with a few ingredients at home, you could also get a high calorie, high protein shake. So taking a cup of Greek yogurt, using 2 tablespoons of peanut butter, a banana, a cup of strawberries, and water as needed, blending that together, you would end up with a shake that's 560 calories and 32 grams of protein. So you don't need to buy anything pre-made. You could make it at home or do a combination of both. Whatever works for the person is something we would certainly encourage and support. Foods to limit. A couple of reasons that we would say to limit things. Fried and greasy foods might be harder for somebody with cholangiocarcinoma to digest. So preventing any discomfort or symptoms after eating is certainly a priority. And also the American Cancer Society, as well as the American Institute for Cancer Research, recommends limiting and avoiding processed meats and red meat. So we do talk about this with people that we are counseling, but wanting to acknowledge that if somebody's not eating so well and this appeals to them and they tolerate it, we will certainly support it. Some other things we don't love are concentrated sugary things. They could make people feel full without really getting in nutritious calories or proper protein. They could also spike the blood sugar. So we want to be careful with some of these things. Again, doesn't mean that somebody could never drink any of these. If a Diet Cola is going to help somebody enjoy their meal, we'll support it, but usually not prioritizing these as part of everyday consumption. So for managing some of the more common side effects with treatment, if somebody's nauseous, we recommend not going more than 3 hours without eating. Keeping something in the stomach helps to keep the stomach acid more settled, or keep the stomach more settled by preventing the stomach acid buildup. Dry foods are really good at this, so things like crackers, dry cereal, toast, a bowl of white rice with some chicken in it could be a meal option for somebody that's nauseous. Poor appetite, we love to recommend small and frequent meals, but what does that actually look like? It could look like a couple of eggs and a piece of toast, then a few hours later, a bowl of yogurt, a couple hours later, maybe a sandwich. If that whole sandwich is too much for somebody to finish, we'll say maybe eat it open face, try to get all of the protein consumed there. A smoothie could be the afternoon snack with a hearty bowl of soup for dinner and then some pudding in the evening. This could be a pattern that could help somebody maintain their nutritional status by getting adequate calories and protein throughout the day. We also encourage movement. Moving a little bit throughout the day helps stimulate the appetite. So if somebody's not so hungry, not really looking to eat, some movement might help a little bit. If somebody's having diarrhea, we recommend lower fiber or easier to digest foods, replacing those fluid losses like I was discussing before, and monitoring for any exocrine pancreatic insufficiency or bile acid malabsorption. So in practice, a meal of some salmon, string beans, pasta, that might be something that's tolerable if somebody's having diarrhea. If somebody's fatigued, we like to lean on our caregivers. If we have people in our lives that are helping out, having them do some grocery shopping for us, having ready to eat or easy to prepare foods in the house are really helpful on those days post-treatment or recovering from surgery. A jar of peanut butter and a loaf of bread could help make fill some gaps between meals. Also a place that we encourage increased movement. Being tired, we're not expecting anyone to do any vigorous exercise or activity, but getting up and moving around throughout the day is often helpful. If somebody's feeling full very quickly, again, we're back to recommending small and frequent meals. This we might focus especially on easy to digest foods and nutrient-dense liquids, things that are going to pass through the system a little more readily, not leave somebody feeling full for hours after they eat it. And then with exocrine pancreatic insufficiency, people might need pancreatic enzyme replacement therapy. This would be something that would need to be taken in supplemental form of enzymes, taken with all meals and snacks that contain fat, and then adjusting based on the fat content is something we discuss with patients often. A little bit more on enzymes, usually needed post-Whipple or if there's any pancreas involvement in the disease. Some of the signs and symptoms that we ask about and look for that indicate insufficiency would be light-colored, floating, or oily stools, urgency, or like they need to get up from the table or go right after you eat. That's something that we ask people about to evaluate if they're having any difficulty digesting food, which would indicate the need for enzymes. If people are having a lot of foul-smelling gas, like really large amounts, that's another symptom, as well as the inability to gain weight. If somebody is prescribed enzymes, we reinforce the use, taking the capsules at the beginning of eating with the first bite of food. We often find people are not taking them with protein shakes and just highlighting that because of the calorie, protein, and sometimes fat content of those, the enzymes will help with the breakdown and absorption of those. And then if meals are lasting a long time, if somebody is having a multiple course meal or they're just a very slow eater, they might need to space out the capsules or take additional capsules through the duration of the meal. We always recommend starting at the lowest dose, which is 500 units of lipase per kilogram per meal, and increase based on symptoms, using the medication Creon as an example, which comes in 36,000 unit capsules for somebody that is between 100 pounds, 150 pounds. That might be one capsule per meal, one per snack. And then if somebody's over 160 pounds, they might need 2 capsules per meal, 1 per snack. But what's it considered a meal versus a snack is a very common question that I get. And I tell people to really think about the fat content rather than the time of day or what the typical thought of that food is. So a snack of peanut butter on toast might require a meal dose of enzymes because the fat content in peanut butter is going to be harder to digest, versus a meal of cereal and skim milk might be okay with a snack dose because it is a lower fat meal. So really adjusting based on the person's eating habits and preferences and tolerances. And then some vitamins to remember. With malabsorption and difficulty eating fat-soluble vitamins might be something to consider supplementing. Again, we would ask for tests to assess if the need is there before recommending supplementation most of the time. B vitamins, including B12 or thiamine and folate, or other vitamins that might be impacted by the disease itself, the treatments that somebody's going through. So we do like to test to avoid any unnecessary supplementation, and then often educate people on avoiding mega doses or really concentrated amounts of these vitamins because they might not be necessary or beneficial. So in summary, refer to a registered dietitian early if the person's having difficulty eating for nutrition optimization. We will help them adjust their diet according to their symptoms, focusing on adequate calories and protein to prevent unintentional weight loss and really help them get through their treatment and recover as easily as possible, and consider pancreatic enzymes or vitamins if needed. These are my references, and happy to take any questions.
How do you feel about the old chestnut that sugar encourages tumor growth?
Great question. Sugar feeds cancer, our favorite. It's a very complicated topic. There is not a direct correlation with sugar and tumor growth. We can't starve tumors. We can't starve cancer growth by eliminating sugar from the diet. It's also really hard to eliminate all carbohydrates from the diet. Often we are working with people through active treatment as well, so highlighting that it's not an appropriate time to be thinking about limits like that. Again, we're not going to encourage drinking a Frappuccino every morning to start your day and getting all of the sugar that's included in that, but we're not going to recommend completely eliminating carbs.
Hi, it's not exactly a question, but I just want to show you that I do have Creon. I had the Whipple, and that presentation was spot on. It was like really good. Hit the nail on the head, so thank you.
Thank you. Good for you carrying it with you. It is the reality of post-Whipple digestion and eating. You need to take it with you when you leave the house.
What was that pill pack, though? That looks good. It only has 3, is it 3?
This is my Friday pack, so it's got morning, afternoon, evening.
Oh, okay.
It's all Creon.
Oh, great.
Good idea.
I have that same pill pack. It's great. I saw it on one of your slides. I wanted to ask you to talk a little bit more about cachexia. I think it's not talked about enough in this world.
Yeah. So I had it in relation to protein needs. Cachexia is the loss of muscle mass with or without the loss of fat mass. And we do see cancer cachexia often, especially going through treatment, on presentation, after surgery. It really could be something we're seeing at any point in time. Cancer cachexia has the higher end of the protein needs, all the way up to 2.5 grams per kilogram of body weight per day. So really trying to help people understand how much protein that is, trying to help them build 6 small meals throughout the day, because that will help maximize the protein absorption by not front-loading it or back-loading it with like a protein-heavy dinner. We want to space it out throughout the day. And pairing that with exercise. I didn't really highlight exercise in the talk besides moving around throughout the day for a poor appetite or if you're tired, but protein plus exercise is really what's going to rebuild and repair the muscles. So working with people, referring to physical therapy, telling them to get up and move around as much as possible, use light weights or resistance bands. I always say if the physical therapist in the hospital showed you exercises, do that after discharge. If they're only coming 2 or 3 days a week, there are other days to still do those exercises. So pairing adequate protein, adequate calories, and physical activity or exercise is really going to help prevent the cachexia from worsening or reversing cachexia, which is an uphill battle but can be done.
Hi, Rebecca, thank you. This is great. Just a question about sugar.
Yes.
If you are going to use sugar?
Yes.
If you're making some sweets, because my
Yes.
sister wants sweets, what's the best sugar to use?
A very complicated answer that I could give my long response to. So 2 buckets, real sugars and artificial sweeteners. I usually say let's try to minimize all of them. Not eliminate, but minimize. And also thinking about it in the picture of what else are you eating in the day. If you're eating a pastry for breakfast, if you're having fruit-flavored yogurts throughout the day, if you're drinking juices and sodas, if you have a lot of sugar in your diet, let's try to see where we could make some adjustments. I can't really confidently say there's a best sugar. All of the natural sugars, white sugar, brown sugar, molasses, honey, are all going to break down to glucose in the body. So at the lowest level, they're all really the same. The artificial sweetener buckets, there are some more natural ones, and they don't impact the blood sugar as readily. So for people with diabetes or that need to keep the blood sugar more controlled, that's the better choice, but really trying to minimize all of them. Again, not eliminating it, but where could we cut back on it? Where could we really cut out those super high sugary things? Foods with a lot of added sugar is usually the place I'll start with that. Did that answer your question?
Yeah, thank you.
Yeah.
I actually have a question
Yeah.
for you. My question is related to supplements, particularly the protein shakes, whether they be pre-mixed powders. I hear a lot from patients that they hate them. They hate the flavor. They're too sweet, they're too thick, etc. We talk about different things like cutting them with water, doing the fruit-based ones, but what do you tell patients and how can you help them with that part?
Yeah, so first, I'll usually not recommend more than one in the day. Sometimes I have patients come to me and say, my doctor told me to drink 3, and with anything, 3 a day every day is gonna end up being not palatable very quickly. If somebody is able to make homemade smoothies, that's usually what I would tell them to do if they don't like the pre-made ones. You could really customize these. You could use flavors that you like. You could use fruits that have a stronger taste. So like bananas are gonna be a strong flavor. Mango and pineapple could blend well into a smoothie and kind of mask some of the tastes of other things. Greek yogurt is something that not everyone loves because it's so sour for some people. So I'll say still use it as the protein source in the smoothie, but let's mask that with adding a banana or pineapple to make it taste sweeter. Thinning it with water helps, but we do want to balance the volume and nutritional concentration of things, because if it's too thin and it takes too long to drink, they might not finish it and get all of the nutritional benefits. If making homemade smoothies is not accessible for somebody, because I know it's not for everyone, with these, sometimes if the person's willing, I'll say use like a vanilla flavored one in their coffee instead of milk. You're not gonna put the whole 8 ounces in there, but it's still gonna be better than nothing. Also mixing it into their oatmeal is something that I talk to people about, like make the oatmeal a little bit thicker after it's cooked, mix a vanilla or a chocolate one in there. So little ways to sneak it in if they're not really drinking the whole thing at one time. Yes, on this list, what I hear is the insurers are very sweet. The Kate Farms are not so much. They're plant protein, they use pea protein, so they taste different. They're less sweet. So if that's something that somebody is not drinking them because of the sweetness, the Kate Farms might do better there. But some people don't like that pea protein taste. So it's really, we give a lot of these out as we have them as samples, and I always end saying the best one is gonna be the one that you'll drink. So trying a couple might be the way to find the one that somebody will actually consume. Yeah.
Plant protein, like what proteins do you suggest? Because how do you balance like nutrition but like obviously core powers aren't the best for you, like aside from the cancer, like they don't recommend, like there's a lot of additives, just not natural things in it. So like, how do you balance that? And also on top of that, like what proteins do you think are the cleanest maybe, or like the best?
Yeah, so the ingredients are usually a whey protein or pea protein. Pea is certainly plant-based, it's vegan, it doesn't have dairy. There's no great research saying we need to avoid dairy in chemotherapy or in cancer. So I usually do start with whey protein for people. Again, I find it more palatable. Reports back from people often find it more palatable. The ones that are pure whey protein will need to be mixed with something else to give it a taste or a flavor that leaves it to be palatable for the person. So it is a little bit of personal preference. There are powders out there that are just the individual protein ingredients. So yeah, we do take into consideration the list of ingredients in these things, but if it's a question of somebody really struggling to eat and eating nothing or having something with some added sugar or some artificial ingredients, we're going to be okay with that to get them through treatment. And then let's focus on whole foods and whole protein sources after treatment versus reliance on the powders. So it's a bit dependent on the current scenario.
Anyone else?
Could you speak a little bit about whole milk, almond silk milk, or oatmeal milk, which is preferable?
Yeah, so again, it will depend on what's going on for the person. Dairy, so whole milk, skim milk, any actual milk itself, plus soy milk contain protein. Almond milk, oat milk, cashew milk, very low in protein. So usually I'm working with people to find calories and protein every which possible place we could get them. So I do tend to recommend dairy milk or soy milk over the other milk alternatives. Otherwise, it's really personal preference and taste. It's also thinking about the ingredients that might be in some of the dairy alternatives. If they're sweetened, there are added sugars in there. Some of them have other ingredients we might not want to be consuming every day. Again, there's no solid research that dairy is a problem in cancer, so I don't shy away from it. If somebody doesn't tolerate it, my next recommendation would be a lactose-free dairy product. Yeah. Yeah.
Or supplements are difficult. I mean, everyone has an opinion on which one is better to take. And if we're looking to add into our nutrition, is there anything you'd recommend?
Yeah, so the question was, aside from a multivitamin or the vitamins themselves, are supplements recommended? Fish oil has some good research behind it in terms of it is anti-inflammatory. There's some papers saying that it does help preserve muscle mass or prevent muscle wasting. So that is certainly a benefit we like to hear. That's one that I'm more confident in recommending. We also think about the interactions with chemotherapy and radiation itself. So again, depending on what's going on for the person, if they're actively going through chemo or radiation, I will speak for myself and tend to go more conservative and say, the chemotherapies that your oncologist recommended, we know work for your disease, your tumor type. The supplements at the same time have not been tested, so we don't know what's gonna happen. There are theories out there that antioxidants, so something as benign seeming as vitamin C, taken in high doses in an oral form, might protect cancer cells or make the chemotherapy less effective. So we take all of those things into consideration when we're talking about supplements to someone. Multivitamins usually don't have those mega doses of the individual vitamins. They're more balanced. So usually we feel pretty confident saying that those are safe to be taken, even with chemotherapy and radiation. But ultimately, we want things from our food. We always think of a food first approach. So if we could cook with turmeric, if we could cook with ginger, if we could eat salmon or sardines or mackerel and get those antioxidants and omega-3s from our diet itself, we know the body processes those so much more readily and absorbs them well, and you get other health benefits from eating the foods versus just looking for a pill.
Hi, I just have a question. Is eating rice every day a bad thing? And also sardine and tuna in a box, is that bad as well?
So eating rice every day, or I'll lump it in with other carbs, not a bad thing at all. I wouldn't recommend a meal that's rice by itself. I'd say let's eat rice with some fish or some chicken, some protein source, and round out that meal with some vegetables for fiber, for vitamins, for minerals that we get from that. So we can eat it every day. You could eat it more than once in the day, carbs, rice, bread, pasta. But usually I will advise not eating it by itself. And then canned fish is a great protein source. It's convenient, it's readily accessible and available. I would put it on that list of easy to prepare or ready to eat foods to have at home after surgery or when fatigued. So yeah, BPA-free cans are pretty common these days, so not so much of a worry there.
Anybody else? One more.
I'm curious to know a bit more about fats. I know that you mentioned the pancreatic enzymes. However, if someone doesn't have the cancer in their pancreas, should they be limiting fats or focusing on certain types of fats?
Yeah, so it will depend on the location of the disease. If the cancer is in the biliary system and blocking anything, or somebody has a drain, that's gonna make fat absorption harder because the bile isn't flowing properly. If somebody's post-Whipple, also the enzyme production is lower, so the body isn't producing enzymes to readily absorb fats. What was the other part of your question? I'm sorry. Which ones would I recommend?
What type of fats would you recommend
Yeah.
for someone with a limited bile movement, and what are good ways of getting high calories with low fat?
Yeah, so if the body's having problems digesting fat, we would need to go on we would need to limit it, so a lower fat diet overall. If somebody's able to process and tolerate and digest fats without a problem, I'll recommend most of the things above the green line, so like avocados, nuts, nut butters, olive oil, olives. Those all have unsaturated fats, so that's the healthy fat category. Dairy and whole fat dairy is more saturated fat. Those are the same fats in like red meat. So not as healthful, but still could be incorporated as part of a diet if somebody needs the calories from fat. If somebody's needing a low-fat diet because of absorption problems, but we still want to keep the calories high, we'll try to get little bits throughout the day because the body will more readily digest smaller amounts at a time. And then again, focus on those protein sources and probably balance out with some carbs to keep the calories up. Yes. Okay.
Can you recommend a book for us to follow up on?
I would recommend a consult with a registered dietitian. Because again, a book's written for the public. A book is not written for you. So most cancer centers do have dietitians on staff or have a dietitian that they could refer you to. So I do promote the profession and promote meeting with somebody to really get your history, focus on what your body needs. Yes, thank you, everyone.
And I do echo that. I know it sounds, we'd all love a book we can get in the bookstore, but I think particularly for bile duct cancers, there's so many nuances that it really is helpful to work with the folks that meet the patients and know their particular challenges. All right, so we are going to move on to the last portion of today's program, and this is all going to be focused on systemic therapy. I know the surgeons are like really tall and handsome, and they have something to say about everything, but you are all waiting for the medical oncology part, right? Yeah, okay. So with that, I would like to welcome Dr. Rafi Winograd. He is a medical oncologist here at NYU. He focuses on caring for patients with gastrointestinal cancers, including bile duct cancers. In addition to treating patients, he is actively involved in clinical research aimed at improving how cancers respond to therapies, including immunotherapy. And he is dedicated to bringing the latest advances from research directly to the people who need them most, and him most, both. So thank you so much. Let's welcome him to the stage to talk about how he personalizes treatment.
All right, good afternoon. Thank you, Dr. Spencer, for the very kind introduction and also for the invitation to speak here today. I'm a little nasally. I just getting over a cold I got from my daughter, so just bear with me. All right, let's see if the clicker works. What am I doing wrong? Okay, so here's an overview of what I'm going to talk about today. First, we're going to review some of the recurrent mutations that we see in cholangiocarcinoma. We're going to look at a summary 20,000-foot view of some drug approvals in cholangiocarcinoma treatment over the last couple of years. We're going to review how do we treat cholangiocarcinoma with standard of care treatment approaches for metastatic disease and some new potential directions. And lastly, we're going to talk a little bit about adjuvant therapy, which is systemic therapy that we give after surgery to reduce the risk of recurrence. Next slide, please. All right, keyboard works. Yeah. So this is a nice slide from a recent review paper looking, describing some of the recurrent mutations that we see in cholangiocarcinomas. And there really is a distinction in the mutational profile, the types of mutations that we see depending where in the bile duct tree the tumors arise. So for intrahepatic cholangiocarcinomas, that's that pink kind of box, and the top, does the mouse work? Yeah, so these up here. We see IDH1 mutations in somewhere between 10 to 20% of these patients. We see mutations in a gene called KRAS, for which there are no currently FDA-approved therapies, but for which there are some coming down the pipeline. And we see mutations in FGFR2 at a relatively high frequency, whereas in bile duct cancers that arise more distally, either in perihilar or distal bile duct, we can see a different set of mutations. Again, KRAS mutations can be seen at a higher frequency here. This is closer to what we see in pancreatic adenocarcinoma, which is closely related to distal cholangiocarcinoma. And we also see some mutations in genes like BRAF and HER2. So this is kind of a 20,000-foot view. As far as what are the medications that are used for treating cholangiocarcinoma from a systemic standpoint, right? So we heard about surgical approaches and histotripsy and ablation. These are medicines that are prescribed by medical oncologists like myself and Dr. Spencer that go in either intravenously or orally, and that are meant to go anywhere in the body that the cancer might be hiding, right? And so local therapies are great because they can treat cancers effectively where they are. Systemic therapies can go everywhere via the bloodstream that the cancer is, because the cancer ultimately is dependent on blood flow for nutrition and survival. So gemcitabine and cisplatin, which is the base of the chemotherapy regimen used in metastatic cholangiocarcinoma, was first approved back in 2010. You'll notice a very long gap here until any other approved therapies here. And some of these approvals over here, like larotrectinib, are actually what are called pan-tumor, meaning these are not specific to bile duct cancers. These are tumor agnostic approvals by the FDA for any tumor, whatever type it might be, that has a mutation in this particular gene called NTRK. And the two approvals were made in 2018 and 2019 for those types of tumors. And it's not until 2020, so 10 years later, that we have the first specific approval here for a drug in advanced cholangiocarcinoma, which is pemigatinib, an FGFR2 inhibitor, followed the next year by ivosidenib, which is an IDH1 inhibitor. And then the last couple of years, we've seen a lot more activity. First and foremost, we've seen the use of immune checkpoint inhibitors in combination with chemotherapy, which we're going to review on the next slide. And then we see the approvals of some other regimens, including futibatinib for, again, FGFR2 mutated cancers. BRAF V600E mutated cancers have a pan-tumor approval with dabrafenib and trametinib, and then trastuzumab deruxtecan and zanidatamab more recently. And I'm not testing you on all these, so just here to give an overview. So for patients that are newly diagnosed with metastatic or unresectable cholangiocarcinoma, so patients who are not able to get cured with surgical approaches, right, the standard of care treatment for what we call first-line treatment is using an immunotherapy agent, either an inhibitor of PD-1 or PD-L1, which is a receptor on T cells, to augment the efficacy of standard chemotherapy, which is gemcitabine and cisplatin, which has been around now for 15 years. And these studies, which were published in back-to-back years, the TOPAZ-1 study with durvalumab and the KEYNOTE-966 study with pembrolizumab, show that the addition of one of these immunotherapies does improve the overall survival of patients with these diseases. You can see the curves that do separate and stay separated over time. And we see also a hazard ratio, which is a number that we look at to try to estimate the benefit of around 0.8, which is similar in both studies. And that equates to about a 20% reduction in death for these patients. What's interesting about immunotherapy and why we as a medical oncologist are particularly excited about that as far as a modality of treating cancer is what we call the tail of the curve. So what you may see here is kind of a flatlining of this curve around 25% or so of patients who are on treatment. You'll see here this is from the initial publication at 24 months, but this has been followed even longer with subsequent publications, that there is a subset of patients who are treated with these agents, with this combination of treatment, that can be alive in the long term on immunotherapy. The chemotherapy tends to fall off after the first 6 months or so, and that have maintained control of their disease. For the most part, these patients are unfortunately not cured of their disease, but they can essentially live in an equilibrium where the immune system on continued immunotherapy keeps the cancer under control. The holy grail of most of medical oncology is how do we find out who those patients are ahead of time so we can select them? And how do we learn from these patients about why these treatments are more effective for them and apply those lessons to all the other patients that are not getting that same kind of benefit for immunotherapy. And the honest truth is, despite the fact that these studies have been around for a couple of years and then multiple similar studies have been published for over a decade in a variety of other cancers, there has not been a single smoking gun or even a particular good constellation of things that we can look at as far as the tumor genetics, understanding the immune microenvironment that allows us to confidently predict who these patients are going to be. And so this work is something that's ongoing and that's going to require a lot of further research. The title of the talk and was about personalizing treatment. And so I think here the point is that in first-line treatment of metastatic cholangiocarcinoma, the way to personalize treatment is to look for trials, right? And so this is something that all of us as medical oncologists are passionate about, all of us who work in academic centers. This is a big part of what we do. And we work with industry partners, we work with foundations, we work with other institutions, cooperative groups, the government institutions, to try to come up with new approaches for treating patients, and all that has to be done through clinical trials. And that's the way that we personalize treatments for patients in the first line. And I think broadly speaking, we can think about two ways to do that for first-line treatment, right? Because first-line treatment is established, it improves survival. We don't want to compromise on that. We want to improve on that. And so the way that you personalize treatment is either you add a medicine to the current standard chemotherapy and immunotherapy combination that you expect might help most patients, and that would be either another chemotherapy, Abraxane comes to mind, another immunotherapy, a CTLA-4 antibody, for example, or targeting of a VEGF, which is vascular endothelial growth factor, which is a blood cell receptor that's been targeted successfully in a variety of different tumors. So an approach like this would be potentially applicable to all patients and could be looked at in combination with the standard current regimens. The other alternative is to look at adding a targeted therapy, so picking a specific target that is only expressed by a smaller subset of the overall patient population, using a medicine or an agent that's already shown to be effective in later lines of therapy, and we're going to see some of these agents later on in the talk, and then adding them to first-line current chemotherapy, immunotherapy combinations. And I'll just show you briefly an example of this. I know we're going to hear more about these types of trials in ongoing talks that are coming up. So firstly, so this is a first-line clinical trial, randomized phase 3 study that's looking at adding a HER2 targeting antibody called zanidatamab to first-line chemotherapy and immunotherapy. So this is a randomized study. Patients are either going to be getting the standard of care, and that's always important, right, with randomized phase 3 studies, is that you're at least getting the standard of care, chemoimmunotherapy. And then the other half of patients will be randomized to be getting this HER2 targeted antibody. And again, this is a biomarker-selected study, meaning that patients have to have overexpression of HER2 to qualify for this study. And this is a study that's open nationally and open here at NYU. And that's just one example of the type of approach that can be looked at for personalizing treatment in first-line metastatic cholangiocarcinoma. So looking at the second line, this is where the personalization story kind of becomes much more relevant. So once we have progression after first-line treatment with a chemotherapy and immunotherapy, looking at what are the mutations that the patient's tumor has is really fundamental for choosing the best option of treatment for them. So we looked at some of the mutations based on the first slide, and I listed FGFR2, which, as I mentioned, about 10 to 20% of the intrahepatic cholangiocarcinoma tumors will have alterations in this gene, and there's 2 FDA-approved therapies. We'll look at them briefly. We look at IDH1, which is, again, mutated in about 20% of primarily intrahepatic cholangiocarcinomas, and again, there's an FDA-approved therapy for that. And then HER2 in about 10 to 20% of cholangiocarcinomas, more in extrahepatic than intrahepatic, have either amplification on a genetic level or overexpression on a protein level of HER2. And there's now a couple of FDA-approved therapies targeting that pathway as well. So just to go through these one by one. So for FGFR2, there are 2 targeted therapies. Pamigatinib was the first one, as I showed in 2020. That was approved, and interestingly, from a drug development standpoint, us in medical oncology will say, it's interesting to see in the 2020s, we're seeing approvals based on what are not randomized studies, right? So these are single-arm studies, including patients with previously treated cholangiocarcinoma that had these mutations. You'll see the study has about 100 patients, and it showed that about 40% of patients had what's called an objective response rate, and just to kind of explain what that means that about that 30% of the tumor, that there was a shrinkage of 30% of the tumor or more in those patients while they were on the treatment. And so that's a benchmark that we use to determine if a patient's responding for clinical trial purposes. 88% of patients had disease control. So disease control means you either had this objective response, which is shrinkage of the tumor, or you had what's called disease stability, which means the tumor neither shrank 30%, but nor did it progress, which is a growth of 20%. So that shows that almost 90% of patients at some point on the study with this medicine had some control of their cancer for a period of time. The median duration of response, which is the time that patients who were on the treatment had benefit of the treatment until they had to discontinue it for progression, was 7.5 months. So the effect is there, but it's not particularly long-lasting, and that's unfortunately a rather common finding in targeted therapies in cancer, which is if you find a good target and you have a good drug and you block it well, you can see really good responses. But the reality is that tumors often develop resistance under the selective pressure of these medicines. And so that's a barrier that we always have to overcome, and we're always looking for what are the mechanisms by which these tumors become resistant to look for next lines of treatment. Futibatinib was the next one that was approved by the FDA in 2024 based on a very similar study, single-arm study of 100 patients, and you see that the numbers look very similar. So both these drugs are options for patients with these mutations. Just to move to the next slide. So IDH1, I mentioned again, is a commonly mutated gene in intrahepatic cholangiocarcinoma. This one was actually approved based on a randomized trial. This was a 2-to-1 randomization of patients, again, that were previously treated. So all these patients have had previously chemotherapy. Most of them had not had the GemCis plus immunotherapy because these studies were done in the 2010s. And in this case, they had IDH1 alterations. It was a 2-to-1 randomization, meaning two-thirds of patients got the study drug and 1/3 did not. And the study was powered to look for progression-free survival, which is essentially the amount of time patients are on treatment until the cancer starts to grow. And you can see on the curves here that there's a big separation. The blue is the patients who got the study drug. But the actual improvement for the average patient was quite low, 2.7 versus 1.4 months, right? If you're looking at 50% right here. But if you see a lot of patients, maybe 20-30% of patients had controlled their disease much longer than that. And then importantly, the overall survival was also improved by using this medicine. And this is despite the fact that 70% of patients who were on the placebo group on this study at the time of their tumor progression went on the active medicine. So starting it earlier was better and patients lived longer. And so this is now FDA approved in the second line. HER2-targeted therapies is probably the most active area of current research. Again, about 15-20% of patients would qualify for these types of approaches. There's a couple different ways to target HER2. One of them is what's called an antibody-drug conjugate, which is this drug with a mouthful of a name called trastuzumab deruxtecan. So it takes an antibody that targets HER2, and it's bound to a very potent chemotherapy molecule. And this was approved in 2025, and it was actually approved in what's, again, called a tumor-agnostic approval, meaning there was a trial that looked at patients that were overexpressing HER2 in a variety of different tumor types that had been previously treated with standard chemotherapy options, and all of them were treated with this. And you'll see just from this brief slide, cutout from the slide here, that in biliary tract cancer, which is primarily cholangiocarcinomas, patients who had IHC3+, which is the highest level of expression of the HER2 protein, had a response rate of 56% with this medication. A similar study, which was the HERB trial, was a phase 2 trial looking primarily at cholangiocarcinoma patients, again with HER2 overexpression. And in the population that they presented of HER2 3+, which is the higher expressors, or the 2+ FISH-positive patients, which is, again, considered HER2 positive but lower level of expression on the tumor. Again, there was a response rate of around 36%. And so this has led to an FDA approval. And then more recently, zanidatamab, which is a novel HER2 targeting monoclonal antibody, so without that conjugation to a chemotherapy, was FDA approved, where it was shown to have a response rate of 41% with a significant median overall survival in about 80 patients. So these are drugs that are in development. As I showed you, there's currently a first-line trial ongoing with zanidatamab based on these exciting data. Looking at some kind of much more rare subtypes or genetic mutations in the tumors, BRAF V600E, we see in about 5% of cholangiocarcinomas, primarily intrahepatic. There's an FDA-approved therapy, which is a BRAF inhibitor, dabrafenib, plus a MEK inhibitor, trametinib. And NTRK, again, mutations are seen in about 3% of intrahepatic cholangiocarcinomas, and they're FDA-approved regimens. Again, both of these drugs were studied in groups of patients with a variety of different tumor types that just had this particular mutation, which is kind of a newer way of approving targeted therapies for rare tumor types or for tumors that, for mutations that are only seen rarely in certain tumor types. And then just as I promised to get back to the surgical setting, right? So after surgery, the surgeons have done their job, the patient's recovered. They come see the medical oncologists, who are the ones who are going to follow the patients for recurrence with scans, with blood work. We do talk to patients about postoperative or what we call adjuvant chemotherapy, right? And the idea there is, even though the surgery is successful, the margins are negative, there's no sign of cancer in the body, we know with certain cancers, and biliary tract cancers are among the most notorious, despite all those good things, the risk of recurrence is too high, right? And this has led to us studying, us as a community, as a field, adjuvant therapy, postoperative therapy, to reduce the risk of recurrence. And I think it's always a tough conversation to have with patients, right? Because they're just coming out of a surgery, everything goes well, they have no cancer that we can see, and now we're telling them that they need more treatment. And I think the important way to describe it to patients, and the way that I talk to patients about it, is that we know from the data, from looking at 100 patients in similar situation, that X% are going to have a recurrence of their cancer if we do nothing, right? And that X% depends on the staging of the tumor, how advanced it was, how many lymph nodes were involved, how aggressive does it look under the microscope. But we know that risk is too high to stomach, and that we can do something after surgery to reduce that risk. So the BILCAP study, which was published about 7 years ago now, was famously technically a negative study, as some of our colleagues like to remind us at every meeting. But despite that fact, those of us who treat these patients will still talk to most patients about this treatment. So it uses capecitabine, which is an oral chemotherapy pill. And the study was randomized versus placebo, and patients were treated for 6 months with this pill. And you can see here that the curves are clearly separated, meaning that patients who received the capecitabine were more likely to be alive and live longer on average compared to those who got the placebo. The study was technically negative because of the statistics here in the intent to treat analysis, which is the appropriate way to analyze the study. But if you took out the handful of patients who never actually got treated on study in what's called a per protocol analysis, you see these curves look exactly the same, but the statistics actually show significance. And so most of us look at these numbers and at these curves and tell patients that we still think it's worthwhile to reduce the risk of recurrence by about 20% in taking 6 months of chemotherapy. Now, capecitabine isn't easy, and so it's not an easy 6 months, but we can get patients through that. As far as personalizing adjuvant treatment, right, there are trials that are looking at doing better, right, because obviously these curves are not flat. We're not, preventing recurrences in all our patients. And so a recent study that I can point to as an example was a study called Artemide Biliary 01, which we actually had open here, but it was an international randomized trial that looked at the addition of an immunotherapy, in this case a drug called rilvegostomig, which is a bispecific antibody that binds both PD-1 and TIGIT, which are both immune receptors. And that was added to standard of care chemotherapy. It was a randomized study and placebo-controlled, and we got the email, I think, a week or two ago that it completed accrual. And so we'll see the results of that probably in a couple of years. So just to summarize, I know I threw a lot of graphs up there, a lot of names of a lot of complicated sounding medications, but the big takeaway is that in first-line treatment for metastatic cholangiocarcinoma, gemcitabine and cisplatin chemotherapy, when combined with an immunotherapy that blocks either PD-1 or PD-L1, in this case durvalumab or pembrolizumab, is the standard approach and has been shown to improve survival compared to standard chemotherapy alone. But the way to improve on this is trials, right? And that's the key here. And that adding targeted therapies for specific subsets of patients that harbor mutations that we know that we can target is one interesting way of trying to improve on those first-line therapeutic options. And I showed you one example of that with a HER2-targeted therapy. In the second line is where we really can start targeting our therapies for our patients, and that's based on their mutations, right? And that comes down to, it's really fundamental as a medical oncologist to sequence your patient's tumors early when you first meet them, to have that information on hand, and also to sequence them at time of progression, right? So most of us will do our best to get repeat biopsies at the time that a tumor progresses, because we know there can be changes in the tumor's biology that can help inform and allow us to select more targeted therapies for them. And lastly, in the postoperative setting, that capecitabine is generally used to reduce the risk of recurrence, despite it being a negative study, and that further clinical trials are ongoing to see if we can improve on that capecitabine monotherapy, for example, by adding immunotherapy. Just want to thank my patients and their families. Obviously, that's why we all do this, and we know as GI doctors that these are hard diseases to deal with as the patient. These are hard treatments to deal with. Clinical trials aren't easy, and the family support is everything to our patients. The organizers for inviting me, the Cholangiocarcinoma Foundation and Dr. Spencer, and then to my colleagues, oncology really is a team sport. We work with surgeons, radiation oncologists, GI, IR, radiology, pathology, nutrition, and then the clinical research teams that are really fundamental to getting these trials done. And I'm happy to take any questions. No questions.
We're going to hold questions till after the next presentation, and then I think we're going to do a little case panel. So I get the pleasure of introducing Dr. Kristen Spencer. She's a medical oncologist and director of the Phase 1 program here at NYU. Her commitment to cancer care is deeply personal, shaping by losing her mother to metastatic colorectal cancer at a young age. Today, she focuses on expanding patient access to innovative clinical trials and new therapies, particularly for pancreatic and hepatobiliary cancers, while partnering closely with patients to support both treatment decisions and quality of life. I also want to say I've worked with her over the years, and she is a true patient champion. She does a lot of research in cholangiocarcinoma. She's done surveys with me. She's brought the patient voice to many presentations at ASCO, and I just appreciate her so much. Thank you for being the first one to do a New York City, Chicago Symposium. Chicago, New York City Cholangiocarcinoma Symposium.
I was going to say, am I doing Chicago's also? Oh, God. I haven't invited anyone to Chicago's.
Chicago replaced cholangiocarcinoma.
Close enough. All right, so the first thing I'm going to say is I apologize that my slides are not on the right template, but I made them and I have absolutely no idea how to get them onto a new template the right way, so I figured they're just going to stay as they are. And my role here is to be very briefly up on this stage because you've seen me enough today, but to give you a primer for Dr. Harding to then come and talk about clinical trials. And the reason I thought that this was, or I should say emerging therapies by way of clinical trials. And the reason I thought that this was really important was because as I spend time in clinic with patients, it has really come to my attention that the education that we give patients around clinical trials is really poor to non-existent. Most patients come into my clinic ready to sign for a clinical trial without even hearing what a clinical trial is, what it involves, how it's different from standard of care. And I think we really are sort of doing them a disservice. So this is, again, meant to be a primer. It's not meant to cover everything. These are the things that I most commonly hear about in clinic that I thought might be practical for you all to hear, and hopefully we'll set the stage for Dr. Harding to come up, talk about the good stuff. So, and I should mention that we had Dr. Winograd up here who was talking about standard of care, meaning things that are already approved through your insurance and the FDA, as well as things that are in the later lines of therapy, meaning that they've made their way through most of the clinical trial process and they're near approval. So we're going to focus on what's going on in the early phase. What are the emerging therapies? So what do I mean by an early phase clinical trial? This means generally a phase 1 or a phase 2 clinical trial. I'm going to tell you what that means in a second. A phase 1 clinical trial is the very first step in testing a new drug, new combination of drugs, or a new treatment in people. And really, we have two very specific goals at this point. Yes, I, as your provider, am interested in whether this drug is helping you. But the trial is asking, is it safe? Meaning, what are the side effects? Can the body tolerate them? And we do this by predefining what we call dose-limiting toxicities and collecting from you all the toxicities or side effects you're having and assign them a grade, meaning how severe they are, and whether we think they're related to the medicine. And then a second goal of what is the right dose. And so how we do that is by starting with a very low amount and slowly increasing in cohorts of patients. So this is what we call a dose escalation. And we do this in different groups of patients to find really what's going to be the safest and most effective dose for future studies. So to put this in the context of clinical trials, generally speaking, you can see on the left, we have discovery, preclinical, we spend time in the lab with mice, we figure out where we want to go next with the medication, and then it enters a phase 1 trial, where again, we're looking at safety. This is anywhere from usually as few as 10 patients to as many as 80 patients. It takes about a year to complete, sometimes can be done faster than that. However, most of these trials are enrolling patients with what we call all solid tumors, so any tumor type available and eligible to enroll. It's not until we really hit the phase 2 setting where we're starting to look at patients with certain tumor types. So we may say, we had a couple patients in the phase 1 trial with cholangiocarcinoma that did really well with this drug. Let's now look at it in 50, 100 patients with cholangiocarcinoma, and that's a phase 2 trial. A phase 3 trial, same thing. Let's look at it in patients with cholangiocarcinoma, this time 500, 1,000 plus patients, and let's compare it to some sort of standard of care and see if it's better. So that's the general framework, okay? So what does this mean for you, participating on an early phase clinical trial? A couple of things that I tell my patients. The first being, while we have a sense of the expected dosing and side effects before we start, you are all a part of completing this information. So I tell my early phase clinical trial patients that they are some of the bravest people that I know. You are monitored closely on the trial. We don't leave you out, don't see you 6 months later on an investigative agent. We don't do that. But what this means for you, you are likely to be with me many times in clinic, sometimes weekly, sometimes more than once a week. Those days can be long. So we're trying to understand not just what the drug is doing to the body, is it helping your cancer, what side effects is it giving to you, but what is the body doing to the drug? So often we're measuring drug levels over time. So you may have taken a pill at 8 a.m. and you're just sitting in a chair for the rest of the day while we draw blood levels and actually look at those in groups of patients to see whether we can correlate who's having side effects, who's having a response, and does it have anything to do with what the body is doing to the drug. There may be biopsies involved, sometimes more than one biopsy. There may be a biopsy to enter. There may be a biopsy, what we say, on treatment after you've had the medicine for a certain period of time. There may be tests that you have never done before, you have never heard of before throughout your cancer journey, things like echocardiograms, eye exams, skin exams. These are all done because we have some information about the medicine that we need to know about you, whether you're going to be safe taking it. And I like to tell my patients that clinical trials in the early phase space, it's basically like controlled chaos, particularly in the beginning. So I will tell you to please be as flexible as you can, bear with us. It does get better, but controlled chaos is pretty much an expectation. The other thing that's really important to know is that this means we have very limited information on the activity of the drug in your specific tumor type, so in cholangiocarcinoma, for instance. So at this stage, we may not be able to give you great estimates of how likely the medicine is to help you, how long it's likely to help you, but we, again, are not going to keep you on a clinical trial if it's not helping you. We, as your providers, care about that information, even if the trial is trying to figure out dosing. So the flip side is you might then ask me, well, why the heck would I participate on one of these things? Well, the caveat is that, or I guess the other hand, is that you get access to a promising drug, possibly as much as 10 years prior to its approval by the FDA. So that's why you might want to participate. Some common questions that we get. Can I participate on a trial but have all the assessments done at my local oncologist's office? And unfortunately, at this time, as much as we're trying to decentralize clinical trials, use telemedicine, things of that nature, for the most part, the answer is no, with the exception of sometimes maybe we're going to just check your liver function for a one-off and see how you do. You might be able to do that close to home. Everything else has to be done here. Will my current oncologist continue to care for me? Again, unfortunately, the answer is no. So your care actually transfers over to the care of the physician who's enrolling you on the clinical trial. If that was the person that was caring for you the whole time, then wonderful, still remains your oncologist. But what I can say about all of us is that we really do try and keep your prior oncologist up to date on how you're doing, particularly if we're getting a sense that you're maybe struggling a bit with side effects or things aren't going well. We do really re-engage them early. How long will the study last? Well, typically, like your chemotherapy, it will go on indefinitely as long as 3 things are met. One is that the drug is helping you. Again, no new cancer spots. The spots that we knew about when you started are the same or smaller. As long as you're tolerating the drug, so I will remind everyone nothing comes for free, but when we say tolerating, what we mean by that is that we're managing it with medications, whether they be prescription, over the counter, transfusions. We can hold the medicine, we can reduce the dose of the medicine. We have a lot of things in our toolkit, but we're managing the side effects. And then the third is that you want to continue. So I will remind all of you that a clinical trial is always at every single part of the clinical trial voluntary. So that doesn't end when you sign the consent. You have the right to remove your consent at any time. Will I get a placebo? Typically, no. Placebos, again, are used really in a phase 3 clinical trial when we're trying to determine if one medicine is better than another that is already FDA approved. I actually have to tell you that in cancer clinical trials, placebos are even rare in the phase 3 setting. Oftentimes, we're comparing to a standard of care the addition of something else. But the phase 1 level, you're not going to see a placebo. Will you continue your current chemotherapy? No. So this will be in place of your current treatment. And I'll remind everyone that the reason that you're probably being referred for a phase 1 trial is that your current treatment is either potentially not working as well as we'd like it to anymore, or it's making you too sick. So there's a reason that we want to take that away and give you something else. So who pays? Well, complicated question. I know Melinda referred to this. You heard her talk about this when she was enrolling on a trial many years ago. It's gotten better, but I would say anything that we are typically doing to monitor or treat your cancer is still going to be billed to your insurance. So you will still have copays, CAT scans that we would normally be doing, accessing your port by the nurse. All that will be billed to your insurance. So if they do not cover something currently, for instance, they never want to pay for the medicine, the nausea medicine Zofran, they are still going to be a pain in the tush about it when you enroll on a clinical trial. That's not going to go away. They don't care. Everything else, though, that we would not otherwise be doing, so these are things like the screening tests that I mentioned, providing the drug itself. These will be paid for by the sponsor of the study or provided by the study. How far should I travel? I get this one a lot, and I think this is a really tough one as it's a very personal question. But what I would remind everybody is that the schedule is going to be very hard at first, and how you're feeling at the beginning of a study is not how you're likely to be feeling after you start that study. So add things like potential side effects, add the schedule of coming in and out, back and forth. You have to go wherever you have a very viable plan to stay nearby for long stretches of time and get back and forth when you need to. And then finally, all of these questions: what is the idea behind the trial? What is the data so far in support of it? What will the schedule be? How many times do I have to be here? What will the side effects be? What will be required of me? Will there be biopsies? What will the sponsor pay for? What will be out of pocket? Will participating on this trial make me ineligible for X other trial I want to do? I'm going to put under one blanket of wonderful questions. Really depends on the clinical trial, so ask us. We are more than happy to answer those questions. We're happy to help you strategize. I say to my patients who have multiple clinical trials as options available to them, this is a wonderful place to be in, but really hard to make decisions. So ask us to help you. That is what we are here for. And I think that is it for me. And now, without further ado, I will introduce Dr. Jim Harding, who is a board-certified medical oncologist at Memorial Sloan Kettering Cancer Center, our neighbors just up the street, who are collaborating with us on this symposium today. And he specializes in treating liver, gallbladder, and bile duct cancers, as well as other gastrointestinal cancers. He is deeply committed to providing individualized care and works closely with a multidisciplinary team to ensure patients receive the best possible treatment and support. So please join me in welcoming. He is going to talk about emerging therapies to look out for. So some of the things we've been investigating in these trials I was just referring to.
Thank you so much. I really appreciate the invitation to speak in front of you today. Dr. Spencer, I noticed that you referred to the surgeons as being really tall. I'm 6'1", and I think I am wearing some green as well on my tie. It's a pleasure to be here with you, and as stated, I really focus on the treatment of patients with bile duct cancers and liver cancers. And I've been at Memorial now for 15 years in this space. And here are my disclosures: grant support, research support, and various consulting. And today's agenda for me, I'll expound on some of the things that were already talked about We'll expand on precision medicines. How can we make those better? Talk about novel drug classes. How can we make this more broadly applicable to patients that don't necessarily have targets for gene-driven medicine? And then we'll talk about targeting the immune synapse and maybe end a little bit with some perioperative approaches. And so, as was stated by my colleague earlier, the general treatment paradigm for patients with biliary tract cancers is as described here: chemoimmunotherapy, then really matching to precision approaches based on the underlying molecular events. But of course, clinical trials are really critical to advance the science and advance the field. And I know the audience is comprised of some physicians and also people not in medicine. What is precision medicine, right? It's important, I think, for the discussion. The gene is the blueprint, and the gene makes a protein, and that protein makes the work happen. It's the cells, the underlying building block of cells. And so in cancer, one of the hallmarks of cancer is the genes can become mutated, and that then makes a mutated protein, and that protein just doesn't listen to what it's supposed to do. It keeps making its action and is one reason that we develop cancer. And so precision medicine, which you heard a lot about, is when we use a small chemical, a small molecule that can fit into the protein that's abnormal and disrupt it. Usually these are what are called small molecules. Sometimes they're called antibodies. And as you saw earlier, there are a number of precision targets that can be leveraged to treat cancer. One not on here are NRG1 fusions with a novel bispecific, and we'll hear more about that at another time. But over the last decade, the amount of precision medicine in biliary tract cancer has increased pretty substantially. This is data from Memorial. We've now sequenced about 3,000 to 4,000 tumors of patients with various forms of biliary tract cancer. And what I'm just showing you here is the level of actionability based on a gene report from 2017 to 2023. Green means there's an FDA approval. And so for all bile duct cancers, we're about 30% actionable. For intrahepatic, it's approaching 50%. So understanding the underlying gene is critical, important to treat the disease. And from this work, we really have documented that you must do the molecular profiling. We've done work with liquid biopsies where you can detect those abnormal DNA fragments in the blood, and that seems to work very well. We also do protein staining. And so my pitch here is, of course, always do and know the genotyping. And that's important because one of the studies that I was intimately involved with in co-led was that of zanidatamab. It's a targeted therapy for HER2 amplified or overexpressed bile duct cancers. And, if you find an alteration, sometimes that can be quite transformative. One of the reasons why I got into medicine is I like to make people feel well, right? And what I'm showing you here is the first scan of a patient that had extensive bile duct cancer in their lungs. Now, you don't need to be a doctor to know that this must be a lot of suffering, and it was. This is a patient with bile duct cancer, had standard treatment, it failed. There was no effort to genotype necessarily at that time. We did it through our molecular profiling and linked her to a clinical trial. Very short of breath when I first met her. In fact, the first day of treatment, I couldn't be there, and someone called and said, listen, very short of breath, should we go to the hospital? I said, no, give the treatment. And 8 weeks later, you can see all of this white gunk in the lungs was really almost gone, and that continued for really over a year plus and really reconstituted health. So we need to understand and know those mutations. But one of the problems and issue number one for precision medicine is that only a proportion of patients with biliary tract cancer with these abnormal genes will respond to a targeted therapy. And the reason for that is multiple, but we have to think about cancer as not one entity. Here, multiple people can have the same cancer, but if you compared those cancers, they would be different. In an individual person with multiple sites of cancer, the cancer may all maintain that initial gene problem, but they may have subtle differences depending on where they are. And if you take out a tumor, there are various different types of cells within it. It's not all the same. So cancer is, under the same flag, a legion. It's multitudes of the same thing, but they're all slightly different. And so that's called heterogeneity, right? And so one thing that we can do, particularly with relationship to HER2, is can we understand HER2, heterogeneity in these patients with HER2? And in fact, one thing that we're trying to do across all of these cancer-specific mutations is can we identify patients upfront who may or may not respond? And one way that we're doing it in a hopefully soon-to-be funded grant is to use imaging, an external imaging modality, to look at the gene and protein product throughout the whole body. So you've heard of HER2, you've heard of PET scans, right? It's basically an image that shows the whole body, and wherever the cancer is, it lights up. That's usually using sugar to do that. But here, we're actually developed a molecule where you can look throughout the body for where the HER2 expressing cancer is. And what our goal of this research, and other people are doing research like this, is to sample different parts of people with cancer. And we've already genotyped over 4,000 samples, probably 20 to 200 paired samples. What we're going to do is look at HER2 expression and heterogeneity. We're going to see how that compares to this imaging, and we're going to assess that based on how treatment may play out. And so this is an example of sort of this preclinical or translational work that Dr. Spencer spoke about. We're going to understand better how this specific mutation in biliary tract cancer works, such that we might be able to select patients better and might be able to define new therapies. Another solution to this idea of why doesn't everyone respond, it comes back to that heterogeneity. Maybe the cancer contains all of the same genes and proteins, but they're all slightly different. We might need to target multiple nodes in the pathway of cancer. And one such study for HER2, which I co-lead, is really already discussed. It's a randomized study, but what's interesting about it, it is adding zanidatamab upfront, a HER2-targeted therapy, with chemotherapy, with immunotherapy. So we're blocking multiple pathways in cancer at an earlier stage in the disease, and the hope is that this would, lead to a better outcome. This is not the only study that's doing this. So there's, for IDH1 mutant tumors, there's a frontline study now looking at chemotherapy, immunotherapy with the drug ivosetinib. There is, for other HER2-targeted cancers, combination immunotherapy with a drug called trastuzumab deruxtecan. And the idea behind this, very simply, is can we extinguish the heterogeneity of cancer by doing rational combinations of treatment? Another issue for precision medicine is if there's a response, remember, it's not all the same thing. It's a little bit different. Some cancer cells don't like that targeted therapy. They hate it and they die. But other cancer cells have mechanisms by which they can escape it. And what's been documented for particularly another type of biliary tract cancer, FGFR2 fused, is that when you use an inhibitor of that pathway, multiple resistance mutations develop. And that brings up the idea of, well, can I then sequence therapy? And that's happening too. Right now, there's a phase 3 clinical trial that's evaluating sequential treatment for patients with FGFR-altered cholangiocarcinoma. The idea is to take patients that have had already 1 or 2 lines of prior therapy, chemoimmunotherapy, an FGFR inhibitor, and then give a novel drug called tiningatinib versus a physician's choice chemotherapy, with a hope, again, that if we can sequence therapies, we may lead to a better outcome for patients. And so we're seeing that in the space of precision. But one thing that I'm always cognizant of, and when I interface with patients, there's a lot of disappointment when people hear, well, I don't have a specific mutation. So what am I going to do if I don't meet one of these categories? And that's important. We need to be aware that we have to evaluate multiple different ways of treating cancer to see if there's a way we can improve outcomes. And thankfully, over the last 5 to 10 years, there are a number of new targets for bile duct cancer that were previously thought not to be druggable or even weren't considered. And I can't, in the interest of time, go over every single one of those, but I'll say that KRAS mutations, MTAP deletions, MET and MDM2 amplifications, these are genetic abnormalities that lead to abnormal proteins that now, in current drug development, there are inhibitors of. And the one that I'll just speak about is KRAS mutations here a little bit, because I do think it's important. From our experience in our first report of about 1,200 patients, genotype 17% had mutations in KRAS. It's a gene that makes an abnormal protein. And there are specific mutations in this that are in cholangio, intrahepatic, extrahepatic, and gallbladder cancer. And what we're starting to see is that we can use different drugs to block these pathways. One of them is already available for a rare type of KRAS mutation. It's called KRAS G12C. And this drug in small trials showed about half of patients with a KRAS G12C mutation had a response and benefit, and this was in the context of a basket study. But we're expanding now the availability of KRAS-targeted therapies, and this is mostly occurring in a disease that's similar to bile duct cancer, pancreas cancer, at least for extrahepatic. But people are looking at a variety of different drugs for that, with some called pan-RAS inhibitor, isoform selective inhibitor, novel combinations. And so I think over the next several years, some of these targets will show merit and have further development. Right now, most of these are in phase 1 drug development, which was just talked about by Dr. Spencer. Another issue is, can we keep expanding, right? So what I told you before is that the gene is abnormal. It makes an abnormal protein. We can make a molecule that targets that protein, yeah? But there are also a number of proteins that are overexpressed in cancer. They may not necessarily be mutated, but they may be preferentially expressed in cancer. Maybe, it's more critical. They can be inside the cell. They could be outside the cell. It might be useful for the function of cancer. And we can use those proteins with modern chemistry and biochemistry like a honing beacon. So we can exploit the cancer-associated protein. And there are a number of ways we can do that. I'm going to show you a few examples of this. Again, the things that are kind of being developed across solid tumors and even cholangiocarcinoma. And so some of those molecules might be something called an antibody-drug conjugate, a radioligand, a degrader, or even cellular therapies. And so antibody-drug conjugate is a unique class of drug, and the way that works is, our immune cells, or B cells, they make antibodies. And the antibodies are designed to surveil and kill bacteria and proteins. And they basically can bind to these pretty well. And what we can do is manufacture antibodies where we then put something that cancer doesn't like on them. So like a chemotherapy being common. And so a protein that's preferentially expressed on cancer is the honing beacon. And then this antibody comes along, it gets brought into the cell, it releases whatever its warhead is, its payload, something toxic to cancer cells, that then kills the cancer cell and then leaches out to other cells around it. And so there have been a number of FDA approvals for antibody-drug conjugates across solid tumors, specifically not in biliary tract cancer per se, but in HER2 overexpressed cholangiocarcinomas and bile duct cancers, there is an agnostic approval for an ADC called trastuzumab deruxtecan. But we need to look at this in more depth, and we and others have looked at this using patient samples from resected samples. So what we did in a project led by one of our fellows, basically we looked at common antibody drug conjugate targets like TROP2, c-MET, Nectin-4, Claudin-18, HER2, which is already has an approved space, but it's not very common. And what we saw was that across all types of bile duct cancers, there's some positive expression, and some of these tumors are very heavily expressing. The darker the brown means the darker the expression. These are just panels of patient tumors showing that these are potential targets. And indeed, right now, there are trials ongoing with antibody drug conjugates to TROP2, and that includes cohorts with biliary tract cancer, a protein called MET, a protein called Claudin 18.2, and we've put together a study based on this and some preclinical data, which is ongoing, looking at Nectin-4 as a potential target, and that study will open at Memorial shortly. So antibody drug conjugates, I think, are a way to expand therapeutics to patients with biliary tract cancer. There's also some thought that these can be paired well with immunotherapy, and there might be some synergistic effect. That's also occurring across drug development and solid tumors. We've seen a great deal of success with that related to bladder cancers. And we do, as I've stated, for at least HER2-positive disease, have an ongoing phase 3 study there. Another way to expand the arsenal, and this is still a little bit far away, but we're starting to see this percolate, is what's called radioligand therapy. And so you talked earlier today about Y90. There was a talk about external beam radiation. You use basically high-intensity radio waves to kill cancer. Radioligand therapy is a mechanism by which you can put a radioactive particle, this is the symbol for radioactivity, you can attach it to an antibody, you can attach it to a small molecule, and that can hone to proteins that are abnormally expressed in cancer and deliver to that cancer cell radiation. And so I like to refer to it as liquid radiation. And the way it works is you first get a specialized scan that targets or visualizes the tumor to see if there's expression of its target, and then you follow that with an infusion of the same molecule, but now it's radioactive and it goes selectively to the tumor. So we're seeing a number of different radioligand-based therapies now being evaluated across solid tumor, one being FAP, fibroblast-associated protein. This is a very early study with not a wonderful tracer, but showed some proof of principle that you could target cancer this way and that there could be some durable responses. But now there are many that are coming into the pipeline that are looking at a variety of cancers, including cholangiocarcinoma. And so that's another one that I think is quite interesting. And then finally, protein degraders. So I told you proteins are throughout the body. Some of them are mutated. We target with small molecules, but some of them are not mutated. They're just overexpressed, and they're really important for a cancer cell to grow. And with modern chemistry, what you can actually do is you can make certain molecules, they're called PROTACs or molecular glues. And what they do is they bind to your target protein that's abnormal, that's causing cancer growth right here. And what they bring it to is this special protein complex in cells that breaks down breaks down that protein. Like in our normal cell, we're making proteins and degrading proteins all the time. It's a constant process, making protein, degrading protein. And in this way, what is essentially happening is you can select an abnormal protein that then can be degraded. And in models of cancer, this is quite potent. And we're starting to see this for KRAS-altered tumors, but a variety of others that may have applicability. Again, this is a phase 1 type concepts, and you'll see these, and some will have cohorts for cholangiocarcinoma. I told you that cancer is a legion, right? It's not all the same cell. That's why it's hard. It's hard to manage. If we had one secret warhead that could kill it, we'd be all set. We don't have that yet. But we have our own legion, and it's a silly cartoon. I like to think that our immune cells are much, much, much tougher than this one. But we have a variety of cells in our body that are specifically designed to find and eradicate things that are not completely ourself. And it does a really good job because we're walking around in a mess all day and we live just fine, right? And these guys could include innate immune cells like natural killer cells. They can include, T cells and helper T cells. And what we've learned over this last, 10 years is that we can develop molecules to these cells. We can even reinfuse them, and this can be our legion against the legion of cancer. And so I do think there's going to be continued development across a variety of across a variety of cancer. I'm not actually going to spend that much time on this because it's really extensive. I only have actually a few more slides. But basically, we are going to broaden immunotherapy, and I think that will come to fruition over the next several years to decades. We've talked about drugs like durvalumab and pembrolizumab. Those are monoclonal antibodies. And what they do is they work in this immune microenvironment to unleash our T cells and even our B cells against cancer and kill cancer. So those are cells that are already there that recognize the cancer as foreign. And so what people are trying to do are make newer versions of PD-1 antibodies. They're called bispecifics, where one portion blocks something called PD-1, the other one blocks another immune factor called TIGIT. You can switch out TIGIT. You can put something called CTLA-4 there. You can put vascular markers like VEGF. And you can even, with newer chemistry, make like tri-specific antibodies that can look at multiple targets that bring the immune system together in such a way that you can fight cancer in that way. And this is one example of a drug called rilvegostomig, which was ongoing in a phase 1 study in the front line with gemcitabine that's showing some reasonable data of shrinkage and control. And the idea is that as we determine new immunotherapies, we need to bring them to the earlier stages of cancer, right? If you can remove cancer when it's small, you extinguish its heterogeneity, then you might be able to use combined therapy to treat the cancer in such a way. And you heard about BILCAP and probably ASCOT1 that demonstrated fluoropyrimidines were useful. This is the global Artemide study that is just, it's been enrolling patients where you basically have resected biliary tract cancers. You receive a standard chemotherapy up to the physician and the patient with a placebo, and then you have this with rilvegostomig. And the idea is to see if we can bring immunotherapy to earlier stages of disease. This isn't the only such study like this. There are a number of studies across the country for cholangiocarcinoma about looking at immunotherapy and combination chemotherapy before surgery to elicit an immune response. There are other modalities that are being looked at. This is just medical antibody-based therapies. People are now investing in vaccines and cellular therapies, which I won't go into at length, but we can talk about it in the questions. This I always find fascinating is the work of my colleague, Andrea Sursic. She asked the question, for certain cancers that have mutations in them, which we were talking about earlier, MSI high or mismatch repair deficiency, it's like 1 to 2% of biliary tract cancers. And she asked the question in colon cancer, well, we know those are really immune-sensitive cancers. Can we give immunotherapy sort of at an earlier stage of disease? And they did this in colon, and essentially they were able to remove the need for surgery. This study continues to enroll, and we are enrolling patients with biliary tract cancer as well. And so this is a small proportion of patients, but my hope for the future is one day with this continued drug development, which truly is a partnership with many, many people and you, that we'll get to a point where we can do that for more patients. And so, at Memorial, I just highlight, for each stage of disease, we have various studies that are open for biliary tract cancer, running the gamut from phase 1 to phase 3. And my hope is that together, we're going to see, greater improvement in this disease. My conclusions would be mutational profiling is critical, and it's really important in the current age of treatment for biliary tract cancer. So much so, it's always in the back of my mind, I got the question wrong earlier today. I didn't see the mutational profiling. The ongoing work is really focusing on improving precision approaches by having biomarker selection. I think we're going to see inroads with RAS targeting, perhaps MET and MTAP-deleted tumors. We need to expand the availability of quote-unquote targets, and I do think that will occur with the use of ADCs, potentially novel IO therapies, radioligands, protein degraders. I do think the evaluation of these compounds now at earlier stage of disease is critical. We've seen it across a number of studies that this is really important. And then I'll end with some thank yous. I thank Dr. Spencer for the invitation. The Cholangiocarcinoma Foundation, as always, work as part of a much broader group, all of my various funding sources. And if you're around on March 6th, I'm leading a Cycle for Survival team, so you can see me indoor cycling. So thank you so much for your time, and I'm happy to talk more and answer any questions, okay?
All right, and I'll have Dr. Winograd back up. So much like we had the surgeons do, we are going to also walk through a case. So this is actually a case of a real life patient who's been a patient of mine. And really the purpose of this is for you to hear different perspectives on how we make clinical decisions, how we decide what to recommend to you all at what point in time and why. Obviously, again, not meant to be all inclusive of every situation. We will then have a question and answer afterwards. And so welcome everyone to start thinking of your questions, and you're welcome to come to the microphones. But for now, we will start with a case of a 47-year-old male who had a past medical history of choledocholithiasis, so basically stone disease in the bile ducts. Had a cholecystectomy in the past for this. You'll note that there was a pretty significant family history of multiple cancers on the maternal grandfather's side and father's side. So how did the patient initially present? Actually with upper back or neck pain that was refractory to corticosteroid injections, not uncommon. They had been referred to rheumatology. Rheumatology actually did an appropriate workup for this and noted that the liver numbers, the LFTs, we call them liver function tests, were found to be high. They then did a spine MRI and actually incidentally noted a left hepatic lobe or liver lobe mass that was measuring almost 10 centimeters. And there looked to be some tumor in the left hepatic duct. They had some dilation of their bile ducts because of this tumor, and there was involvement of the vasculature that was in and around the liver surrounding this tumor. And these are abbreviations for lack of space to spell them out, but what's important to note is many of you are probably familiar with your bilirubin that we measure on all your blood work, so that level was 1.5, which is really a little bit above normal where we like it, right, sort of at the upper limit of what we like to see, at least as medical oncologists. So our next step in this scenario is often to get a liver biopsy to tell us what exactly is going on. And so unfortunately, in this situation, liver biopsy showed a moderately differentiated adenocarcinoma, and it was consistent. This is a very common finding we get back from pathology where they say it's consistent with a pancreatobiliary primary, meaning that, as you all know, I sort of explained to patients that the bile duct system is like one of the routes at a four-way traffic stop. And so the small intestine, the pancreas, the bile duct system, they all kind of come together at one big junction. And so it often is difficult to tell exactly where a tumor is originating when it's in that space. So we put together the clinical picture of what we're seeing on the scans, what we're seeing on the pathology, and the patient was diagnosed with a bile duct cancer. So just a little bit more workup. So we then did a CAT scan of the chest, abdomen, and pelvis. In particular, the CAT scan can give us a little bit more information about what's happening with the nearby vessels. The CAT scan of the chest can obviously tell us the extent of disease. And this unfortunately showed that there was a soft tissue mass on the left side. It was engulfing much of the left side of the liver, the veins, the branches of the pulmonary artery, the portal veins. This was actually compressing the vena cava in the lower belly, which is the large vein in the abdomen. And there were multiple lymph nodes that were enlarged sort of near, regional to the liver and near the tumor. However, there was no distant disease that was noted. And so this is where I think you heard Dr. Torres Hernandez talking about a PET CT. So I'm actually a very much also a firm believer in PET CT scans in patients that are otherwise considered eligible for surgery, because it has been demonstrated to actually not just change management, as he was saying, but actually upstage about 30% of patients. So we really want to know what the extent of disease is before putting someone through a big surgery. And so the PET CT showed this large mass. They had, again, a lot of dilatations. They had localized lymph nodes, but again, nothing that was distant. And I'll show you that on the next slide. So this patient had clinical stage 3 intrahepatic cholangiocarcinoma. Showing a video of what, I know I said I'm technical, I did not do this. My 26-year-old research assistant made that video, So just showing you the tumor. Looks pretty large, but the surgeons thought they could get that all out. And then there's the PET scan that's on the right side. All right, so I've already given it a little bit away, but I'm gonna now defer to my colleagues here. So what are you all gonna do next? And I'm giving you some options here. There's no wrong answer, I won't buzz you. You're welcome to add options if you'd like, if you don't like any of these. But would you take this patient for surgery up front? Would you do TARE, which is otherwise known as radioembolization, chemotherapy with a triplet of gem-cis-Abraxane, chemotherapy with gem-cis and one of the immunotherapies, or radiation?
So thank you. I think the case illustrates some nice points that, of course, you went to the disease management team and reviewed it in detail. For me, I think that although you alluded that this might be technically resectable, the tumor, quite large, seems to be impinging on vasculature. There's nodal disease. And my sense would be that surgery might be a very morbid in that there could be long recovery, and I'd be worried about micrometastatic cancer. And so my approach would probably be systemic therapy. In the current age, I'd probably do gem-cis durvalumab. But I think probably at the time of this, combination chemotherapy would be reasonable as well. The reason why I would try to go that route is because we know that this can shrink tumors. There's been data for perioperative approaches, particularly now out of China. You know, it also is associated with a known survival advantage, right? Like, I'm giving a therapy that I know extends life. And so even though I hope that we would get to surgery, if there's something that pops up in a month or two, then at least we've started with that. That's how I would approach it. I would do systemic therapy up front.
Yeah, so I agree. I mean, I think it would have been an interesting thing to have one of the tall, handsome surgeons here to pitch their, not my words.
I'm really getting insulted,
I know.
guys.
Okay, all right.
I mean,
You know, I was
next
gonna-
cholangiocarcinoma meeting, there better be some.
I was going to wait to the end, but I am getting so many. So let's give a round of applause to our tall, handsome medical oncologist.
But the point is, you know, these are the discussions we have in the tumor boards, and I think, you know, part of that back and forth is the surgeons kind of giving their thoughts on what the risks are for the patient with this particular tumor. But I think some of the key points, you know, were already made, which is the lymph node involvement is highly concerning, and I think part of our job as medical oncologists is sometimes to slow the horse down a little bit, right? The notion of getting the tumor out surgically is always very appealing to patients, and the surgeons with the amazing technical abilities they have, some of the new technologies, the robotics approaches that they have at doing these surgeries are able to do things technically, right, that may not have been possible 5 years ago, 10 years ago. But the biology of the tumor is one, as we know, that even when we have an R0 resection, meaning all the margins are negative, the risk of recurrence is really quite high, right? And ultimately, that comes not from the tumor that was taken out, but the tumor that's left behind. And that's, as we just heard, microscopic disease that we can't see on a scan. And for that reason, starting with systemic therapy first would be my preference. And in today's day and age, that would be Gemcis with one of the PD-1 or PD-L1 antibodies. You know, I think you put Gemcis Abraxane up here. There was some nice data from Dr. Maitel and others about that combination. I think this was in the pre-immunotherapy age, but showing the ability of that regimen to shrink tumors and get patients to surgery. So I think it's a reasonable option, but in the age of immunotherapy, when we know about the potential long-term benefits of treating with immunotherapy, and certainly in the neoadjuvant setting in a lot of tumor types, giving immunotherapy before surgery has been associated with improved long-term outcomes. That would be the approach I would take.
And may I just add one other comment? All right, thanks. So I'd also say that for the local therapies of TARE, so this is selective, you know, Y90 or internal, you know, radiation or external beam radiation, you know, both of those modalities have shown that they can potentially control the tumor for an extended period of time, but both of those modalities may have a negative impact on the remnant liver. And so I think that although those could be entertained, and you know, I think we could talk with the surgeons about it, if they think there is a pathway to surgery, it's really, I think, their call, because you don't want to diminish the liver function in any way. And so I do think Gemcis with immunotherapy would probably be safe, but based on this, I'd still pick systemic. What would you do?
Well, I was part of managing the patient, so I'm going to reserve my.
All right.
But no, I agree. I agree with you. Systemic, you know, for all the reasons that you all said. So I would do the same. And, you know, I would just add that, you know, this patient had the benefit of having a multidisciplinary evaluation, and I think that is the first step. That is the most important thing. So for any of you that, you know, now I'm considering all of you advocates, because we're informing you and giving you all this information. If you hear of folks that are diagnosed with bile duct cancers, they really should all be evaluated at an academic center of excellence where all of these disciplines are able to weigh in and make decisions. It's really incredibly important. And, you know, that's because you've heard from everyone today that these decisions are very nuanced, and everyone should be considered and have the option available for all types of therapy. So the last thing I want is a medical oncologist to make a decision that a tumor is too big and this patient can never go to the operating room and they're never afforded the benefit of that option. So that's the first thing I'm going to say. The second thing I'm going to say is this patient actually did start neoadjuvant chemotherapy with Gem/Cis Abraxane. And as Dr. Winograd alluded to, there was actually a feasibility study called the NEO-GAP trial that looked at giving Gem/Cis Abraxane prior to surgery for patients with high-risk disease. This was surgically defined. And that trial really just told us that this approach was feasible, but what that was born out of was some data in patients who had metastatic or locally advanced, not surgically amenable disease. And that came from a phase 2 trial that showed that this regimen had a lot of promise to it. And so this, I think, is a really a poster child of why we go through the clinical trial process, stage 1, 2, and 3. Because when Gem/Cis Abraxane then went into a phase 3 clinical trial, PI was Dr. Rachna Shroff, you may have heard of her. That trial ended up being a negative clinical trial. So while this was all going on, there was a lot of promise to this regimen, so that's why it was used in this particular patient. Well, what I will say is that regimen on subset analysis, so what we often do in clinical trials is we go back and we look at the whole population of patients that was enrolled, and we look to see, is there anything we're missing? Is there any subset of patients that did actually particularly well on this regimen, even though the whole population did not? And patients with gallbladder cancer and patients who had non-metastatic disease actually did derive a benefit from this regimen. So it is still sometimes used in some places. I will say that it is more toxic, has a lot of side effects, a lot of bone marrow suppression, a lot of fatigue. So you really have to use it very judiciously. But it is an option. We used it here. The patient was restaged after 4 cycles. And you could see that the CAT scan, I'm telling you, the CAT scan actually demonstrated a nice response. The tumor went down to 6.2 centimeters. So that's a, 4 centimeter decrease. I mean, it was almost 10 centimeters when they presented. In the interim, to the point of what we've been saying here, early tumor sequencing. So this patient had profiling that showed what, a number of things, but what's actionable here that Dr. Harding alluded to is MTAP loss in their tumor. So not at this point going to change management or really do anything, but information that we want in our pockets for sure. So this patient went to the operating room. This is a really gross picture. Of the number that I had to offer, this is the least gross picture that they gave me, so we'll take it. But they had a extended left hepatectomy, so that means they had a large portion of the left liver removed. They also had lymph nodes taken out. They had some vasculature reconstructed. And the good news is, I mean, we don't know what, we saw the scans before they went to the operating room. We don't know really at a microscopic level what was happening here, but after surgery, after chemotherapy, you could see the lymph nodes were all negative for cancer. Patient recovered really well and was discharged on post-op day 9. So now that I've unfairly told you what happened and you didn't get to choose either of these things, what would you do next? Would you give this patient, quote, adjuvant therapy, which we learned from Dr. Winograd is systemic therapy after a surgery? And if you would, what would you give and for how long?
Yeah, I mean, I think by the book, right, this patient would not be somebody who would meet any of the criteria for the adjuvant trials that I presented. One of them, BILCAP, which was for patients who went for surgery first, right, and had completely the tumor removed, and then were assessed and either given treatment or not, and that showed a benefit. This patient obviously doesn't meet those criteria because they received 4 cycles of chemotherapy and immunotherapy before surgery. And so I think it's hard to extrapolate from that, what the benefit would be of adjuvant treatment. I would look at the fact that they had significant reduction of their tumor with the regimen that you did give them before surgery. And I would probably try, as long as they were recovered adequately and fit enough to tolerate it, another 4 cycles of Gem/Cis and durvalumab, probably without the Abraxane postoperatively, and then would weigh consideration of continuing durvalumab, with an informed discussion with the patient and tumor board.
I think I generally agree with that. I mean, you don't, Yeah, I don't have much more to add. I mean, I think that if, general principles when you're giving, I mean, in this case, I guess this would be considered a neoadjuvant, a true neoadjuvant approach, because it was resectable from the get-go, right? So, conversion therapy is generally when something is way too large to be removed and you're doing treatment to shrink it down in the hope that you could have a surgery. Neoadjuvant implies that it can be removed now and I'm giving chemotherapy up front. It's a subtle distinction. I think that it's totally reasonable if you have the tumor resected and you see pathologic response to resume a component of that after surgery, for a set period of time, if it was tolerable and recovered well. I don't think that's a wrong thing to do. There's less evidence to do it, as you noted, but I think that would be in the lines of what many medical oncologists might practice.
Yeah, so I'll just add that, this is a really, this is a gray area, very much. Go ahead.
Yeah, go ahead, please.
No.
Oh, yeah, I was just going to say, I mean, alternatively, sometimes if you've given enough therapy up front and you could just stop and watch, I wouldn't have a problem with that either. And I probably would have kind of just a discussion about that with the individual patient, the surgeon, maybe review with the pathologist to make that decision, as, again, it's not clear what to do.
Yeah, I mean, and to your point, I think, in spite of the NeoGAP trial that I mentioned, neoadjuvant therapy is not currently the standard of care. So patients that are eligible for surgery are taken to surgery right away, with the exception of what we call conversion therapy. So those are patients that are technically not resectable. And what actually happened in this case was, while the patient was technically resectable, this was more of a let's see the test of time, because we didn't know what was happening in those lymph nodes. So this was not a standard approach, and it happens more and more these days, and we don't often know what to do with patients now after they went to the operating room. The two things that both of you are hinting at that we have to guide us are, number one, how the patient did with the regimen before they went to the operating room, and how they are after the operating room, because it's not always the same person that went in and that comes out. And so can they tolerate the same regimen that you had them on now that they've had this very large surgery? The second thing is the pathology. There's a measure called tumor regression grade. I didn't include slides on it here because it's not standardly done everywhere, but it is a question you can ask the pathologist to actually score what the response was in the tissue to the systemic therapy that we gave the patient before surgery. And if there was little to no response to what you gave, it doesn't really make sense to give them any more of that. If there was a great response, you might want to do the same thing again if the patient can tolerate it. We tend to pick a chunk of time of 6 months just because that's what's been done over and over, and that was what's studied, so often we'll complete the rest of that. If the patient had no response to the treatment you gave them before surgery, you might want to start all over again and give them a full 6 months after if they can tolerate it, but it's a very nuanced discussion. So this patient had a response pathologically to that regimen. They completed 6 months. However, unfortunately, just over a year later, you can see that this patient had a recurrence. It was actually a multi-site recurrence, so the patient had multiple lesions in the liver, they had pulmonary nodules, so they were diagnosed with metastatic disease. And this is where I'd really like to have the bulk of our discussion. So I'll start with what would you guys do now? Would you go back on the same pre-op regimen because it worked before, and it was a period of time between when you gave it and when this happened again? Would you go to what's now sort of the standard of care, GEM-CIS with a checkpoint inhibitor? Would you incorporate MTAP-directed therapy, which we didn't get to dive into in a granular way, but you mentioned it as a really exciting biomarker that this patient actually has? I have their sequencing findings up there for you. Would you give it by itself with chemotherapy? And when would you incorporate a clinical trial? And how do you decide that with patients?
Yeah, so for my perspective, I always think about a clinical trial at every, point. And so, you have recurrence of the disease, and, that I would certainly think to that. From a standpoint of, the MTAP-directed therapy, so this is an investigational therapy. This is basically a target for which there are a number of molecules that are in development, and there's preclinical rationale to do something like that. They're though in the context of generally phase 1 studies, which is what you talked about, where it's looking at safety, and now they're maybe moving into phase 2, so trying to define better the efficacy of that. Many times such an approach might require that you've had the standard of care beforehand, which I think is reasonable, because with an investigational therapy, we're not 100% sure what's going to happen, even though it's a venue to get like a new treatment that may be helpful. So my probable vote here would be to go with more of a standard of care, and if the patient didn't have immunotherapy before, I would be keen to do that, given that it's extension of life, there's a maintenance approach with it, and so that's probably what I would do. I don't know, what are your thoughts about that?
Yeah, I mean, echoing a lot of the same points. I think for me, unless there was a trial that they were eligible in the first line, which, as you alluded to, MTAP targeted therapies are in early phase development. It's probably going to be some years before we see any trials that might think about incorporating those into first line therapy, right? The basic principle is you really want to give the patient the best chance at a regimen that's been shown to improve survival. Now, I think this question is answered differently if the patient recurs 3 months from completion of their adjuvant chemotherapy, right? If they got GemCis before and after surgery, and the recurrence is 3 months later, would I go back to a GemCis-based regimen? I'd probably be a lot less excited about that, because the notion would be that the tumor cells that are growing out are probably pretty resistant to that chemotherapy regimen, and I would probably be looking at this very differently. 14 months later, and it's a regimen that did have some activity at around the time of surgery, I would be inclined to go back to that with a combination of immunotherapy, which is the standard approach. And so assuming that there's no incorporation of additional agents on a trial for first-line therapy, I think that's the way I would go. And then you would resequence, right? That we talked about that as well. You have this MTAP loss from the initial mutation. I guess, is this a repeat biopsy or is this the initial one?
This is the initial.
Yeah, so I mean, I would be inclined, again, there's not, what we would call selective pressure significantly since that initial biopsy, and this one, meaning the patient had surgery, presumably the sequencing was done on that, and then they got a few more months of the same chemo as before, so you don't expect necessarily a change in the mutations in the tumor, but sometimes we can see things, so I would probably, offer the patient a repeat biopsy. Sometimes it's technically difficult. In this case, I think one of those liver lesions was pretty peripheral. That's going to be a relatively low-risk biopsy to do, and I would get that in order to kind of inform the next potential options for this patient. But here, probably GemCis with Durva or Pembro.
Yeah, so totally agree. This patient, although given the time of, to your point, 14 months later, there are many situations where we do what we call re-challenge with the same regimen, because there's been such a long period of time. We do consider the prior treatment with that chemotherapy successful, so we may want to use it again. But remember, what I said about this particular regimen was that the ultimate trials of this regimen were negative, except for small subsets of patients, of which this patient does not fit. So it begs the question of whether the toxicity we see with the triplet would offer the benefit in a very different setting now, right? We're not trying to get the patient to the operating room. We're trying to control the disease, keep things stable for as long as we can. So this patient did go on GemCis durvalumab. One thing to mention about difference between the TOPAZ and the KEYNOTE trials, the TOPAZ trial, the GemCis actually stopped after 6 months of treatment, and every patient continued on just the immune medicine. Whereas with the KEYNOTE trial with pembrolizumab, you could continue the gem. So a lot of difference on how, I don't know if you guys want to weigh in on how you feel about that, whether you continue the gem, whether you stop at 6 months. You know, I would say, I think for many patients after 6 months of this triplet regimen, they're pretty tired of the chemo, but what are you all doing in that situation?
I usually make it a shared decision-making model, you know, to think about it. I mean, you know, if you look at the studies, although you really can't compare them to one another, the way that experiment was done, very similar in terms of the outcomes. Maybe the only difference was for Keynote 966, the duration of response was longer, and you know that might lead you to say, well, maybe I should continue the chemo gemcitabine a little longer. I don't think we can really know that, and I would make it shared decision. So if, for example, if, you know, as you said, it's been 6 months and there's problems with the bone marrow, fatigue, you know, people are tired and they've had, you know, response or control, I would really offer the single agent immunotherapy. I think that would be very reasonable. I think on the other hand, you know, you come to some instances where these tumors continue to shrink even at 6 months, and somebody is very fit and really not having as much toxicity. Removing the cisplatin is, you know, preserving, I think, the bone marrow, and you can continue on the gem. I might do that. I think that it's really a kind of shared decision. Some people are uncomfortable with stopping chemotherapy. I wouldn't push cisplatin like a lot. That's always hard to do, and it can have long-term toxicities that I think we could always resume it at a later point or re-challenge with a platinum at some point.
Yeah, I mean, I agree with all those points, so that, you know, those decisions are ultimately individualized to each individual patient and are made in discussion with the patient. I'll say my practice really has been to stop. I think 6 months of chemo is a lot between the bone marrow and the fatigue and the side effects. And the reason I do that is often because then it does allow me to re-challenge with GemCis, and I've done that successfully a number of times in patients with this regimen, where you see initially a very nice response that kind of seems to be plateauing towards that 6-month mark. You transition to immunotherapy alone. You get a couple more months mileage out of that, and at some point we see some progression again. The patient's marrow has recovered, they're feeling better, they've been coming to clinic a lot less, once a month for durvalumab, and I'll re-challenge with GemCis at that point, getting back on the original regimen. If insurance allows. I've run into one time where they were absolutely against that. But and I've had patients go back and have responses again with reintroduction of the chemo, and I find that that's been a productive way to extend the duration of the first-line regimen.
I swear I didn't prep them. This is exactly how I do things. And so this patient, a bit older, was running into marrow toxicity issues, particularly platelets. So we did the GemCis Durva for 6 months, and then we took out the GemCis. Patient continued on single agent Durva for some time, and actually just recently started to show some signs of the cancer growing again. So we were preparing to re-add. I often try to just re-add the gemcitabine and see if I can get away with that. I mean, before adding both. Doesn't always work. Sometimes it does. But actually yesterday we were just informed that they had a slot awarded to them on an MTAP-directed clinical trial. So that's what we're going to do.
I think that would be reasonable, yeah. I mean, I think that you know, the time of progression on this, I really do think it's moving towards the molecular targeting and clinical trials, but I do, you know, think that you know, cytotoxic chemotherapies can still be helpful, and you know, that's always I think a possibility. I think in certain instances, and we don't have the whole multidisciplinary team here, depending on the extent or the location of the recurrence, you might consider other things. Maybe not in this case, where it seems that there were multiple sites of tumor progression, but you know, some people might have one tumor grow, and perhaps there could be some local therapy related to that, and so that you have to be mindful of that as well.
Great point. All right, so phase 1 to hopefully with the win, to bring us full circle. All right, so now any questions for the audience. This is the last Q&A of the meeting today. Everyone's tired and thinking about their grocery list. My father told me to buy protein bars. I'm like, what am I going to do with protein bars in the storm? But can I eat real food? Yeah, right?
Hi, my question is, reading a single patient case study when he had progression of disease on durvalumab alone, and then switching him to pembro and having an incredible response. Is this routinely done? Would you ever consider this, even if you were to put the gem back on? Do we see any benefit in switching the immunotherapies?
Yeah, I mean, so Durva and Pembro are pretty similar molecules, right? There's a little bit of a nuance as far as a PD-1 versus a PD-L1 binding. So, to just talk about that for a second. So PD-1 is a receptor expressed on activated T cells, whereas PD-L1 is a receptor that binds PD-1 that's expressed on normal cells in the body, but also overexpressed by tumor cells. And when PD-L1 is overexpressed on tumor cells, it shuts down the effect of PD-1 expressing T cells. And by interrupting that interaction, you're allowing the T cells that are activated and coming into the tumor to no longer be getting suppressed by the tumor and to kill the tumor cells. That's kind of the basic biology of that. And so theoretically, blocking either side of that interface should have similar effects, with the small caveat of that there are some other ligands for PD-1, specifically PD-L2, that in some tumor types seem to be upregulated as well. And so biologically speaking, blocking PD-1, such as Pembro does and nivolumab and some other similar compounds, may be theoretically slightly better than blocking PD-L1. I can't tell you that I've seen ever a patient who is progressing on a PD-L1 then having a response to the PD-1. It speaks to potentially something else biologically happening, whether there was a radiation or some other event, or even an infection, right? I mean, there's been case reports of patients having certain infections that are causing systemic inflammatory changes that somehow are potentiating the effects of immunotherapy. So positing with absolutely no data other than what you gave me, I think in general we think about these as pretty interchangeable compounds.
Yeah, I agree. I've never seen that happen, but
If I'm understanding this correctly, the first line of defense is basically you're using the drugs for the chemo and immunology, the systematic approach. For the second line of defense, or when would you look to do the Y90 or the PUC? Like, at what point do you make that decision?
Yeah, I think that's an interesting, it's a good question. I think in that space, it really depends to me, again, on the individual characteristics of the tumor, right? And I think the time course of the treatment and how someone is doing. Regional therapies are generally, a lot of devices and regional therapy is basically used on small, like single-arm trials that are not necessarily randomized, and to know their full benefit is still challenging. And we saw today there are many randomized studies going on, and so in the absence of trials to kind of make clarity on what to do, many centers will have external beam radiation that they can offer, Y90, pump. There are a whole variety of them. I think in that instance, it's not necessarily that may be the pathway. It's rather, what does the disease look like and what are we trying to achieve at that time? That's how I perceive that. For example, if somebody had liver-limited liver cancer that didn't necessarily have a mutational driver, bile duct cancer, I apologize, it's all in the liver. They didn't necessarily have, any potential trial or driver. I mean, I would review that with the tumor board and ask the question, is there a regional approach that could be used in the setting? Sometimes there really isn't, if there's a localized tumor in one area, maybe that's something you could do. But if there are multiple tumors throughout the liver, it's hard to rationalize how that treatment would be helpful, with the exception maybe of pump. I don't know. But I think it's still somewhat investigational, on my view. I don't know what you guys think.
Yeah, I mean, I think, it's so patient-specific, and I think this is one of the things Dr. Spencer was alluding to, that, these are decisions that, we don't make on our own either, right? Meaning, going into a tumor board, I may have a notion of what I think it might be best for the patient, but in discussion with my colleagues who have expertise in giving the Y90 or with our surgeons, my mind can be changed, right? And I think that's important for all of us to go into these discussions with, an open mind to make sure we come out with the group's recommendation, incorporating everybody's expertise as to what we think is going to best help that patient, right? My job in that meeting is to advocate for the patient, know their data, know their history, and be able to present that clearly. But ultimately, these decisions are very nuanced. And so, we like to consider the use of local regional therapies to lump all these things together when biologically it makes sense. And so, sometimes it's liver-only disease, but sometimes occasionally you'll see a single area growing in the lung, right? Meaning the tumor in the liver is being controlled on whatever current treatment you're doing, or there's no tumor in the liver and there's a single recurrence somewhere else. And can you radiate that? Can you ablate that, right? And there are ways to do that outside the liver as well that can be quite effective for targeting that specific tumor. But ultimately, you have to view that in the context of the overall biology of that patient's tumor, right? If they've been off treatment for a year and a half and there's one side of recurrence, that's a reasonable thing to think about, right? Ultimately, my worry as a medical oncologist is that's the tip of the iceberg and we're going to see more things. But again, in multidisciplinary discussions, we can have those discussions about whether that's a reasonable approach and present those options to the patient. So there's no single answer for your question, but it's a really important one.
Thank you for that. And my last question was about the hepatic artery. I didn't hear enough talk about that, if a tumor is strangling it a little bit. Like, how do you keep that alive and healthy and the blood flow, I guess, going?
Yeah, I mean, the liver is perfused by two things, the portal vein, which does 80% of the blood flow, and the hepatic artery, which is 20%. And when there are tumors that are in the liver, perhaps some of them preferentially will take their blood supply from the hepatic artery. And so you can use the hepatic artery as a means to deliver, regional therapy with Y90. You can use that to do pump-based approach. I think the question there is there a way to preserve like the highway of all those important structures going into the liver? There may be, regional therapies like radiation that can sometimes be used in those settings. But I think that, it really depends on the context of an individual person.
Yeah. All right, let's go ahead.
Do you elaborate a little more on the usefulness of circulating tumor DNA, both in the conventional treatments and clinical trials?
Sure. I mean, I think that we know that tumors will shed their DNA into the blood, and that you can detect that. But there are a number of ways you can detect that. One type of way is, which is called tumor-informed, where you understand some of the genes that cause the cancer to grow, and then you use assays to look at a number of those in the blood, and those genes are thought to not change very much. And those might be called minimal residual disease assays. So what they're looking for is the very slightest amount of DNA from the cancer in the blood. And there are a number of companies and individual institutions that have developed these. And probably across cancers, but more so in colon than any other, what you can tell is that if there is no, circulating tumor DNA in the blood, let's say after a surgery, perhaps the outcome may be better. And likewise, if there is, perhaps there's a greater risk of recurrence. And so it may be having a prognostic effect, right? there's something like called the harbinger's dilemma, in that you might know that something is worse, but you're not sure what to do about it. And that's kind of where we are, I think, with cell-free DNA right now. That at early stage with MDR-based assays, if it is positive, it's not clear what to do, and there are ongoing trials to see if you should add chemotherapy to it, or you should do some treatment. So I would say that detection of cell-free DNA can be done. It may have some value in other more, common cancers. It probably has some value here, but to do something based upon that or not, I don't think we know where we're at. Another form of cell-free DNA is it's looking for the mutational profile of cancer. And, this is helping us in that, sometimes, as I told you, it's like a whole mix of tissue. You do a biopsy, you try to do some genetic analysis, you're not able to, but you might be able to detect mutations in the blood. And using this type of assay, it's complementary, and that could guide you to a targeted therapy. The question about sequentially following that with treatment, I think still, too, is unanswered, because we know that resistance mutations develop, but we don't yet know if they do develop, can you give something else that would get rid of them? And so I think that, in an investigational way, you can continue to follow these, but I'm not yet 100% sure how it would dictate the treatment plan at this point. For FGFR inhibitors, there's specific mutations that will develop, and that may suggest you can use a different FGFR inhibitor, and that might be good to know, but it hasn't been like a proven, it's not been truly proven in prospective data. And so sometimes insurers will not pay for that based on that. But I think that, I do that. I will periodically check cell-free DNA, but it's in the auspices of research, funding from the government and like philanthropic organizations.
All right, let's just do one last question since we're way over time. Go ahead.
Yeah, just going back, sorry we didn't have questions in between your first presentation, but to the FDA-approved drugs. So, for example, like the FGFR2, like how do you decide which one would be better? So, like the futibatinib or the pentajavanib, probably botch them, but and then once you decide, like since they're taking the place of they're the second line, like when, like you were saying, you start to like add things back, like do you add the immunotherapy back, or like what is no, you won't add
Yeah, no, it's a great question, right? And so when there are multiple agents approved, right, and we have a little bit of this in the first line as far as Pembro versus Durva, there we think they're probably pretty equivalent, and it's really about whether you want to continue that gemcitabine beyond 8 cycles. That probably is going to dictate your choice. With some of these agents, there are going to be slightly different toxicity profiles, right? And so that's usually the thing that's going to drive the decision. So you kind of review what are the most likely side effects with your patients for these individual medications. Often when their medicine's targeting the same target, the side effect profile is going to be quite similar, but based on each individual kind of molecule's biology and chemistry, really, you're going to see some potentially slightly different patterns of side effects. And so based on your patient's prior medical history, their medical conditions, the things that are priorities for them, you might pick one or the other in consultation with your patient. Occasionally, insurance is going to dictate. Some insurances will say, we're only going to pay for this one because we struck a deal with the company that makes that one, so sometimes your hands are tied. But if you're asking from a medical standpoint, that's usually what's going to drive that. As far as your second question, would you add something to that? Again, not outside the context of a clinical trial, right? Because we know based on phase 1, 2, and actually really just phase 1 and 2 trials with some of these targeted therapies, what the safety is of those medicines when given in patients, right? And they have side effects, they really do. We don't know necessarily what the safety is of combining those medicines with immunotherapies, right? And while we might postulate, based on our understanding of the medicines, that actually they probably would be tolerable together, and maybe we can come up with some biological reason why we think they might work together, you actually don't know that, right? And there's been plenty of times where we've had good ideas, we being the field, has had good ideas that based on preclinical data look good, that based on phase 1 data seem okay, and then they come to larger trials, And not only are we not helping patients, potentially we're harming patients, right? And trials are designed to have interval assessments for that, right? To make sure that if patients are potentially not benefiting or God forbid, being harmed by the study intervention, that studies are stopped early. But doing so outside of a trial runs that risk, right? And so I think all of us in our daily practice are doing things that are not quite within the bounds of what the trial would have prescribed, because the real world is not the world of the trial world that the patients were enrolled on. But I would not certainly combine, immunotherapy with an FGFR inhibitor, just based on the fact that we don't know what the safety of that is.
I think it's hard to know whether Foodie or PEMI, which is the best choice. I mean, there's biochemical differences. Both of those were tested on single-arm studies, and with 100+ patients, and the response rates are similar. Futibatinib was a little higher, pemigatinib was a little lower, but it's hard to make a decision like that because of the number of patients treated. Often, I will talk afterward because I'm going to go on, so I won't. We can talk
Yes,
afterward,
unfortunately, no, we were going to have to end. We're like 40 minutes over, but you guys are, the medical oncologist will stay behind and answer any of your questions offline. So let's give them a round of applause one more time. Even though you can barely see the green in his tie, and I don't know, they're handsome, but I don't know how tall compared to Dr. Halazoun, but we'll do a measuring contest next time. So thank you so much for all your insights, and so wonderful to have you here tonight. So Melinda, you're on the agenda to do closing with me. Did you have closing remarks?
I have one remark that somebody left their bag out at lunch, but there was a pair of sunglasses. So if they're anybody's, I don't want to take them home. And two, I just wanted to thank you all for coming out and for all the amazing questions that you offered to our panel. And three, thank you, Dr. Spencer, for being such a wonderful host and doing this with us. And we look forward to having another one next year, and we'll figure that out. Dr. Harding, thank you for coming over from MSK and for the great presentation, as well as all the presentations were wonderful, even from the surgeons. So again, thank you all. We appreciate you.
And I would just add that I think, days like today remind us that, while this is really a challenge for patients and providers, we're not standing still. We're making progress, and New York City is the heart of innovation. So this is where we need to keep working together to generate ideas and to move the field forward. And I'm so thankful for my colleagues who are all very collaborative, brilliant, tall, handsome people. Thank you so much for all of you for being here. And again, the patients, you are at the heart of this for all of us. Thank you so much for your time. Be safe this weekend. Go get your protein bars. All right.